Good morning, ladies and gentlemen. A very warm welcome to the National Symposium on Good Laboratory Practice GLP 2026, organized by the Department of Standards Malaysia. The theme of this year's symposium is Strengthening Trust, Ensuring Quality, Advancing Sustainability. We are delighted to have all of you with us today, both physically here at the Glass House, Paskrandi Hotel, Putrajaya, and those joining us through the online platform. To officially begin our program this morning, may I invite Encik Irwan Tahir to lead us in the recitation of the Doa. Please welcome.
We start our prayer with the recitation of the Fatiha. Amin amin bismillahirrahmanirrahim. Alhamdulillahi rabbil alamin. Wassalatu wassalamu ala ashrafil anbiya'i wal mursalin wa ala alihi wa sahbihi ajma'in. In the name of Allah, the most gracious, the most merciful, all praise is due to Allah, the Lord of the worlds. Peace and blessings be upon the noblest of prophets and messengers, our Prophet Muhammad, and upon his family and all his companions. Ya Allah, Ya Rahman, Ya Rahim, we gathered here this morning with hearts full of gratitude, thanking you for the blessings of health, time, and opportunity to convene at this National Symposium on Good Lab Practice 2026. We seek your divine grace and blessings upon this gathering held here at Hotel Pas Grande, Putrajaya. Ya Allah, Ya Alim, Ya Hakim, we ask that you illuminate our mind and grant us beneficial knowledge as we strive to realize our themes today, strengthening trust, ensuring quality, advancing sustainability. We ask for your guidance, bestow upon us the integrity, precision, and dedication required in all our lab practice. Make our work source of safety, health, and prosperity for the Ummah, nations, and of course for the world. Ya Allah, Ya Hafiz, protect us from any shortcoming and ease our affairs throughout the symposium. Unite our hearts in purpose, forgive our mistakes, and accept our share efforts as righteous deeds performed for your sake. Grant us a successful event from this morning until its end, and grant us a safe journey home thereafter. ربنا آتنا في الدنيا حسنة وفي الآخرة حسنة وقنا عذاب النار وصلى الله على سيدنا محمد وعلى آله وصحبه وسلم سبحان ربك رب العزة عما يصفون وسلام على المرسلين والحمد لله رب العالمين.
Thank you to Encik Irwan Tahir for the prayer. Ladies and gentlemen, may I kindly invite everyone to stand as we proceed with the singing of our national anthem, Negaraku. Thank you, ladies and gentlemen. Please be seated. Ladies and gentlemen, it is now my pleasure to invite Encik Warukiman Wan Mamat Director of Accreditation Division, Department of Standards Malaysia, to deliver the opening remarks and officially welcome us to the National Symposium on Good Laboratory Practice GLP 2026. Ladies and gentlemen, please join me in welcoming Encik Warukiman Wan Maman. Encik Warukiman, the floor is yours.
Macam mana? Tak bunyi. Bismillahirrahmanirrahim. Terima kasih pengacara majlis yang berusaha, Puan Fadilah Hasbullah, Ketua Seksyen Good Clinical Practice and Good Laboratory Practice bahagian Regulatory Farmasi Negara, yang berusaha Encik Baharudin Basri, Pegawai Pertanian Jabatan Pertanian Malaysia, yang berusaha Encik Tim Ching Min, Test Facility Manager ALS Technicam Malaysia, yang berusaha Dr. Ahmad Razlan Rosli, Residue and Human Safety Scientist, Bayer Crop Science, yang berusaha TS Hasnizawati Muhammad Dahlan, Deputy Test Facility Manager, Makmal Biosasi Alaf UKM, Miss Laina Iwa, Principal Administrator OECD Good Laboratory Practice, Dr. AM Hessin, Specialist Inspector, Netherlands, Mr. Mark Ubrana, Chief Specialist Department for Good Laboratory Practice, Poland. Tuan-tuan dan puan-puan sekalian, Assalamualaikum warahmatullahi wabarakatuh dan salam sejahtera. Alhamdulillah, marilah kita sama-sama memanjatkan kesyukuran kita kepada Allah Subhanahu Wa Taala kerana dengan izinnya kita dapat bersama-sama pada hari ini dalam National Symposium on Good Laboratory Practice 2026. Bagi pihak Jabatan Standard Malaysia, saya ingin mengalu-alukan kehadiran semua peserta dan mengucapkan terima kasih atas komitmen dan sokongan yang diberikan kepada penganjuran program ini. Saya dimaklumkan bahawa seramai 94 peserta hadir secara fizikal, manakala 42 peserta telah berdaftar secara dalam talian dan akan mengikuti simposium ini melalui YouTube GSM. Kehadiran ini amat membanggakan dan jelas menunjukkan terdapat minat serta keperluan yang semakin meningkat terhadap pemahaman dan pelaksanaan good laboratory practice di Malaysia. Pada kesempatan ini, saya merakamkan penghargaan kepada 3 organisasi pempamer yang turut sama-sama menjayakan program ini, iaitu Malaysian Institute of Pharmaceutical and Nutraceutical, National Institute of Biotechnology Malaysia, Makmal Biosasi Center for Natural and Physical Laboratory Management, ALAF UKM, dan Rakit Malaysia Sdn Bhd. Penglibatan pihak industri dan organisasi berkaitan seperti ini amat penting dalam mewujudkan ekosistem GLP yang lebih dinamik serta menggalakkan perkongsian pengalaman dan amalan terbaik. Tuan-tuan dan puan-puan, Jabatan Standard Malaysia atau JSM merupakan sebuah agensi di bawah Kementerian Pelaburan, Perdagangan dan Industri (MITI) yang ditubuhkan di bawah Akta Standard Malaysia 1996. Selaras dengan fungsi yang diperuntukkan di bawah akta tersebut, JSM berperanan sebagai badan standard kebangsaan dan badan akreditasi nasional yang bertanggungjawab terhadap pembangunan standard serta pelaksanaan aktiviti akreditasi badan penilaian pematuhan. Dalam konteks GLP, GSM merupakan satu lagi peranan yang amat penting, iaitu sebagai Compliant Monitoring Authority, CMA, yang bertanggungjawab memantau pematuhan test fasiliti terhadap prinsip OECD GLP. Pelaksanaan prinsip OECD GLP amat penting bagi memastikan kajian bukan klinikal yang dijalankan menghasilkan data yang berkualiti, boleh dipercayai dan mempunyai integriti. Seterusnya boleh diterima oleh pihak berkuasa, kawasan di peringkat kebangsaan dan antarabangsa. Kepentingan GLP tidak terhad kepada satu sektor sahaja. Data daripada kajian bukan klinikal yang dilaksanakan mengikut prinsip OECD GLP menyokong proses penilaian keselamatan dan keberkesanan pelbagai produk termasuk produk farmasetikal, racun makhluk perosak, bahan kimia industri, bahan bioteknologi serta produk kesihatan. Oleh itu, kejayaan pelaksanaan GLP bukanlah tanggungjawab GSN semata-mata. Ia memerlukan kerjasama dan komitmen semua pihak kepentingan, termasuk regulatory authorities, test facilities, syarikat penaja kajian GLP, pihak industri serta organisasi yang menjalankan kajian bukan klinikal. Tuan-tuan dan puan-puan, kita juga sedang berhadapan dengan perubahan yang begitu pantas dalam lanskap sains, teknologi dan regulatory. Pendigitalan sistem makmal, penggunaan sistem komputer, kecerdasan buatan, peningkatan keperluan terhadap integriti data serta perubahan dalam keperluan regulatory di peringkat global menuntut kita untuk sentiasa meningkatkan pengetahuan dan keupayaan. Dalam keadaan ini, persoalan yang kita perlu fikirkan bukan sekadar sama ada sesuatu kajian mematuhi GLP, tetapi juga sejauh mana kita mampu memastikan bahawa data yang dihasilkan benar-benar boleh dipercayai, boleh dikesan, mempunyai integriti dan boleh dipertahankan apabila dinilai oleh pihak berkuasa kawal selia. Atas keperluan inilah GSM mengambil inisiatif untuk menganjurkan simposium ini. Program ini merupakan julung kali dianjurkan oleh GSM. Kami melihatnya sebagai satu platform penting untuk menghimpunkan tuan-puan semua pihak berkepentingan dalam ekosistem GLP. Melalui simposium ini, kita bukan sahaja ingin berkongsi maklumat, tetapi lebih penting kita ingin mewujudkan dialog, persemahaman dan kerjasama strategi antara semua pihak yang terlibat. Antara objektif utama ini adalah untuk menyediakan platform bagi membincangkan isu-isu semasa dan cabaran pelaksanaan prinsip OECD GLP, mengharmonikan kefahaman mengenai keperluan GLP serta berkongsi perkembangan terkini di peringkat antarabangsa. Kita juga berpeluang mendapatkan perkongsian daripada pakar OECD GLP, khususnya berkaitan perkembangan semasa, integriti data, sistem komputer dan pengurusan test item. Saya percaya perkongsian seperti ini amat bernilai kerana ia memberikan kita perspektif yang lebih luas mengenai bagaimana GLP sedang berkembang di peringkat global dan bagaimana kita boleh memastikan pelaksanaan GLP di Malaysia sentiasa relevan dan bersedia menghadapi perubahan masa depan. Selain itu, simposium ini juga bertujuan untuk memperkukuhkan hubungan GSM dengan pihak berkuasa, kawasan industri dan test facilities. Saya berharap sesi ini telah disusun pada hari ini bukan sekadar menjadi satu pembentangan satu hala, tetapi dapat dimanfaatkan sebagai platform untuk bertanya, berbincang, berkongsi pengalaman dan mengenal pasti penyelesaian bersama. Tuan-tuan dan puan-puan, sebagai suatu program yang julung kali dianjurkan, GSM melihat simposium ini bukan sebagai satu platform yang berakhir pada hari ini saja. Sebaliknya, ia adalah permulaan kepada satu platform libat urus GLP yang lebih berterusan. GSM berhasrat untuk memperbanyakkan lagi program berkaitan GLP pada masa akan datang dengan kerjasama semua pihak yang berkepentingan dalam ekosistem GLP di Malaysia. Saya percaya melalui kerjasama yang kukuh dan berterusan, kita dapat membina ekosistem GLP yang lebih mantap, kompetitif dan diyakini. Akhir kata, harapan saya agar usaha kita dalam memperkukuhkan pelaksanaan GLP di Malaysia dapat diteruskan dengan lebih agresif dan tersusum. Semoga pada masa akan datang, kita dapat melihat lebih banyak test facilities yang mampu memenuhi keperluan OECD GLP, lebih banyak kajian bukan klinikal yang berkualiti tinggi dijalankan di Malaysia, serta lebih banyak data kajian yang mempunyai integriti boleh dipercayai dan diterima di peringkat antarabangsa. Saya berharap agar Malaysia bukan sahaja menjadi negara yang melaksanakan GLP, tetapi mampu membangunkan ekosistem GLP yang kukuh dan berdaya saing yang menyokong pembangunan produk, inovasi, penyelidikan serta kepentingan keselamatan dan kesihatan masyarakat. Akhir sekali, saya merakamkan penghargaan kepada semua pihak yang telah memberi sokongan dan kerjasama dalam menjayakan simposium ini. Kepada penceramah dan pakar yang sudi berkongsi ilmu dan pengalaman, para pempamer, pihak berkuasa, kawaselia, test fasiliti, pihak industri serta peserta yang hadir secara fizikal dan dalam talian. Terima kasih atas komitmen dan sokongan semua. Dengan lafaz Bismillahirrahmanirrahim, saya dengan sukacitanya merasmikan National Symposium on Good Laboratory Practice GLP 2026. Sekian, wassalamualaikum warahmatullahi wabarakatuh.
Thank you, Encik Waluliman, for the inspiring opening remarks and for officially setting the tone for today's symposium. Ladies and gentlemen, before we proceed with our technical session, we would like to invite all our distinguished speakers, invited guests, organizers, and participants to join us for a group photograph. May I kindly invite everyone to proceed to the designated area for the group photograph.
Sini je kan? Center je kan? Ini microphone. Aktivitas ni termasuk kan? Ini kita penuhi itu. Oh, why? Maka tak ada evidence dah datang. Kita akan hantar ke PF bagi dia. Kita letak tangan dulu. Depan.
Dah, kat duduk ni.
Cik Mama dah sampai ke?
Itu sini, itu kain.
Kejap sini.
Yang kosong, boleh tempat kosong ni. Ini ada tempat yang kosong.
Ya.
Tak ada di kampung.
Apa yang depan lagi? Yang kat tengah tu boleh depan. Alright. Kita pergi. Berais.
Okey. Yang ini boleh kita dengar sikit.
Ada di sana. Baik kat sini. Orang online dengar. Apa?
Thank you, everyone. Please return to your seats as we continue with the next session. Ladies and gentlemen, we shall now proceed with our first session of the day. Our first presentation will provide an update on the GLP compliance program of the Department of Standards Malaysia. It is my pleasure to introduce our first speaker, Puan Sharifah Azlinda Syed Abu Bakar, Head of GLP and Filas Unit. She will be sharing an update on the GLP compliance program of the JSM, including the current program and its implementation. Ladies and gentlemen, please join me in welcoming Puan Sharifah Azlinda Syed Abu Bakar.
Assalamualaikum warahmatullahi wabarakatuh. Can you see me now? The rostrum actually is too high for me. Okay. Assalamualaikum and good morning, everyone. First of all, thank you to the Ms. Najiha, our MC today. I will proceed to my presentations. I will update about the DSM GLP compliance program. Okay, the next one. Can you provide me so that I can control myself? Yeah, which one?
So press that button, okay?
This one.
Yeah. Just follow the arrows.
So for those first time heard about the DSM, okay, we are the Department of Standards Malaysia. It's a national standard body and also the national accreditation body governed by the Standards of Malaysia Act 1996 at 549. Okay, we are under preview of Ministry of Investment, Trade and Industry. I heard actually certain people still think that we are still under MOSI. Actually, since 2018, we already put under VP. Okay, no more under MOSI, although we are the science officer. Okay, so the next one. Okay, this one is actually, I think I need to take out this one. Okay, this one actually our two main role as a national standard body. As a national standard body and also national accreditation body of Malaysia. So in Malaysia, we only have one national standard and also national accreditation body. Okay, this one is our governance structure for the first timer known about DSM. Okay, we are the under the MITI 1 year. Sorry, the laser pointer is not function. Okay, so this one is under MITI, and then we also have the Malaysian Standard and Accreditation Council, call it MASDAM. And then we have two national committee under preview of MASDAM, which is National Standard Committee, focusing on the discussing about the standard development, and then pertaining the accreditation activities, we are reporting to the National Accreditation Committee, NAC. Okay, this one, DSM top management. I'm not sure whether all of you know about our new DG. Okay, we got the new DG starting, I think, September or August last year. We got Mr. C. Chi Kong, okay, our new DG. And then we have 4 director, which is the first Mr. Mirza Hussein for the Senior Director of Standardization, followed by Mr. Wanukiman Wan Mamat. We just meet him, okay, Director of Accreditation. And then the Madam Nur Latifah Hussein, Director of Strategy Planning. And the last one is Encik Izzat Abdullah, Director of Management Services. Okay, this one, under the accreditation divisions, we have we actually just restructure our divisions. So we got the GLP and Philips unit. This one under my preview starting February this year. And then we have an inspection body unit, PTP, RNP, then certification body. We have management system and halal, certification body unit pertaining for person, product, and ADB. We have medical testing and biobank unit, calibration, mechanical and electrical laboratories unit, testing laboratories unit. So if your laboratory got the accreditation for 17025 is under these two units, calibration, mechanical, electrical laboratories, testing laboratories unit. We also have the assessment coordination unit, accreditation liaison unit, competency unit, QMS unit. We have a boss of the competency unit here, which is Puan Aina. Okay, this one's the DSM GLP inspector. We have a Puan Fariza, okay, lead inspector and also OECD evaluator. We have Puan Rohas Miza, GLP officer, lead inspector, OECD evaluator. And then we have a Puan Siti Rohaya, inspector, me myself as a GLP officer and also lead inspector. And then we have Prof. Dr. Nurfadila Raja is a lead inspector. We also have Dr. Jahangir Kamaldin as a lead inspector. And then we have a newcomer, which is Encik Irwan, is our training inspector. Okay, we are hoping next year we are going to train a few inspectors so that we can enhance our pool of inspector. Okay, this one's the DSM GLPCP infrastructure as of 1st July this year, where currently we have 5 lead inspector, 1 inspector, and 1 training inspector. Then pertaining the test facility, we have 14 compliant test facilities and we have 1 applicant. And then the total currently involved in the GLP inspection activities from DSM, we have 5 permanent staff. This one actually is the hierarchy of the GLP-CP documentation under DSM. Okay, firstly, of course, we are referred to the OECD series of documents. We have 25 essential documents covering GLP principle, data integrity, computerized system. So total, there's a 25 OECD GLP documents. And then we also refer to the OECD publication and guidance, including the test guideline. And then the internal one, the tier 3, we have referring also to the DSM GLP-CP circulars. So we have internally also have our own circular. And then we also come out our GLP-CP manual and appendix. So for the new applicant who is planning to apply with DSM, so I really appreciate if you can download this GLP-CP manual. You can get it from the DSM website. I can show it to you after this. Okay, this is a DSM website. Can Irwan, where are you, Irwan? Can you share the screen for the DSM website? Okay, because I believe all of you actually is not familiar with the DSM website, especially in getting information about the GLP. So it's good if you can see it through the live showing here. Okay, you can go to the accreditation, okay, www.gsm.gov.my. You go to the accreditasi or accreditation, okay, and then you can go to the, okay, you go to the, this one, the program, you can go to the GLPCP, you can see this one, actually the field that, can you scroll down? Okay, this one. Can you change it to English, please? Sorry. Okay, this one actually, okay, feel we as DSM as CMA, Compliance Monitoring Authority, the field that cover under DSM is industrial chemical, feed additive, pesticide, biotechnology, non-pharmaceutical, and also others item which is regarding the industrial chemical related to chemical is under DSM. Okay, so in Malaysia we have a 2 CMA currently, which is DSM and also NPRA. So our field is different. The next one, you can go to the resources. Okay, this one, the pyramid that I showed it to you. This one, the OECD GLP, a series of document, OECD publication, okay, and then the guidance document, the circular, GLP-CP manual, and appendix. Okay, you can get all the information here. Okay, so you can download it. Actually, all these OECD document, you can also access it through the direct to the OECD website. Okay, that's it. I hope you understand and can easier access this DSM website. Can we go back to my slide, please? Okay, this one, OECD document number 1, I just write up is the summary about the OECD document number 1. Okay, I believe all of you already familiar with all the OECD document number 1 principle. Okay, because there's I think most of the tax facilities under DSM actually is attended here. Thank you so much for your attendance. Okay, this one, the categorization, the active OECD series, based on the category, as I categorize it into admin and inspection, role and responsibility, data integrity, and IT, specific study type, alignment, and material. So total, there's a 25 document OECD. Okay, this one, the circular, is a GLP-CP circular number 1, 2021. This one is a guideline for remote inspection. This one, we are not withdraw it yet because we are not sure whether this remote assessment is still needed or not. So it depends on the situation. If the remote assessment is required, so we will do remote. That's the reason why we are not withdraw this circular yet. Okay, the local engine, this one is our GLP-CP manual, published on the 13 March 2023. Okay, this one, the table of content of the manual, you can see here, there's a process of the GLP-CP conduct by the GSM, and then we have a lot of the appendix here. Okay, easier for our applicant or also existing test facility to use as a reference. Okay, for the organizational structure, you can go to the appendix 1 and also appendix 2. Is pertaining the inspector integrity, you can go to the appendix 4, appendix 5, and then pertaining the framework of the OECD GLP-CP program, you can go to the appendix 3, explaining about the inspection process, appendix C, list of evidence document, appendix 7, evaluation report, appendix 8, master register GLP-CP. Okay, and then pertaining this one also the inspection life cycle toolkit available in our website. Okay, you can see here from the starting from the initiation, pre-inspection, the audit until you got the certification process, which is your compliance GLP status. So it's all explained in an appendix mentioned here. Okay. For your information, DSM already have our own system, which is e-accreditation system. So all the appendix 1 that I mentioned just now actually as a reference, but all the information, all the report actually already embedded in the system. Okay, so throughout the inspection process, all the appendix actually already embedded in the e-accreditation system. So this one, the GLP inspection process flow chart, I believe the process is similar. Okay, I believe all of you already familiar with this inspection process. Starting from the application, pre-inspection, inspection, which is study audit, recommendation of GLP compliance by our DG, by the DA dulu, which is the Director of Accreditation, followed by our DG, and then you will got the GLP compliance status, then followed by the surveillance. Okay, this one that I mentioned to you, our new system is a e-accreditation system. We are in the process of the upgrade this system so that all our user can easily access all these informations and also easy to implement your inspection process. Okay, in terms of fee, this information actually is available in the DSM website. Okay, if you want to know for the new applicant, if you are interested, you can go to DSM website to see how much that you need to pay for the application fee, for the surveillance. So each inspection, there's a fee. Okay. This one, the summary of the GLP inspection conducted by DSM starting from 2020 until 2026. Okay. 2026, we have total 10 inspection conducted by DSM inspector. So most of them, actually the highest one, of course, is the 2024. We have 11 inspection. Okay, this one actually is a summary from all the findings received during the inspections. Okay, normally it's for pertaining to the test facility organization and personnel. The common finding is pertaining the training, competency, the JD, job descriptions, the responsibility, and also the study director roles. And then pertaining the QAP, quality assurance program, normally pertaining the QA inspection plan, the documentation, QA statement, whether this QA independence or not, and then the follow-up of the finding of the QA inspections. All of this is a common finding that normally received by the test facility. Okay, and then pertaining the third one is the equipment, material, and reagent. This one, number 1, number 2, number 3 is actually is the highest common finding received. Okay, pertaining the calibration, whether the equipment has been calibrated or not. In terms of the maintenance, the identification, traceability, and also suitability. And then the SOP, of course, the outdated SOP, incomplete procedure, then there is no version control and also deviation from the SOPs, which is you are not identified with the current and also the obsolete documents. And then the third one, pertaining the test and reference item, the process of receipt, identification, storage, characterization, and traceability of the test and reference item. Then pertaining the study plan also, normally is about the approval of the study plan, amendment of the study plan, required content, which is the format of the study plan. It's not as per mention of the OECD GLP principle. And then, and also, about the recording consistency with the study conduct. And then the next one pertaining the study conduct, which is pertaining the raw data, deviation, or observation, recording, and also data traceability. Normally the highest one actually about the raw data, especially the chromatogram. Okay, your chromatogram is not complete. You just cut and paste, so you don't put the whole set of the chromatogram record. Okay, the next one is about the reporting of study result, the final report, the amendment of the final report, consistency with raw data, and then storage and retention of record and material, pertaining the archiving, retention, retrieval, and record integrity. And then the last one pertaining the facilities and also the test system is related about the environmental condition of your laboratories, contamination control, housing, care, and also monitoring. Okay, this one update about the GLP activities. In 2026, where the first one, we have a Mesyuarat Agensi Pemantau Pemantauan Kebangsaan together with the NPRA. Okay, on the 3rd April 2026, we also have a sesi taklimat berkenaan GLP to the Pesticide Board on the 11th March 2026. And then we also have a walkthrough session on e-accreditation 2.0 conducted on the 15th to 16th June this year. This walkthrough session is very important because we try to standardize the all procedures in a hard copy, and then we put it and then emphasize it on the system. Then the next one is pertaining the National Symposium that occurs today. This is a part of our program this year. And then the next one is about the revision of the DSM GLPCP manual, where we are planning to do it on the 7th until 9 December this year. The next one is the GLP activities attended in 2026. The first one for third meeting of the Working Party on GLP held on 14 to 16 April 2026, attended by Puan Fariza. And then training test item categorizations on the 1st April 2026, this one conducted online. And then the 17th OECD training course for GLP inspector. This one will be conducted on 2 to 5 November 2026 in Germany, where it will be attended by Encik Irwan and also Prof. Fadila. Okay, this one's just a little update and pertaining the GLP Malaysian 2026 outreach update. Okay, beside all the national activities and also OECD GLP activities, we'll also receive the outreach program from the overseas country that would like to see and also oversee the inspection process in Malaysia. Okay. This one is held on 9 to 10 October 2025 in Malaysia. And then the person actually is sorry. Okay, this one, for Fariza, this one from the Kazakhstan, right? Yeah, this one actually the officers from the Kazakhstan come to Malaysia and observe GLP inspections. Okay. So the DSM objective actually is so in terms of the GLP actually to affirming the continuous commitment in upholding the integrity of OECD GLP, like what my director said during the opening remark. We try to strengthen it and then to have a close collaboration with our regulator, CMA, the industry, the test facility, so that we can together enhance the GLP infrastructure in Malaysia. Okay, that's the end of my presentations. Is there any other question? Do you have any questions that you would like to ask? Okay, don't worry, during the coffee break, you can actually communicate with the speaker, not only myself. If you have any question, okay, don't hesitate to approach us. That's all. Thank you so much, everyone.
Thank you, Puan Sharifah Azlinda, for the comprehensive presentation and for sharing the latest update on the GLP compliance program of GSM. We certainly appreciate the insights provided, particularly on the expectations and implementation of GLP compliant activities. We shall now continue with our next session from another important regulatory authority. It is my pleasure to introduce Puan Fadilah Hasbullah, Head of GLP Section, National Pharmaceutical Regulatory Agency, NPRA. Puan Fadilah will be sharing insights on the GLP compliance program of NPRA, including the current program and its implementation. This session will provide valuable perspective on the regulatory requirements and expectation from the pharmaceutical regulatory perspective. Ladies and gentlemen, please join me in welcoming Puan Fadilah Hasbullah.
Oh, okey. Bukan yang ni. Ini untuk online. Ada. Dia punya remote ke tak boleh? Dia orang ke klik? Ada apa ni dia? Remote yang tadi. Remote tak berfungsi. Oh, tak nak klik dia. Untuk klik dia. Ada apa dekat atas ya? Okay. Ini mic dia. Mic pakai dulu. Ini untuk on. Okay. Okey, Assalamualaikum and selamat pagi, salam sejahtera semua. Nama saya Fadilah, saya daripada Seksyen GCP GLP Bahagian Regulatory Farmasi Negara atau in short NPRA. So untuk pagi ni, the first section, my slide will be focused more on the role of NPRA as CFA. And after that, I have another slide which focus on NPRA role as the regulatory authority, RA. Okay, this is the first slide and it's a bit of updates. I believe some of you have seen this slide because I presented this slide in the Mama Registrasi workshop last 2 weeks, 3 weeks, I guess. And also the same slide during NPRA GLP forum end of last year. Okay, all right. NPRA, we have okay, all right. So this is the first slide. So I will this slide focus on more on the NPRA rules as the Compliance Monitoring Authority, GLP in Malaysia. So our scope is on the pharmaceutical product, cosmetic, battery products, and also feed additive. And the other scope, it is under Department of Standard Malaysia, JSM. So in NPRA, we have three main centers. The first one is Center of Product and Cosmetic Evaluation. So basically, this first center is the activity of the NPRA, where they act as the regulatory authority for the registrations of pharmaceutical and also cosmetic in Malaysia. Next one, we have Center of Compliance and Quality Control, which support the registration of activity in NPRA. And the third one, we have Center of Regulatory Coordination and Strategic Planning for the coordination on the regulatory matters in Malaysia and also planning. So we have 3 technical centers and 1 center of administration for the management of the environment. Okay, so MPRA has involved as a participants or member of this collaboration. We are the WHO Collaborating Center for Regulatory Control of Pharmaceuticals since 1996. We also participate as the PICS, Pharmaceutical Inspection Corporation Scheme, since 2002 for the Good Manufacturing Practice, GMP. And since 2002, we have been actively involved in the activities related to the GMP program and also QA program in relation to the GMP. We also an adherent to OECD MED the GLP framework since 2013. And MPRA is also an observer in the International Council for Harmonization, or in short ICH, of technical requirement for pharmaceutical for human use since June 2018. So we're still observer and we're aiming to become one of the participating member. And lastly, we're also the member of International Pharmaceutical Regulatory Program, IPRP, since November 2019. All right, so CMA under MPRA is under the activity for GLP program is managed by the Good Clinical Practice and Good Laboratory Practice, and it is under the Center of Compliance and Quality Control. So this center is a big center. We have 8 sections under this Center of Compliance and Quality Control. So basically all the GXP activities is under this center. We have GMP section, Good Distribution section, Good Manufacturing Practice section, GCP section, GLP section. We have BE Inspection and Ethics Committee section. We have Pharmacovigilance GDP section. And we also have Surveillance and Complaint section, Product and Cosmetic Testing, and also Service and Supply section. Okay, so under GCP and GLP section, we perform GCP inspection to the trial site, sponsor, and contract research organization. We also conduct MPRA phase 1 unit inspection for the early phase of clinical trial. We go and inspect this early phase of clinical trial, particularly first in human trial. And we also act as the GLP Compliance Monitoring Authority for MPRA for Malaysia, for pharmaceutical scope. And under MPRA GLP Compliance Program, we perform GLP inspection for the non-clinical research facilities or studies. So our inspection is to the facilities under our GLP program and study specific for those that are related to the product registration. And we also manage serious risks reporting and protocol evaluation related to clinical trial. And we also involved in the foreign bioequivalence inspection program. Okay. So under this section, we have about 7 officers headed by me, sorry. Okay, and we have 3 full-time officers under GLP and 3 full-time officers under GCP. While all these officers are qualified in both GCP and GLP. All of them are qualified as GCP inspector as well as GLP inspector and also bioequivalent inspector. And we have 1 pembantu tarbih administrative staff to help us. Okay, so this is a little bit of update. So we have updated a few things on our webpage. Can the Secretariat click to the MPRA website? The first one for the Okay, so I shared with you the link. So this is the web page for GCP GLP section. All right, so you have 2 tabs there. One is for GCP and one is for GLP. And if you can see the blue tab above, we have directive, procedures, form, FAQ, guideline, GCP GLP inspection activities, and also list of GLP compliant test facility. So all the directive and circulars, the recent one would be study-specific inspection for foreign GLP inspection. And then under procedures and form, you can click to know how to apply and to get the latest form for NPRA GLP program. Yeah. So the first one is for GLP and the second one is for the GCP FIH. So this is the latest form, January 2026 for GLP application form. Can you click back? Okay, so we also have updated our FAQ part. All right, for GLP, we have updated it November last year. And we have updated for GCP as well. So this, if you have any queries, just refer to this FAQ tab. So, and if not, you can always drop us an email at sgcpglp@npra.gov.my. All right, can you go back? Website. Okay. So the major update will be the guideline. We have updated our manual and we rebrand it into a guideline. All right, we have, yes, the first one, Malaysia Guideline for NPRA GLP Compliance Program, first edition, updated 15 May 2026. You can click to the link. Okay, so this is the latest guideline. So it's the first edition because we rebrand it. Everything is here regarding the NPRA GLP compliance program on how to apply, what are the process involved, what are the process involved during inspection, post-inspection, and so on. Okay, boleh click back dekat website. Okay, boleh go back to the slide first. Okay, okay, we also have a GCP GLP inspection dashboard, which is a live inspection statistic. Can you go back to the website again? Yes. Okay, so these are like a live dashboard where you can click and play around to see what are the statistics for a particular GCP or GLP or BE inspection by year, by category. We can go to the standard. Can you click? Okay, and unclick the GCP. All right. So in total, since 2019, we have performed 20 inspections for surveillance for GLP inspection. Okay. Then can you scroll down to the page? Okay, bawah lagi. Okay. So these are the statistics for GLP inspection since 2019. For this year, we have performed so far 5 GLP inspections. And this also involve 4 surveillance inspection and 1 pre-inspection to the test facility. We have 2 more pending for this year. Can you go up? Okay, can you go to the statistic finding for inspection? And click GLP inspection. Okay, so you can see here the distribution of the finding by grading, whether by major or minor. And we categorize it according to the principle of GLP and also by year. Right? Okay, can you go up? Findings data chart? GLP inspection. Okay, so these are the trends: inspection by category and classification. All right. So you can see the bulk or the majority of the major finding is usually at the QA program and also at the performance and reporting of the study. And the bulk of the minor is at the performance of the study. So you can play around with this dashboard to see the statistic, and maybe you can do your own risk assessment to your test facility based on this analytics. Okay. Can you go back to the slide? Thank you. Okay, so I've covered this slide. Right, next. Okay, this is basically an extract to the dashboard just now that I showed you. Okay, for the 2026 updates and activities, as I said before, we have do a revision and rebranding to the MPRA GLP Compliance Program Manual, and we rebrand it into a guideline. And the last MPRA manual update was during the OECD in 2023, which was 3 years ago. And this new guideline, first edition May 2026, was made effective in 15 May with the issuance of directive by the Director of Pharmaceutical Services under Regulation 29, Control of Drug and Cosmetic Regulation 1984. So I'll talk more about this regulation for pharmaceutical product in my next session. All right. Okay, we also updated our Malaysian GCP guideline. This is to revise to align with the newly effective ICH GCP E6R3, and also Malaysian GCP inspection is also to align with the newly effective ICH GCP E6R3. These are the activity for this year. And these two guidelines are under revision. So this year is guideline revision year for us. We are trying to finish these two guidelines so that we can we can revise all these 5 guidelines over this section. But I'm not sure whether we can finish these 2, but we have completed 3 at least, the major guideline for GCP and GLP inspection. Okay, for 2026, among the activities that we involve, this was in 2025, Forum on OECD GLP CMA with stakeholder online. We held this last year online. We also attended the 40th meeting of the GLP Working Party in Paris in April this year. And MPRA also organized National Regulatory Conference, NRC in short, in June and July this year. We are the organizer. And recently we involved with the Mama Biostrasi OECD workshop as a speaker and also participants. Okay, so these are some photos from the OECD Working Party in Paris, April this year. So one attendee from MPRA and one attendee from JSM. And this is the biggest event for MPRA this year. We organized National Regulatory Conference. It's every 3-year conference. For this year is a regulatory, the theme is RegTech. And it's driving innovation and efficiency in regulatory. So basically, during this 3-day NRC, we discuss about the regulatory technology in pharmaceutical registration, as well as TMHS, traditional medicine and herbal health supplement. So I'm not sure when the next one will be, but usually it will be held every 3 years. A little bit about the NRC. So in total, we have 622 participants for this year for this NRC 3 days event. 369 are the participants for the pharmaceutical track, and 253 for the traditional medicine and health supplement track. We also have overseas participants, about 27, from country across Southeast Asia as well as Australia. Okay, so last year highlight will be a new policy, which is the GLP study specific inspection to the test facility of the non-mutual acceptance of data MAD countries. So this inspection is risk-based, a study-specific inspection. We only inspect study and not the facility. Is following an evaluation and or request by the MPRA evaluator, and it's for the purpose of product registration with the MPRA only. And it is not a compliance program for overseas test facility, which is not under MAD framework. Okay, last year also we introduced the electronic certificate. Previously we issued signed hard copy certificate, but since last year we only produced electronic certificate for GLP compliance program and other GXP program. Okay, last year also we we did a research and present a poster for the analysis of a decade of GCP inspection in Malaysia, and analysis of finding from various site sponsors, CRO and phase 1 unit over 10-year period from 2015 to 2024. And we present this poster at the DIA global meeting in DC, USA. Okay, for this year planning, we will involve as a steering committee for the 17th OECD training course for GLP inspector. And for next year, we will send one attendee for the 41st meeting of the OECD GLP Working Party in Paris. Okay, that's all from MPRA.
Thank you very much, Prof. Fadila, for the informative presentation and for sharing the latest update on the GLP compliance program of NPRA. We certainly appreciate the insight provided, particularly on the expectation and implementation of GLP compliant activities. Ladies and gentlemen, we will now take a short coffee break. The coffee break will be approximately 15 minutes. Please enjoy the refreshment prepared for you, and we kindly invite everyone to return to the hall by 10:30 a.m. as we will continue with the next session immediately after the break. Thank you and enjoy your coffee.
Okay, semua makanan ada dekat belakang. It's new lah, baru makanan yang baru lain daripada pagi tadi. Makan lagi. Hari ni kita makan. Boleh?
Ladies and gentlemen, we shall now proceed with our next industry session. This session will provide valuable insight into the regulatory expectation for GLP studies in pesticide registration, particularly from the perspective of the regulatory authority. Understanding this expectation is important to ensure that studies submitted to support pesticide registration are conducted in accordance with the relevant regulatory and GLP requirements, and that the data generated are of the quality, reliability, and integrity expected by the regulatory authority. For this session, we are pleased to have a representative from the Departments of Agriculture to share their perspective and regulatory expectation on this important area. Without further delay, it is my pleasure to invite Encik Baharudin Basri, Agriculture Officer from the DOA, to deliver the presentation entitled Regulatory Expectation for GLP Studies in Pesticide Registration. Ladies and gentlemen, please join me in welcoming Encik Baharudin Basri.
Okay. Tak pakai flip sendiri. Tak ikut sendiri.
Bismillahirrahmanirrahim. Assalamualaikum. Senyap eh? Dengar ke tak? Ke masih makan lagi? Okay, terlebih dahulu saya mengucapkan ribuan terima kasih kepada Jabatan Standard Malaysia kerana memberi peluang saya lah untuk berkongsi sedikit berkenaan pelaksanaan GLP di Jabatan Pertanian, terutama di aspek racun makhluk rosak lah. Okay, saya Baharudin bin Basri bertugas di bahagian kawalan racun rosak dan baja Jabatan Pertanian lah. Okay, sebelum saya pergi, pernah dengar racun rosak ke? Saya macam nampak muka-muka yang saya kenal lah. Okay, sebelum saya meneruskan, saya nak intro sedikit berkenaan dengan apa itu racun makhluk rosak. Boleh eh? Biasa dengar ke racun makhluk rosak? Pernah dengar? Dah makan kan? Ni tak ada suara ni. Ke tak nak saya kongsi? Tak nak saya kongsi, saya balik lah. Takut eh? Okay. Okay, tak ada, saya share saja. Okay. Untuk makluman, racun perosak ni kalau dia ada dua definisi. Secara umumnya, racun perosak dia boleh sama ada membunuh, menghalau, merengsa atau apa? Makhluk perosak. Racun makhluk perosak. Menghalau, saya nak tanya orang industri lah. Boleh saya tanya? Saya jalan boleh eh? Boleh kan? Saya kenal beberapa orang industri lah. Saya nak tanya lah. Boleh tak? Apa itu racun makhluk perosak?
Sesuatu yang membunuh,
Membunuh. Lagi?
menghalau.
Menghalau.
Ha.
Lagi? Membunuh, menghalau. Lagi? Tak ada? Tak tahu? Pelik kalau tak tahu ni. Okay, selain daripada tu, kita dia merengsakan jugalah. So, itu secara umum kita punya definisi. Tetapi kalau itu ikut akta, so untuk racun makhluk perosak ini kita dikawal di bawah Akta Racun Makhluk Perosak 1974 ataupun Akta 149. So, untuk definisi akta, racun makhluk perosak adalah sesuatu bahan yang dijadualkan dalam atau tersenarai dalam Jadual 1 Akta Racun Makhluk Perosak 1974 lah. So, untuk racun makhluk perosak ni dia boleh jadi satu bahan kimia atau kombinasi beberapa bahan kimia untuk membentuk sesuatu racun makhluk perosak. Okey. Kita bahan kimia yang tersenarai dalam jadual 1 itu kita panggil apa? Bahan kimia tu. Jangan confuse Sebanyak orang sebut pewaris. Bukan Kita tak mewariskan apa-apa. Kita adalah panggil perawis aktif atau active ingredient dan campuran dengan inert ingredient ataupun bahan lengai. So itu adalah kombinasi racun makhluk perosak yang kita senaraikan dalam jadual 1. So untuk tuan-tuan dan puan-puan melihat apa yang kita senaraikan dalam jadual 1, so tuan-tuan boleh semak kita punya website. Dalam website Jabatan Pertanian, kita ada nyatakan kita punya jadual 1. So boleh check lah. Ada berapa ratus 200, 300 lebih kurang begitulah. So semua itu dikawal di bawah akta racun makhluk perosak 1974. Okey, kat sini saya diminta untuk mencerita berkenaan dengan pelaksanaan GLP. So apa kaitannya racun makhluk perosak dengan GLP ni? So untuk kaitan dia adalah bila sesuatu racun makhluk perosak tersenarai dalam jadual 1, perawis aktif itu perlu didaftarkan. Kenapa racun makhluk perosak perlu didaftarkan? Sebab kita nak kawal lah. Apa tujuan kita nak kawal? Supaya racun makhluk perosak yang berada di pasaran selamat, berkesan dan kita kawal juga untuk melindungi manusia dan juga alam sekitar. So kita mengambil kira ke semua aspek dalam proses pendaftaran lah. So bila kita lihat, bila tuan-tuan dan puan-puan terlibat atau melihat chemical yang tuan-tuan handle terjadual dalam jadual 1, so kita perlu mengikut beberapa peraturan lah. Yang pertama adalah peraturan pendaftaran 2004. So bila kita tengok dah check dalam jadual 1 ada chemical yang tuan-tuan puan-puan handle, so dia perlu datang kepada bahagian kawal racun perosak dan baja untuk didaftarkan. So dalam akta kita ada peraturan. So dalam peraturan tu kita ada peraturan pendaftaran 2004. Peraturan ini telah dibuat 2004 dan dipinda pada 2009. Okay. So dalam untuk pendaftaran ni, so kita melihat beberapa checklist ataupun data requirement macam mana tuan-tuan dan puan-puan nak daftarkan racun makhluk perosak. So benda tu pun tuan-tuan dan puan-puan boleh tengok dekat kita punya website. Okay. Okay, itu introduction sebagai apa itu racun makhluk perosak, apa kaitannya dengan GLP lah. Okay, next. Okay. Kenapa GLP diwujudkan ataupun dilaksanakan untuk mengawal di bawah pendaftaran racun makhluk perosak? Yang pertama macam saya cakap tadi, kita punya peraturan pendaftaran tahun 2000 berapa? 2004, pindaan 2009 kan. Tetapi kalau kita tengok kat depan ni, okay, titik tolak permatuhan kita bermula 1 Mac 2013. Okay, kenapa kat sini? Untuk ni kita telah mengeluarkan satu perkeliling di bawah akta berkenaan dengan satu pematuhan bermula daripada 1 Mac 2013. Semua permohonan pendaftaran racun hanya dipertimbangkan sekiranya mematuhi amalan makmal baik, good laboratory practice lah, GLP. Kita start daripada 2013. So kenapa kita tak start 2009? Okay. Sepanjang 2009 hingga 2013 kita menerima laporan ataupun report-report yang dihantar oleh pihak pendaftar lah. Tetapi kenapa kita keluarkan perkeliling 2013? Okey, kita kata terus kenapa kita melaksanakan GLP? Yang pertama adalah perlindungan integriti. Ia nak mencegah manipulasi dan pemalsuan data saintifik makmal. Kenapa macam ni? Sebab apa kita keluarkan satu perkeliling? Saya cerita dari perspektif saya sebagai penilai lah. Pada tahun 2009 tu, 2008, saya masuk 2008, penilaian 2009. So saya lihat semua report-report itu yang dihantar oleh pendaftar. Muka-muka pendaftar pun saya pernah tengok kat sini kan. Mungkinlah bukan yang ada di sini. Menghantar report-report yang cut and paste sahaja. Faham? So dia ambil mana-mana, dia cut and paste. So dia punya nombor, dia punya font, penyusunan, reporting sangat-sangat berterabur. So bila kita lihat apa benda yang kita nak kawal, macam mana kita nak kawal dari segi reporting. So kita pun pergilah jumpa Jabatan Standard. Okay, kita belajar Jabatan Pertanian, pergi belajarlah macam mana good report kena ada. So kita diintroducekan berkenaan dengan GLP lah. Start from there, kita belajar apa itu GLP. Pernah dengar GLP kan? Dah belajar GLP tak? Dia ada pillar kan? Pillar untuk GLP. Pernah dengar? Saya tak tahu lah, mungkin saya dah lama, mungkin ada yang barulah. Yang tahu, kita tahu ada pillar untuk GLP. So dari situ kita belajar setiap pillar dan kita nak applykan untuk kita punya reporting di bawah racun makhluk perosak. Disebabkan isu cut and paste, dia suka-suka hati type, guna font, dia pilih yang mana-mana dia suka, dia hantar kepada PB, pesticide board lah. Lembaga lembaga racun makhluk perosak kita panggil pesticide board. So dihantar, biarlah pegawai yang baca. Ikut sukalah. Lulus ke tak lulus selain cerita, tapi hantar report sahaja. So titik daripada itu, kita nak mencegah manipulasi dan pemalsuan data saintifik makmal. Kenapa kita perlukan macam ni? Sebab yang saya cakap adalah, bila kita nak mendaftarkan suatu racun makhluk perosak, dia perlu berkesan. So kalau reportnya itu pun dah cacar marbah, macam mana kita nak pastikan racun yang kita letakkan di pasaran itu berkesan lah. So kita nak screening through dulu. So kita base by report dahululah. So seterusnya, GLP ni adalah standard kualiti antarabangsa, memastikan kajian kimia tepat boleh dipercayai dan diterima oleh pihak berkuasa. So kita tak enforce GLP pada peringkat awal, walaupun dia ada dalam garis panduan kita. Cumanya pada masa itu kita tak explore lagi apa itu GLP, dan selepas kita explore apa itu GLP, barulah kita implement lah. So dia adalah standard kualiti antarabangsa. Apa yang saya difahamkan oleh Jabatan Standard, untuk GLP ni dia boleh terima, kita adalah signatory kan? Kita signatory kan? Kepada GLP, OECD GLP. Kita signatory kan? Full adherence. Okay. So apa-apa yang makmal yang ada GLP, kita menerima. Tak kisah daripada dalam atau luar negara lah. So dia kualiti antarabangsa. So kita adapt benda tu. So apa-apa report yang ada GLP certification, so dia adalah kajian yang kimia tepat, boleh dipercayai dan diterima. Okay. Yang penting tepat tu dia boleh dan diboleh percayai lah. So sini adalah ketetapan mandatory. Kegagalan mengumpulkan dokumen sah mengakibatkan permohonan ditolak serta-merta. So pada sebelum 2013 kita terima saja. So kita tak ada buat penolakan. Tetapi as the time ni kita telah menambah baik. Apa-apa kalau syarikat pendaftar nak mendaftar, kita dah buat satu checklist awal. Checklist. So kalau report yang dihantar oleh pendaftar tu tak ada GLP, kita tolak siap-siap. So dia tak boleh masuk untuk dinilai dan didaftarkan lah. Betul syarikat pendaftar ada kat sini ni. Seram pula saya nak ni kan. Saya yang seram dengan diorang. So dia tak boleh salah satu checklist untuk pendaftaran, dia kena ada GLP certification. Okay. Next. Saya nak tunjuk realiti yang kita terima lah. Ada ke nama-nama syarikat yang saya paparkan kat dalam ni? Okey. So ni adalah realiti semasa statistik penolakan pendaftaran. So saya ambil beberapa tahun sahaja. So yang pertama adalah tahun 2023 lah. Daripada kesemua 115 permohonan yang dihantar oleh pihak syarikat, kita tolak 4.2%. 4.2%. Sebab apa? Dia melibatkan syarikat ni lah. PCTS. Saya sebut Saya sebut je. Kalau tak puas hati boleh tepi dengan saya lah. Nanti saya senaraikan apa yang tak betul lah. Okay, PCTS, Agros Bay dengan Hes Bio lah. So untuk 2024 kita terima permohonan lebih kurang 68. Ini setelah melepasi checklist kita tau. Permohonan sangat banyak. Kita ada checklist kan. So kita akan terima lebih kurang 68 untuk tahun 2024. Kita terima dan penolakan sebanyak 3.4%. Melibatkan 3 TOR dengan sumber chemical lah. Saya sebut je. So untuk inilah kalau untuk kita punya pembelajaran bersama. So apa punca kita tolak? Okey, yang pertama tiada GLP sah dikemukakan bagi data teknikal. Sebab kita buat checklist, kita masuk dalam sistem. Sistem you boleh upload lah apa-apa pun untuk lepaskan dia punya mandatory requirement. Tetapi ada pegawai yang akan nilai lah. Kita manual pun kita buat, manusia pun kita buat lah. So kita bila kita melihat setelah dia lulus untuk dalam sistem, kita melihat data-data dan punca utama kita buat penolakan adalah tiada GLP sah dikemukakan. Dia kata dalam sistem kata ada GLP, bila kita semak dokumen tak ada GLP. So dia jadi mandatory kan. Saya cakap tadi mandatory. So ni adalah asas utama kita menolak permohonan inilah. Okey, ada soalan tak? Tak ada? Boleh je tanya. Kalau boleh saya jawab lah. Okey, senyap je ni macam seram Seram ke perkongsian saya ni? Menakutkan? Aduh. Okay, puan menakutkan. Lepas ni tak boleh panggil saya lah. Slide saya menakutkan. Okey, seterusnya. Okey. Untuk peraturan pendaftaran 2009, kita ada beberapa kriteria ataupun benefit yang kita perlu patuhilah untuk pendaftaran racun malu perosak. So beberapa field, ada 2 field yang mandatory racun malu untuk GLP. Data GLP yang diperlukan untuk pendaftaran racun malu perosak. So field dia yang pertama adalah data quality, product specification. Data yang kedua adalah product toxicity. Product toxicity. Data ketiga adalah data efficacy ataupun keberkesanan. Pernah dengar kan? Keberkesanan. Dan yang terakhir adalah residu. So kita ada 4 major kriteria yang kita perlukan, tetapi hanya 2 sahaja yang kita enforce untuk GLP. So yang lagi 2 tu kita boleh kupaskan jika ada yang sudi nak buat. Kalau ada sudi nak buat, baru kita akan masukkan dalam kriteria lah. Setakat ni lab-lab tu tak mampu lagi untuk memenuhi GLP lah. Kalau kita mandatory kepada semua, saya rasa tak ada produk kot. Ada, mungkin sangat lambat lah. Ataupun dia punya very costly untuk mendaftar racun malu perosak. So yang 2 ni pun saya rasa memang a big cost juga for the industry. Okey, data yang pertama ataupun kriteria yang pertama kita melihat dari segi produk spesifikasi ataupun produk kualiti lah. So untuk ni kita tengok data spesifikasi mesti patuh GLP. Ni adalah parameter-parameter yang kita tengok untuk product specification lah antara saya tak nak list semua kalau tu nanti panjang. Okey, yang antaranya adalah boiling point, melting point, warna, pH. Semua ini perlu dilaksanakan dalam ataupun reportnya perlu dalam GLP lah. Okey. So ini antara sifat fizikal dan kimia, analisis 5 batch termasuk profile impurities. Kita juga mewajibkanlah untuk makluman tuan-tuan dan puan-puan, racun makhluk perosak sekarang kita dah ada tarikh luput. Okey, ini perkongsian sajalah. So kita ada tarikh luput bermula tahun 2023. So sejak itulah pihak syarikat diminta untuk menghantar data juga. So data ni kita beri sedikit kelonggaran. Kalau ada GLP, kita GLP dululah. Kalau tak, minimum ISO. So ada yang hantar guna GLP. So it's good lah. So kita tak naikkan lagi standard untuk data requirement, hanya GLP sahaja. Kita cuma hanya untuk physical and chemical property sahaja untuk product specification lah. Okay, ini antara sifat parameter yang kita lihat. Selain daripada product specification, area yang kita tengok adalah kajian toksikologi. Mesti mematuhi standard GLP sepenuhnya. Okay, ada dua untuk produk spec dan juga toksikologi. Okay, untuk toksikologi ni dia besar sikit, dia punya coverage. So untuk produk racun makhluk perosak, kita kategorikan sebagai proprietary dan juga komoditi. Okay, untuk proprietary ni adalah produk-produk racun perosak yang memang baru, yang tak ada dekat Malaysia. So itu kita panggil produk proprietary lah. So dia lebih kurang berapa tahun lah? 10 tahun 10 tahun dia punya period lah untuk proprietary. After that it can consider commodity. Commodity ni adalah produk generic, kita panggil lah the generic pesticide. So untuk toxicology dia ada dua kategori. Dia kena pilih. Kalau dia klik commodity, berapa packs? 6 pack Siapa ada di sini 6 pack? Yang lelaki lah. 6 pack ke 1 pack? Kita untuk komoditi kita minta 6 packs. Acute oral. Kalau saya salah saya tak pernah ada di bidang ni dah. Okay. Acute oral, acute dermal, acute inhalation, skin sensitization, eye irritation. Dan satu lagi, tak ingat lah. Corrosion lah. So ada 6 pack yang kita minta, kesemuanya perlu GLP. Okay. Untuk kes toksikologi, kesemuanya data perlu dalam GLP. Okay. Kalau untuk proprietary, dia kena ada chapter 3 which is impact on human and animal. Lepas tu dia ada juga impact on fate and behavior and also effect on non-target species. So saya cakap untuk racun perosak dia kena cover semula. Manusia, alam sekitar, beneficial insect, semua kita cover. Dan satu-satunya kriteria ini kesemuanya perlu GLP untuk data requirement kita lah. Okay. Okay, next. Okay, saya sikit-sikit lah based on pengalaman berkenaan dengan bila kita menilai data yang dihantar menggunakan atas tiket GLP ini platform GLP kan. So kita cerita atas tiket GLP lah. So antara kriteria penolakan bahagi data teknikal untuk produk spec dengan juga tox, antara dia punya in compliance, yang pertama adalah isu akreditasi makmal. Okay, makmal Uji kaji tidak menerima pengiktirafan GLP atau tiada dalam semakan OECD GLP atau ISO. Pernah semak ke? Siapa yang dah accredit GLP? Siapa daripada lab ah? Lab. TF. Test facility. Ramai ya? Boleh angkat tangan tak test facility? Dah semak website? Ada nama tak? Ha? Ada? Betul Ada So boleh semak Selain daripada itu kita semak untuk international punya lab lah kalau ada. So isu dia sekarang ni kalau kat Malaysia tak apa, kita ada jabatan standard untuk kita refer. Tapi kalau untuk luar negara kita ada website yang kita perlu lihat. So bila dia hantar tu, first kita punya kriteria kita semak dulu sama ada lab itu di certified GLP atau tak lah. So bila kita semak tak ada GLP. Dalam borang dia kata ada GLP kan? Declare je. Nanti pandailah pegawai nilai kan. Okay, bila kita nilai tengok tak ada. So kita tolak So sebenarnya in compliance yang kedua adalah ketiadaan sijil. Ni ada sedikit pertikaian lah. Sama ada di under US ke, US hanya compliance saja, tak ada GLP cert ke. So kita dah verify lah dengan CMA kita. So dah ada lah kita punya pegawai dah train macam mana kita nak lihat sama ada dia compliance ataupun certification lah. So benda tu kalau dia tak ada memang kita dah check, kita akan tolak juga. Okay. Seterusnya in compliance adalah tarikh luar tempoh sah. Ada pengesah kan? Berapa tahun? TF. TF bantu saya. Berapa tahun? Validity untuk GLP.
Depends on CMA lah kalau ada isu cert.
Depends on CMA. Berapa lama? NPRA 3. Ada yang lebih daripada 3 tak? 5 tahun ada? 5 tahun. Oh, 2, 3. 4 ada? 4. Ada? Tak ada? 5 tahun? Tak ada. So minimum 2 atau 3 lah. 3 tahun kalau dekat Malaysia lah. So yang ni adalah kita punya data kajian ni lebih kepada ujian 5 batch atau physical and chemical yang dilakukan daripada luar tempoh kelulusan GLP. Ini maksudnya dia hantar tahun 2025 tapi report dia 1983. So kita akan tengok balik lah sama ada perlu kita boleh terima atau tak. Sometimes kita ada justifikasi kenapa kita terima. So dia berbeza kalau tuan-tuan dan puan-puan, kita takkan pukul rata. So setiap produk kita nilai individually. So kita tak sama rata lah. Kita ada garis panduan tapi kita tak nilai okay, GLP je boleh terima semua. Tak. So kita akan ada juga kriteria yang ditetapkan. So kalau dia macam dah lama sangat, so kita akan minta dia buat yang baru lah kalau ni possible. Okay. Seterusnya format tidak piawai. Format laporan data tidak mengikut standard GLP. Okay, masih berlaku Start daripada 2009 hingga 2013 tu memang banyak saya tengok kan. Siapa nak keluar biji mata saya tengok. Saya rasa nak lempang-lempang je orang yang cut and paste tu. Report tu kan. So still lagi berlaku sampai sekarang. Masih. Tapi dia punya tak banyak lah. Tapi masih juga berlaku. Okay. Seterusnya adalah rantaian sumber terputus. Laporan 5 batch dibiayai oleh pihak yang bukan penghilang sumber. Kita tengok sponsor. Boleh tak? Sponsor A buat report, syarikat B datang, dia kata sponsor A bagi dia guna data. Boleh tak? Boleh? Kalau boleh, dia kena apa? Letter of authorization. Maksudnya sponsor membenarkan menggunakan data lah. Tetapi kita tak jumpa benda tu. Ataupun manipulation of letter of authorization. So kita check satu-satu. Makin pertebal lah cermata saya ni melihat data-data tuan-tuan dan puan-puan. So kalau rantaian sumber terputus ataupun kita tak ni so kita inilah tak benarkan. Next. So ni antara contoh yang kita terima iaitu akreditasi palsu atau tidak diiktiraf. Kebanyakan kita yang tak iktiraf tu daripada GB, China punya ni lah. So kita tak terima. Seterusnya adalah akreditasi palsu tadi. Baik. Okey, yang ni isu dia menggunakan si GLP, tapi dia tak ada dalam inilah makmal tidak berdaftar. Dia gunakan si GLP lah. Manipulate ni pandai ni, berani dia manipulate. Okey. Next. Ni pencanggahan. Bila dia hantar maklumat, kita cek sampai maklumat dia tally dekat SDS atau tak. Sampai macam tu sekali tuan-tuan dan puan-puan, data tuan puan-puan ni kita semak. Bukan hantar, terima je. Okey, next ada pemalsuan maklumat makmal. Okey, yang ni saya nak ni lah kes ni lah anti-cam technology inventor. Pemohon meminda tempoh kajian lemah 2022 menggunakan sijil dari GLP yang mempunyai nama dan alamat berbeza pada tahun 2022 semata-mata untuk meluluskan data lampau. So ni pun kita dah dapat tangkap kita cek one line by line even sijil pun kita tengok betul ke tidak. Okey, yang ni pun kita dah beri amaran lah. Okay, next. Ni adalah kesalahan major bagi dokumentasi dan kekaburan lah. Dari segi yang pertama adalah isu tiada tanda tangan sponsor dan quality assurance. So yang ni saya mungkin nak throw kepada all lah. Sama ada saya sebagai regulatory authority perlu tak tanda tangan ini. Adakah major mass ataupun minor mass sahaja. Kalau minor mass kita takkan isu perkara ni lah. So boleh dibincangkan. Kalau ada boleh maklumkan. So saya akan bawalah mandat ini kepada rakan-rakan saya di pesticide board sama ada perlu atau tidak signatory ni. Siapa yang hantar yang bagi duit ni yang sponsor ni sama ada dia perlu semak atau tidak. Itu adalah tanggungjawab Bila you hantar, you bagi test item, report tu you baca ke tidak sponsor ni. Test facility boleh buat. Tak ada masalah. Sponsor tu macam mana kan? Sebab report tu akan dihantar kepada regulatory authority lah. So kita nak tahu perlu ke tidak as a sponsor. Okay, seterusnya adalah isu makmal berdaftar secara sah tapi tiada dalam akreditasi area of expertise. Kadang macam ni toxicology, kadang-kadang on product specification. Dia kata dia GLP, betul. Tapi area of expertise tak ada. Tak disahkan tak inspection semua tu kan. So tak ada, so kita pun tak terima lah report itu. So next. Ni yang terkini. Ni yang terkini. So bila dia hantar report, lepas tu kita tolak. Okay. So ada isu yang dekat sini, kita tolak. So kita minta dia buat report baru. Kita minta dia report baru. Lepas tu kita nak justifikasi. Justifikasi. Kenapa dia buat macam tu? Sebab bila berlaku benda ni, kita CMA, RA telah melibatkan CMA. So kita perlu report kepada CMA what's really happened lah untuk this product. So kita tak terima report ni dan kita minta dia buat report baru hantar kepada kita lah. Okay, next. Ni dia punya justifikasi lah. Cepat sikit. Okey, ni dia punya rentetan dimohon 2025 kita start menilai, lepas tu ada isu dan kita tolak laporan ni lah. Okey, next. So ni adalah antara justifikasi yang kita terima secara ringkas lah. Disebabkan kata jawapan dia adalah untuk pencematan bahan piawai, untuk kecekapan kos ujian dan juga integriti kimia kekal. Dia katakan kekal, so kita akan bincang balik dengan Jabatan Standard perkara ni. Ada perbincangan Kita akan buat nanti formally bersama Jabatan Standard lah. Okay, next. Okay, ini adalah kita dah ada, selain daripada tu kita buat juga apa ni, kempen kesedaran. Okay, kita menyedarkan kita punya pendaftar apa berkenaan dengan tanda-tanda sijil yang red flags lah. Yang pertama, kalau tu tak ada nombor rujukan. Yang penting apa? Kita bagi apa ni nombor rujukan? Kenapa kena ada nombor rujukan? TF boleh tolong jawab tak? TF lain. TF kat depan je kesian. TF lain. Tak nak jawab? Tak nak jawab, saya tolak je lah mana-mana TF yang tak nak jawab nanti Gerah je. Okey, yang pertama kena ada nombor, lepas tu nama makmal tidak dapat disahkan dalam inilah. Dan seterusnya adalah dokumen kelihatan terlalu umum menggunakan template salin dan tampal, copy and paste lah. Dia guna copy and paste. So untuk reporting GLP dia memang ada standard reporting. So kita terima pakai reporting itulah. Okay. Next. Okay. Saya kongsi sedikit sahaja. Saya pun dari masa berapa? Dah habis dah kan? Dah habis Tepat kan? Okay, sekian sahaja. Ada soalan ke? Ada soalan nak dibuka dan boleh tanya. InsyaAllah kalau saya boleh jawab, saya jawab lah. Ada soalan.
Saya Samsulida daripada Sri Berhad. Sementara Mike dekat tangan sini, saya nak introduce Sri adalah one of the GLP facility juga. Dan kami sijil ada berdaftar dengan ESM dan juga MPRA. So ada dua soalan.
Nombor berapa? Nombor sijil nombor berapa? Promote lah.
Okay, soalan saya yang pertama berkenaan dengan six-pack tadi. Oral, dermal, irrigation, corrosion, sensitization, inhalation. So kita tahu dalam OECD, untuk sensitization, kita tahu dalam OECD dia mention that animal test adalah last resort. So ada validated method untuk in vitro input. Tapi dalam guideline pesisai board mengatakan di situ in vitro sensitization, in vivo sensitization. So soalan saya, adakah kita datang kepada kami, adakah kita orang boleh advise untuk tukar method kepada in vitro in situ, atau kami tetap kena buat in vivo sebab guideline memang mention animal is last resort where there is no validated method. Okay, itu soalan pertama. Soalan kedua adalah jika ada soalan lagi, dia kata Encik Bahar dia kata kalau pewaris atau perawis AI lah kita katakan AI active ingredient tu tiada dalam jadual, so kena daftar kan. Tapi let's say active ingredient dia tiada dalam jadual. Dia memang tiada dalam jadual, tapi dia punya ingat sampingan dia tu ada dalam jadual. So adakah penilaian pesisai hanya bergantung kepada AI ataupun dia tengok juga kepada inat?
Okay, first tanyalah dapat persicilan GLP. So ada dua soalan. Yang pertama soalan berkenaan dengan test guideline TG kan, test guideline. So dalam kita punya GP berapa? Kata baca tadi. Saya tanya balik GP berapa? GP Talks GP2. Sebaik ada menjawabkan. So GP Toxicology untuk GP Toxicology tahun 2000 2020 kalau tak silap saya. Okay, ada perbincangan berkenaan dengan isu yang this is international punya requirement juga. Okay, kalau dalam kita punya GP2, kita ada nyatakan test guideline yang dibenarkan bagi setiap contohnya acute oral, acute dermal. Okay, kita pun sekarang sedang review kita punya GP. GP1, GP2, GP3 dan GP4, including GP7. Itu kita akan bincang kemudian lah GP7 tu. Cumanya kita sedang dalam review dan kita memang ambil maklum ada perubahan-perubahan daripada international punya requirement. Okay. Untuk garis panduan itu hanya untuk kita refer. Okay. Kita refer pada yang memang blur, tak tahu test apa nak guna untuk acute oral, untuk acute dermal, oral pun banyak. OECD guideline, JMAF guideline semua tu kan. So kita terima as long as dia follow international punya tu, kecuali GB tadi. GB tu memang kita tak terima lah. Kalau unless siapa dah pergi verified GB tu, datang kepada PB, mungkin kita akan tengok lah. Ataupun siapa nak sponsor untuk pesticide buat pergi tengok GB punya test guideline, kita welcoming lah. So untuk tambahan itu, sebab untuk yang kata kalau international requirement, kita ambil maklum dan kita sedang dalam proses penambahbaikan. So kalau ada, kalau nak juga, kita ni kadang-kadang maklumat-maklumat terkini ni you ada first hand yang dapat, yang tahu. Kalau you ada informasi baru, boleh datang kepada pesticide board untuk menceritakan balik. Kan kadang-kadang you pergi conference, mana-mana conference, which is kadang-kadang kita tak ada berkesempatan untuk pergi. You all yang ada informasi tu. PB ni sentiasa terbuka. Kita dah pindah pejabat, jangan pergi KL pula kita dah pindah Putrajaya. So datang ke Putrajaya lah. Boleh jumpa dengan tim toksikologi untuk menyatakan apa juga perbincangan. Jika benda itu boleh disahkan dan boleh diterima, kita akan masukkan dalam garis panduan kita. So apa dia punya list tu hanya untuk reference. Yang mana kita boleh dapat. Kita refer mana OECD ke JMAF ke kan. So kita masukkan supaya dia tak terikat hanya satu test guideline sahaja. Kalau kata ada isu on animal kan, kalau kata ada conference berkenaan kata tak boleh nak guna animal is last resort. So kalau nak gunakan in vitro okay, in vitro kan boleh je sebab sekarang teknologi semakin maju. So kita pun nak kena catch up dengan ni kan. So kita updated kita punya GP. Kalau ada teknologi-teknologi baru yang kita boleh ni dan dia selari, dan kita boleh terima lah. Dia tak akan rigid sebenarnya. So soalan kedua tadi apa tadi? Tak ingat lah. Oh, AI First tadi, kita regulate kalau ada dalam jadual 1. Apa yang berlaku yang tak ada dalam jadual 1? Soalan saya pertama adalah, AI itu adalah, yang first saya cakap tadi lah, sama ada dia membunuh ke, dia ada pesticidal properties. Kalau dia ada pesticidal properties, dan you boleh mohon dengan pesticide board untuk dimasukkan dalam jadual 1. So kita menilai both active ingredient and also inert ingredient. So kalau inert itu, kita akan tengok percentage. Kalau dia punya lower percentage itu, memang dia sebagai inert, so dia akan kekal sebagai inert. Kalau dia punya percentage adalah melebihi daripada dia punya value yang kita tetapkan, dia akan jadi active ingredient. So dia kena tengok percentage. Dia tak boleh kata semualah. Tapi kalau tu kita akan nilai kesemuanya, both for active dan juga inert ingredient. Okay. So tu saja. Oh, ada soalan ke? Okay.
Soalan satu, maksudnya syarikat pengeluar perlu confirmkan dengan RA whether the in vitro method been accepted.
Ya, maksudnya datanglah jumpa kita kan. Sekarang teknologi sekarang ni kita pun tak update di peringkat PB. So adalah tim toksikologi yang tahu benda-benda macam ni dan mungkin dia aware. Ataupun kita punya sekarang garis panduan contohnya 2020, kita nak updated 2026 ke 27 ke. So kita akan mengambil kira perkara itulah. Okay, saya tutup. Ada soalan ke? Puan Farizan nak suruh tutup dah ni. Mungkin ada PM tepi ke?
Boleh kita korbankan masa lain sikit
Minta maaf lah saya ambil masa tuan-tuan dan puan-puan. Boleh, teruskan.
From Belgium, we are one of the FMCG company producing household and germ protection product.
Okay.
So one of that's the pesticide. My question is because due diligence, extra due diligence and all the higher standards yang dikenakan, can we expect a faster review and approval period from the pesticide board? Because that is one of the main concern for us.
Pesticide board rasanya Okey, untuk makluman tuan-tuan dan puan-puan, kita bekerja secara manual dan juga secara online. Okey, kita pun sekarang semua dah kerja dari rumah, bekerja fleksi. So kita towards tu online system lah. Even pun pagi tadi pun sebelum saya datang sini, kita ada perbincangan berkenaan dengan satu sistem yang akan dibangunkan untuk menggantikan sistem lama, fully system. So sekarang ni kita both untuk manual dan juga online. So bila tuan-tuan tahu so saya nak cerita sikit kenapa tak adalah nak mempertahankan pesticide board ni. Cumanya tuan-tuan kena tahu bila sesuatu data dihantar itu bukan satu helai saja yang hantar kan. Kalau untuk toksikologi tu saya tak tahu, hari tu dia cakap ada 3 troli. 3 troli kalau puan tengok tadi penilaian kita line by line. So kalau katalah itu first tanggungjawab kita adalah untuk memastikan racun perosak yang berada di pasaran selamat. So kalau kita tak go through line by line kan, kita biar je. Even dia kata ada GLP pun, bila kita semak GLP dia tak valid. So dia mengambil masa untuk kita menilai. Bukan kita satu produk sahaja. Siber kita ada lebih kurang 3,000 produk. Dia ada pelbagai kategori, pendaftaran baru, pendaftaran semula, tukar sumber. So dia banyak. Pegawainya ada 4 orang je puan. Kalau boleh saja untuk tambah pegawai, insya-Allah lah boleh kan. Saya ni pun dah tak tahu macam mana kan. So kita akan ada satu lagi sistem baru menggantikan My Pesticide. InsyaAllah kita dah sedang develop My LRMP. So kita akan tengoklah semua one stop center kat situ and fully online. So itu kita cuba mempercepatkan lah. Untuk makluman kita ada yang cepat. 6 bulan, 7 bulan. Ada je. Tahu ke? Tahu kan? Saya nak tanya pendaftar lah. Ada je 6-7 bulan. Syarat dia apa? Semua lengkap. Provided no single queries. 8 bulan. Boleh? Tetapi kita nak kena educate pendaftar ni yang menghantar data ni. Jangan dumping to the pesticide board everything. Baca data anda. Saya tanya tadi kan, sponsor baca tak report sponsor? Report. Test facility dah buat. Sponsor dah ada, tapi sponsor tak sign. Soalan saya lah, sponsor tak sign. Bila sponsor tak sign, sponsor baca tak? Tak. Tak baca. TF lah saya tanya TF. Sebab memang kita terima report. Memang tak ada sign tu kan. So bila kita dapat, kita tanya balik. Dia tanya kita kenapa lambat. So kita kena check sebab banyak partiality kat dalam tu. So kita cubalah. Bukan kita nak menafikan. Ada part yang kita lambat. Sebab dia melibatkan human. Kan? So nak bawa file setebal-tebal tu, nak kita bawa, nak kita baca. Masa ni, bila membaca ni, dia ada melibatkan emosi kita manusia kan. Rasa berdatang 3 troli ni, kalau tuan-tuan nak siap dalam masa sebulan, 3 troli tu kita nak baca. So di time consuming dan melibatkan emosi jugalah. Kalau melibatkan emosi tu kita cubalah. Nanti ada mental health kan. So kita libatkan itulah. Tapi kita cuba, kita ambil maklum. Memang perkara ni banyak diutarakan semasa sesi engagement dengan pesticide board. Kita ambil maklum perkara ni lah. Okey, minta maaf Wan. Saya ni kalau ada nanti kita PM tepi kalau ada saya sebentar lagi. Terima kasih.
Kami teruskan dengan selanjutnya dan ngobrol balik pada the next presenter. Teruskan.
Thank you very much, Encik Baharudin Basri, for the insightful and informative presentation on regulatory expectation for GLP studies and pesticide registration. I believe this sharing has provided our participants with a clearer understanding of the regulatory requirements and the importance of generating reliable and quality data to support the safety and efficacy assessment of pesticide product. Ladies and gentlemen, we shall now proceed with our next industrial session. This session will provide insights into the regulatory expectation for GLP studies supporting the registration of pharmaceutical products, including the importance of reliable and scientifically sound non-clinical data in supporting regulatory evolution and decision making. Without further delay, it is my pleasure to invite Puan Fadila Hasbullah, Head of Good Clinical Practice and GLP section from the National Pharmaceutical Regulatory Agency (NPRA) to deliver the presentation
Tak tahulah.
regulatory
Tak tahulah.
expectation
Sebab
for
kalau dia atas kena
GLP
tailing.
pharmaceutical products. Ladies and gentlemen,
Dia
please
bola.
join me in welcoming Puan Fadila
Saya
Hasbullah.
pakai nama. Saya guna di smart. Okay. Okay. Ya. Okay. Assalamualaikum. Tak boleh. Okay, test. Alright. Assalamualaikum dan selamat pagi. So, saya hanya ada dalam 30 minit untuk bagi presentation. I hope I can summarize this and concise as possible dan dalam masa yang sama information sampai. Alright. Okay, untuk my second topic is on the regulatory framework for pharmaceutical products in Malaysia and also expectation for GLP studies. So untuk pharmaceutical product ni dia besar, dia banyak modul and non-clinical study is part of the module. Right, next slide. Nak control je senang. Okay, so these are the outline of my presentation. Of course, I kena go through the organization overview balik to recap. And then I will touch on the international framework for what are the global expectation for pharmaceutical product, especially from the regulatory overseas such as EMA, US FDA, MHRA UK, Swissmedic, and so on. And lastly is on the focus on the product registration process in Malaysia. Okay, next slide. Right, next. Okay. So NPRA, Bahagian Regulatory Pharmacy Negara ataupun English dia National Pharmaceutical Regulatory Agency ni actually di bawah program perkhidmatan farmasi. So under Kementerian Kesihatan dia ada banyak program perubatan, program farmasi, kesihatan awam, semua tu, and then di bawah program perkhidmatan farmasi kami diketuai oleh Timbalan Ketua Pengarah Kesihatan Farmasi iaitu Yang Berusaha Dr. Azwana Ramli, dan di bawah program perkhidmatan farmasi di bawah Dr. Azwana kita ada NPRA. Pengarah NPRA sekarang vacant sebab beliau baru saja bersara Julai hari itu after NRC. And then di bawah program juga kita ada bahagian penguatkuasaan farmasi, yang ni yang pergi tengok registered and unregistered product. Kalau dia dapat information, intelligence, especially on the unregistered product, then they will conduct this sort of rate. Okay. And then kita juga ada pharmacy practice and development division. Untuk yang enforcement tadi diketuai oleh Yang Berusaha Encik Muhammad Zawawi. Untuk Pharmacy Practice and Development Division yang ini amalan dan perkembangan farmasi diketuai oleh Puan Zuhaini binti Muhrim. So basically service farmasi dekat hospital is under Pharmacy Practice and Development Division. Seterusnya untuk Pharmacy Strategy and Planning Division diketuai oleh Puan Nur Ain sekarang. Dan akhir sekali kita ada Lembaga Farmasi Malaysia yang mana pendaftaran ahli farmasi di Malaysia perlu berdaftar dengan LFM, dan pengarah LFM yang baru saja lantik 2 minggu lepas iaitu Tuan Muhammad Azharuddin bin Aziz. So di bawah MPRA kita ada 3 main center tadi saya dah describe. So kita ada Center of Product and Cosmetic Evaluation, pusat penilaian produk dan kosmetik. Kita ada Center of Compliance and Quality Control, pusat compliance dan kawalan kualiti, dan akhir sekali kita ada Center for Regulatory Coordination and Strategic Planning ataupun pusat koordinasi regulatory dan perancangan strategik. So untuk regulatory activity RA dia di bawah Center for Product and Cosmetic Evaluation. So di bawah Puan Rosiza. So di sini pendaftaran untuk prescription medicine, new drug product, biologics, generic dan juga termasuk non-prescription here is OTC, over the counter drugs. So kita ada prescription and non-prescription OTC. Kita juga ada traditional medicine, dalam label besar dia sekarang known as natural medicine. Tapi di bawah akta dia dikenali traditional medicine. Nanti saya explain further. Dan ada health supplement dan juga vet medicine untuk haiwan. Dan di sini juga bawah RA kita ada issuance of clinical trial import license dan juga exemption untuk mengilang bahan aktif untuk percubaan klinikal di Malaysia. Kalau import dia perlu lesen mengimport. Kita panggil CTIS, CTX. Dan kita juga ada cosmetic notification dan akhir sekali issuance of endorsement letter for ancillary drugs. Okay, untuk international framework for pharmaceutical registration. So basically tadi saya explain yang MPRA adalah observer kepada ICH. And basically ICH ni established in 1990, more than 30 years ago. Dia adalah global platform for RA, regulatory authority and also pharmaceutical industry. Yang mana sekarang terdiri daripada 25 member country, 43 observers including MPRA Malaysia and also nearly 700 experts worldwide. And function ICH ni actually they develop technical guideline untuk non-clinical, clinical and also quality aspect for the registration and also marketing authorization. In global they call it marketing authorization, MA, of pharmaceutical product. And this technical guideline are developed through scientific consensus lah. Basically similar framework with the development of TG under OECD MAD framework with public consultation. These ICH guidelines are published on the ICH website and adopted by the RA all over the world. And this ICH also, they regularly promote harmonization in the interpretation and also application of technical guideline. So how to interpret and application of technical guideline ni memang those who are members and also observers, dia punya expectation for the marketing authorization tu sama, almost similar. Okay, next slide. Okay, so under ICH guideline, dia ada 4 main guideline, which is QSEM, Quality Guideline, Safety Guideline, Efficacy Guideline, and also Multidisciplinary Guideline. So untuk quality for pharmaceutical product, dia cover harmonization in quality area, for example, conduct of stability studies, defining relevant threshold for impurity testing, and also flexible approach in the pharmaceutical risk-based in GLP. And then untuk S guideline, safety guideline, ICH provide comprehensive set of safety guideline for the potential risks like carcinogenicity, toxicity, and reprotoxicity. So ini guideline, what are the requirement for the regulatory authority untuk pharmaceutical product lah. But how to do it, you can refer to OECD TG. So based on the product indication, they provide this list through this guideline. And E is for efficacy. So the main part untuk pharmaceutical is always QSE, quality, safety, and efficacy. E for efficacy guideline, they concern more on design, conduct, safety, and reporting of the clinical trial all over the world lah. And also it covers novel type of medicine derived from biotech. Also yang ni termasuk pharmacogenomic and genomic techniques to produce targeted medicine. And lastly, ICH also have a guideline yang multidisciplinary. So they label it as CM, and this is the cross-cutting topic. So under this multidisciplinary guideline, they have this common technical document, CTD. So CTD ni modul pendaftaran ataupun marketing authorization untuk pharmaceutical across the ICH member and almost all over the world lah. Okay, so untuk multidisciplinary ni dia ada M1 until M18. Okay, let's go to the CTD. Alright. So under CTD triangle, under M4 ni dia specific on the common technical document and they have CTD triangle. So dekat atas sekali kita akan nampak yang tip of the pyramid tu regional administrative information, module 1, yang ni country specific. So basically apa yang country require extra from other country, so they will put in under module 1, country specific or regional specific. And then they have and this one is not part of the CTD, but the CTD is designed like this. And module 2 ni basically on the summary and also overview of the quality, non-clinical and also clinical. And module 3 we have more quality, module 4 non-clinical study report, and module 5 clinical study report. So module 2 to basically summary and overview, but the complete information or for the report for each of the quality, non-clinical and also clinical dekat module 3, 4 and 5 respectively. So QSE ni they assemble in the common format called CTD tadi dah. CTD ni they organize into 5 module. The country specific lies on top. So whatever the country or RA requires akan sit on top, and kalau tak fulfill then ada possibility you cannot be registered lah. Okay, module 1 region specific, module 2, 3 and 5 intended to be common for all regions. So member countries and observers selalu akan follow this. And currently dekat Malaysia, we are also ASEAN member countries. We follow the ACTD format. So ACTD format dengan ICH CTD is sama. It's just difference in term of formatting and also numbering of the information. Documents required are similar. Next slide. Okay, so yang ni untuk CTD, untuk M4R4, number 1 basically the organisation of CTD, standard format yang perlu untuk CTD, used for submission to regulatory authority and organize information required for the registration, especially new product involved and also includes biotechnology vaccine. For M4Q quality, dalam kurungan tu actually a revision number. So kalau nampak R1 tu dia baru revise first time, kalau R4 tu dia dah 4 times undergone a revision. So MQ4 is for quality section, and this provide harmonized structure and format for presenting the chemistry, manufacturing control and information for registration dossier. Next slide. Okay, next is on the M4S for safety. So this one is dia cerita about non-clinical overview and non-clinical summaries of module 2 and also organization for module 4. So this document CTD safety M4S ni guideline akan cerita the structure and format of non-clinical summaries in module 2 and also provide organization of module 4 for non-clinical study report. So you need to refer to this M4 under ICH CTD punya. Okay, lastly M4E for efficacy, revision dia dah 2 kali revision. So yang ni on the clinical overview and clinical summary and also clinical study report of the clinical section for the clinical trial report lah. So CTD efficacy or M4E describe the structure and format of the clinical data in an application, including summaries and detailed summary for the clinical trial report. All right, next slide. Okay, so now kita pergi kepada registration product in Malaysia. Tadi kita dah nampak structure CTD. So Malaysia sekarang ni masih dalam sistem kita, dalam Quest 3. MPRA Malaysia sekarang guna sistem Quest 3 Plus, dan dalam sistem Quest 3 Plus tu dia punya structure is ACTD. Untuk our new system, we are currently at the SRS level, software requirement specification. Kita aim untuk design the system so that they follow the ICH CTD. Tapi benda tu belum pasti lagi sebab sekarang masih dekat SRS level. So we are in talk with the developer to design that the system tu sama ACTD dan juga CTD supaya nanti senang for the submission. Because we also receive feedback from industry during dialogue, dia kata okay, sebab Malaysia kena follow ASEAN, the format dia different sikit berbanding dengan CTD overseas. Jadi bila nak submit kat luar dia macam tertukar-tertukar sikit. Okay. So sekarang we take note on this, dan macam for Quest 5 memang kita cuba untuk harmoniskan this requirement lah. And also for GLP program pun kami cuba untuk buat secara online. In future kalau boleh report kapal submission through MPRA punya Quest 5 system. We'll see how it goes first. Because sekarang masih dekat SRS level. Okay, untuk this part, product registration process in Malaysia. So kita ada dalam 5 akta termasuk regulation dan regulation di bawah setiap akta berkenaan dengan pendaftaran produk di Malaysia. So we have Registration of Pharmacists Act 1951, yang ni basically pendaftaran ahli farmasi dengan Lembaga Farmasi Malaysia. Kalau tengok slide saya tadi, dekat nama saya ada nombor tu, so itu nombor pendaftaran. Yang kedua, Poison Act 1952, and then yang kat bawah ni adalah dia punya regulation yang berkait dengan Poison Act lah. Okay. Next, we have Sales of Drug Act on in short soda, and also 1952, and then Dangerous Drug Act 1952, and lastly we have Medicine Advertisement and Sale 1956. So yang ni adalah regulation yang berkait dengan 5 akta yang dinyatakan. So kelima-lima ni dia berkait dengan pendaftaran produk, especially prescription and non-prescription with the MPRA Malaysia. Right, next slide. Okay, so untuk product registration kita ada dia sebenarnya di bawah regulation 7/1, Control of Drug and Cosmetic Regulation 1984. So di bawah regulation ni, CDCR 1984 ni actually di bawah Sales of Drug Act 1952 punya act. Okay. Di bawah CDCR 1984 kita ada category of product. So kalau tengok kita ada 2 jenis, pharmaceutical products and also other products. So untuk pharmaceutical product ni basically prescription and non-prescription, racun berjadual dan juga OTC, over the counter medicine. Dan kalau kita further divide, dia ada new drugs atau we call it new chemical entity, and we also have biologics and generics. And untuk other products, we have supplement, traditional and veterinary drugs. Okay, untuk traditional tu, kalau di bawah CDCR, nama dia adalah traditional. Tapi kat dalam DRGT, kita ada classification dia natural, traditional and herbal. So CDCR ni 1984 dia dah lama, sekarang sedang semakan dan due untuk dibentangkan dalam kabinet. So kita akan, since dia belum dibentangkan, maksudnya dia belum diwatakan, so kita masih follow old classification lah. Tapi kalau tengok kepada guidance dokumen MPRA, classification dia lain sedikit. Okay, next. Okay, ini adalah reference used for product evaluation. So untuk international reference, kita akan follow WHO guideline, ICH guideline, British Pharmacopoeia atau USP Pharmacopoeia. And also kita akan refer kepada Drug Control Authority, DCA. DCA ini adalah badan yang mendaftarkan dan meluluskan produk. MPRA is secretariat to DCA. So dia ada member of DCA ni lah. MPRA adalah sekretariat kepada DCA yang menilai dan DCA adalah badan yang mendaftar dan meluluskan produk. Okay, dan kita juga refer kepada PICS GLP guideline. So untuk DCA reference country ni kita ada dalam 6 atau 7 bergantung kepada jenis produk tu. Contoh dia US FDA, UK MHRA, European Medicine Agency, TGA Australia, Health Canada, PFDA Japan, Swissmedic dan juga HSA Singapore. Those are our reference country. Untuk regional references, kita akan tengok ASEAN guideline untuk stability, process validation and also variation. And local reference would be DRGD, Drug Registration Guidance Document. So ini adalah kitab atau Bible untuk pendaftaran produk di Malaysia. Dan kita ada additional MPRA guideline yang berkaitan dengan produk tersebut, kategori produk, DCA directive dan juga berkeliling lah circulars. Okay, next. Okay, ini adalah DRGD, Drug Registration Guidance Document. Ini adalah main reference for the registration process kat Malaysia, including QC, inspection, licensing, post-registration, semua ada kat dalam ni lah. And the scope of DRGD include information related to submission and application for registration, which is based on ACTD, ASEAN Common Technical Dossier. And also ada on the submission of application for licensing, manufacturing of importer, wholesaler, semua aktiviti MPRA diceritakan dalam DRGD. Right? So basically proses registration dia ada 6 in summary, walaupun dia timeline dia berbeza bergantung kepada jenis produk. So we have screening step 1, and then dekat screening ni submission through online system Quest 3+, and then nanti ada screener akan check the submitted document. Kalau lulus screen baru boleh proceed dengan acceptance of dossier and submission for sample for lab testing. This is only for the natural product. Number 3 is technical assessment. Bila dah dossier diterima baru proceed dengan technical assessment. Assessment timeline bergantung kepada kategori of product and then number of queries allowed. And then once the assessment completed, baru this evaluation report will be presented at the MPRA Evaluation Committee for untuk persetujuan before proceed dengan DCA meeting. So kalau lulus dekat MPRA punya level, baru proceed to be tabled in the DCA meeting for approval or rejection. Okay. And then last step will be the post-market surveillance for approval untuk dapatkan MAN number. Kalau untuk rejection, untuk pengeluaran surat penolakan lah. Okay. Alright, so data required by MPRA for evaluation for pharmaceutical product basically is in this slide. So untuk ubat baru ataupun biologics, you can see semua part tu diperlukan. Part 1, which is country specific original requirement, yes. Part 2, quality, also yes. Part 3, non-clinical data, yes. And part 4 clinical data, yes. So part 1, 2, 3, 4 ni sama dengan ICH CTD tadilah, sama. This is for new drugs, ubat baru dan juga untuk biologics. untuk generic, so since kajian klinikal tu dah ada, generic ni ubat yang bila innovator tu dah habis patent, so you can make a new drug with similar indication tapi generic. Kita namakan generic. So yang ni untuk generic you tak perlukan, kita tak perlukan non-clinical data dah. Sebab ubat ni dah lama di pasaran. Okay, clinical data kita hanya perlukan BA/BE studies. So you need to conduct BE studies at the NPRA accredited or certified BE center before you can proceed with the registration for generic product. Untuk yang new drug and biologic, memang waktu submission, evaluator akan have their checklist yang mana mereka akan check sama ada untuk non-clinical data ni, GLP studies or non-clinical studies ni conducted dekat MAD punya country or not. And then from that, because there are many modules involved, basically majority of dossier received during submission ni dah ada clinical studies. So this clinical studies is dia punya requirement tu lebih priority berbanding non-clinical. So if for this established dossier, evaluator will prioritize this clinical data during review compared to non-clinical. Unless produk tu baru dan it depends also on the category of the product lah. Alright, next slide. Okay, untuk kita ada new policy NPRA GLP study specific foreign inspection approach yang mana dalam keadaan tertentu NPRA ada terima produk yang hanya mempunyai early phase clinical studies, phase 1 or phase 2 without completed phase 3 clinical studies. So phase 3 clinical studies tu tak complete lagi, tak ada clinical study report. Yang mana under certain circumstances, this product are needed for registration to meet the unmet medical need ataupun interest of public health. Jadi perlu didaftarkan juga berdasarkan kepada keperluan ini. Jadi in that case, untuk yang non-clinical study not conducted in MAD, kita ada study specific inspection di mana kita boleh pergi untuk verify compliance of GLP tu. Contohnya kalau GLP tu dijalankan di China, clinical study dia mungkin multi-regional. Kalau ada kat Malaysia, kita boleh pergi untuk clinical study. Tapi untuk GLP, kita boleh pergi dekat China based on this policy. Okay. So this policy is risk-based for the purpose of product registration in Malaysia for NPRA and it's not a compliance program or for the MAD countries. Okay. Boleh. Okay, so ini kita baru endorse this paper last year, effective this year lah, January. So kita ada option tu, kalau clinical study dia tak complete lagi dan dia hanya non-clinical dekat non-MAD, kita boleh pergi untuk verify compliance dia. Next slide. So ini adalah registration pathway reform. Sebab tadi pun ada question tanya, boleh tak pendekkan timeline? Sama, NPRA pun setiap kali dialog kita terima soalan yang sama. Boleh tak pendekkan timeline? Bolehkah cepatkan registration? So kita ada a few pathway untuk bantu industri dan juga public di Malaysia. Yang pertama kita ada facilitated registration pathway, FRP ataupun in short reliance. So yang ni product approved by this, produk tu kena approved by stringent regulatory authority SRA ataupun WHO listed authority WLA. Ataupun pre-qualified and ASEAN Joint Assessment Initiative. So dia kena fulfill kriteria ni. Kita ada guideline dekat website lah, nanti boleh refer sama ada your product fulfill this FRP reliance pathway or not. And then verification dia akan involve 30 working days and then proceed with 90 working days untuk abbreviated review. So assessment report and list of Q&As to be provided, validity for 5 years. Usually the product cover NCE biologic, generics and biosimilar. Next, we have orphan medicine for rare disease. So this one specifically designed for the orphan medicine based on the list of rare disease in the Malaysian Orphan Medicine Guideline. Priority review is automatically granted 120 working days, validity for 5 years. Then we also have priority review. This is for unmet medical needs, life-saving medicine, interest of public health and emergency supply. Dia punya timeline 120 untuk NCE biologic biosimilar dan 100 hari bekerja untuk generics, validity for 5 years. We also have CR for unmet medical situation. Yang ni dia ada kriteria dia, registered at least at one DCA reference agency. Ini timeline dia, a priority review of 120 working days may be granted. And this is upon request based on the early clinical data, phase 2 data. Usually yang saya cerita tadi, yang GLP study specific tu falls under this category lah, CR. Clinical data dia tak complete lagi, dia submit yang early phase. And then we have to review limited data for clinical data tu and also non-clinical sebab dia tak complete. And we also have conditional registration for pharmaceutical product during disaster. So ni contoh dia hari itulah during COVID-19. Right, next slide. So ini number of product registered in Malaysia by MPRA by year from 2017 to 2024. Kita nampak yang bawah sekali tu prescription, and then yang warna coklat tu non-prescription or OTC. Warna light yellow is natural medicine, including traditional medicine. Hijau health supplement, and yang hitam tu veterinary drug lah. So basically in a year kita ada dalam more than 1,600 lah. The trend is increasing. So this statistic hanya untuk sampai 2024. Right, okay, next. Okay. So ini support, regulatory support untuk product registration kat Malaysia lah. Untuk local kita provide priority review. We also provide product meeting for the product that fulfill the priority review. So ada guideline dia dekat website MPRA, nanti boleh refer in detail. Dan kita juga ada priority GMP inspection untuk local. But it depends on your product lah, meeting the FRP or priority review or not. And then early scientific engagement, assist in product development pun ada, nanti saya cerita. Untuk import, kita ada juga priority review, product meeting the criteria, dia punya clause is kena meet the criteria untuk priority review lah. Kalau tak meet, kita takkan proceed lah. Okay. And then untuk import, kita akan bagi juga exemption from zone 4B storage condition with justification and hybrid flexibility on non-clinical, part 3 and clinical data. So yang ni based on the category of product yang saya mention tadi lah, yang ada slide nombor 19, faster access to medicine tu, yang ada FRP, reliance, offer medicine, priority review and all. Okay, untuk enter, next slide. Untuk enter Malaysian pharmaceutical market, key recommendation from MPRA, pertama sekali kena understand regulatory landscape, Malaysian regulatory landscape, and then kalau you nak proceed overseas, you kena tahu juga RA overseas tu requirement dia macam mana, supaya you tak sangkut. As we all know, Malaysian market is small compared to overseas. So kalau you nak market dekat Malaysia je, mungkin it's not sustainable. Business is about sustainability. Okay, first, you have to understand the regulatory landscape, identify the local partners, familiarize thoroughly with the registration requirement, and ensure compliance with GMP and BE requirement. Banyak company yang start kat sini lah, GMP and also BE. BE untuk generic product. And then engage with local regulatory consultant to navigate approval efficiently. Okay. Number 2, adapt to market needs. Customize product portfolio to meet Malaysian healthcare priority lah. Itu yang sebaik-baiknya dan seelok-eloknya. consider affordable pricing strategies given the market are competitive and also price sensitive market in Malaysia. And also explore innovative drug formulation or biosimilar tailored for Malaysian market. So ini masih ada gap sekarang dekat sini. Number 3 is on the technology transfer, sharing of technology, manufacturing practices and know-how from one company to another, collaboration, and lastly, partnership and business strategy. So these are the key points you have to take into consideration before entering Malaysian market. Next slide. Okay, ini kita ada this pre-submission meeting, we call it PSM, pre-submission meeting, where we give advice regarding the QSE aspect prior to submission for the registration application. So kalau you're not sure, you can just write an email through pre-submission@mpra.gov.my. Dia ada guideline juga, boleh refer to the guideline. So we'll see whether we can arrange a PSM meeting between the company and also the relevant section or stakeholder within MPRA to give some advice to guide you. All right. Okay, I think that's all. And my last slide is on the references. Kalau tak sure, just click on these references lah. Okay, any question?
Thank you very much, Margarita, for sharing your knowledge and expertise with us. We truly appreciate the valuable insights shared on the regulatory expectation for GLP studies in pharmaceutical products. I believe the sharing has provided our participants with a better understanding of the regulatory perspective and the importance of generating quality and reliable data to support the registration of pharmaceutical products. Ladies and gentlemen, we are now moving into our industry session, which will provide us with valuable insight from the industry perspective. Mr. Tim dulu. Okay. For this session, we are pleased to have Dr. Ahmad Razlan Rosli from Bayer CropScience, who also represent Chemical Industries Council of Malaysia, who will be joining us live online. Dr. Ahmad Razlan Rosli will be sharing his insight and experience from the industry perspective, particularly in relation to GLP and industry expectations. Although Dr. Ahmad Razlan Rosli is joining us virtually today, we are very pleased to have him with us and appreciate his time and contribution to this symposium. Ladies and gentlemen, please join me in welcoming Dr. Ahmad Razlan Rosli from Bayer CropScience.
Assalamualaikum. Can everyone hear me? Am I audible?
Please proceed, Dr. Rosli.
Okay, all right. Okay, great. Thank you. Okay, Assalamualaikum, and good afternoon to everyone. Thank you for National Standards Malaysia for this opportunity to have this industry sharing session. Unfortunately, I couldn't be there physically due to personal reasons, so I apologize for that. But thanks for organizers for making this virtual session possible. So I am a residue and human safety scientist for Bayer CropScience. So I'm based in Asia Pacific. So in a nutshell, my responsibility is to evaluate product safety and human safety for crop protection products that are registered in Asia, including Malaysia as well. And if people in the audience are not familiar with Bayer, so we are a multinational company based in Germany. We have three major divisions or businesses. So we cover pharmaceuticals, consumer health. So anyone who is familiar with Baroka or Redoxons in the pharmacies, we are familiar with those products. And of course, crop science, where I am residing, where we are mostly targeting the evaluation of our crop protection products and our seed and traits worldwide, and making sure that our products are safe for farmers globally. So with that, I would share my presentations on industry expectations. I would like to give a more, I would say, broader view of the topic. We will more focus on GLP eventually, but I will see what kind of developments that are happening in Asia and also globally, and how that would eventually lead to the requirements of GLP and what is expected from us as industry, and what we are hoping to engage with national authorities, with government agencies, etc., across the world. So in this first slide, I wanted to give a broader view of what would be the target for most industry, which is the journey to food safety. So if you look at this pipeline that I've presented here in a schematic image, is to represent all the different areas that we cover during our evaluation. And eventually the target is to achieve maximum residue limit. So basically a tolerance for a active ingredient or a pesticide for a crop. So for example, here we have, let me see if I can have this. Just gonna take a highlighter or a laser pointer. Sorry. Okay, I think. Okay, never mind. So basically what we have here is like tolerances for different AIs, which is set at a parts per million or parts per billion kind of concentration, where we set a tolerance for what will be the tolerance limit for these residues to be present in your food commodity, in your supermarkets, etc. So that will be our basically target end goal is to achieve a tolerance whereby we've conducted various safety assessments, not just us as industry players, but also by the global regulators, for example, by GMPR and WHO, which is under the GMPR, which is under WHO arm for scientific evaluations for pesticide residues. So we ensure that the consumers or the public would be safe from residues that you find in foods or in other food commodities that you find in the supermarket. So we end up having a tolerance, but before that, we first define our product label. So for example, if you have a pesticide that includes a use pattern for a country. So for example, what is the dose rates, etc., how many applications you apply in a crop, similar to what you have a dose for, pharmaceutical products. You have a similar case with pesticides. We investigate what would be the nature of pesticide residues, how would that influence the safety in our safety studies. So we do a lot of metabolism studies in plants and animals. We define the residues because there will be a lot of different residue components within the compound that is evaluated by many regulators. And then we go into the essentials, we will call it the safety studies. So we do conduct GLP residue studies, and these are done according to OECD test guidelines, and also by other higher regulatory bodies, by the US, by EPA, and of course by the EU with the European Food Safety Authorities. So we have various different studies that we conducted to ensure that our products are safe before it hits the shelves in the supermarkets. We do like field residue studies, storage stability, rotational crop, where we have subsequent crop cultivations within a couple of seasons, processing studies if certain commodities are leading up to a processed product, for example, like orange juice, apple juice, etc., and other different methods of evaluations and studies. So we do a lot of GLP studies, which I will go into more in depth later. And then we do conduct a dietary safety assessment, where we look at the results from residue studies, what are the residue levels found in those studies, and see whether those residue levels do fall within the safety limits of the toxicological reference values. So those are values that are set by toxicity studies, so acute and subchronic studies that are done either by us, the company, or the manufacturers, or by the international regulators like JMPR, WHO, etc. So eventually we want to ensure that if we do a proper science-based evaluation and do studies based on GLP requirements, we ensure that consumer safety and essentially global trade flow is enabled, because there are many countries that rely on scientific-based studies to show that these products that apply in crops during the commercial cultivations do end up safe when consumers eat the food from the supermarkets or from restaurants. And why science-based decision-making is a strategic priority in Asia is because there are a lot of different underlying, I would say, underlying problems that might occur from different countries, and this is due to a lot of political ambitions or different problems that result in a lot of these bans or blanket restrictions that happened in many of the countries that we see in this agricultural space. So we see a lot of bans in Sri Lanka, also in Pakistan, where there is no scientific reason why these pesticides are banned, purely because of its hazard, and not by other means of evaluation. Like, for example, here you see that there is a lot of evaluations of risk assessment, which we look at exposure level. So risk assessment is not just based on looking at hazard, where it is also important because inherently chemicals are potent and hazardous by nature. They need to be in order to target a pest or a disease in the field, but you will need to evaluate that hazard or that assessment based on the exposure, how much exposure or the level of exposure that you are being done in the field or in when you're eating the commodity or the crop. So it's important to include that in the equation. And a lot of our advocacy in the industry involves on science-based risk assessment. So looking at abolishing hazard cutoffs, and then even if you're looking at highly hazardous pesticides, HHPs, which are a controversial topic in many countries, including Malaysia, we will want to also evaluate more scientifically on these evaluations on HHPs, and not just looking at the hazard component, but also looking at exposure component, where some companies like us, Bayer, we have the luxury of conducting all these studies worldwide to ensure that our products are safe generally when we apply them in the crop. So what are the barriers of science-based decision-making? So there are a lot of different barriers in many different countries. And of course, there are quite a few of them. Authorities always have preference or simplicity over complexity. And this is highly understandable because chronically authorities around the world are understaffed. So this assessment evaluation are inherently very resource intensive, so require a lot of expertise, a lot of engagements with many different experts around the world to ensure that risk assessment done not by purely national agendas, but basically pure on international best practices and based on international guidances by WHO and OECD. Another point I will highlight is the local generics. So there's a lot of highly influential industries which focus on local generics, and this is also understandable in a sense for some countries, including Malaysia, where there is professional treatment, and obviously with lower capacities and lower capabilities in the R&D sectors, they don't have the expertise to do a thorough safety study or safety assessment on the product they're going to register in the country. So governments or authorities would give more incentives for local generics. And there is a drawback for that. They will, you need to compromise on some of the thorough evaluations that are done on those products and the R&D that is lacking would also limit how the product can be traded or can be exported outside the country, because some countries like high regulatory countries like the US and Europe are depending on a thorough scientific evaluation of those products to make sure that those products are safe when they hit the borders in their own countries. And another I would like to highlight before we move on is basically the local knowledge gap. So the path of least resistance. Risk assessment is a like I mentioned before, it's a resource intensive program and practice. So to build it from scratch in the country requires a lot of resources and infrastructure. So which I will present later. There's a lot of different advocacy efforts that we're working on, capacity building in some of the countries to make sure that we work more towards risk assessment. And this, of course, includes the component of GLP, which we'll get on shortly. And some important notion about the benefits of science-based decision-making. Again, farmers and environment, because you have a scientific approach on the assessment, you'll be able to identify where the real risk is, and not just pure guessing or just pure blindness of where the risk is based on hazardous part of the equation. So we look at the exposure part, as I mentioned, impactful stewardship. So when you have a proper risk assessment guidelines, you'll be able to tailor those assessments based on how the risk is appearing. For example, if a certain product is applied in Europe and in Asia, of course, there will be different commercial practices, agricultural practices that are different in those countries. So we need to employ different PPEs, so protective clothing on the operators of those pesticides, and that has to be tailored to the country. Sometimes certain things are done in Europe might not be working well in for example, Indonesia, where it is more humid, more hot. You don't expect them to wear full clothing when you're applying pesticide in the rice field, for example. So there are a lot of different tailored stewardship around that. And then regulators do promote trust scientific risk-based evaluations. So you'll be able to have products that are traded across the world that are trustworthy and reliable and scientifically defensible when it comes to litigations, lawsuits by the public on the use of or overuse of pesticides. And we've seen many in the news lately about overuse of pesticide could lead to other problems, but it is due to the off-label uses of the responsible operators instead of the actual safety profile of the product itself. And lastly, the industry players, the role is, of course, the strength of the industry would be the R&D part, the pipeline. They are well positioned to produce ever improving products that can replace all chemistries that have high hazards profiles. We want chemistries that are reliable, safe for environment and for consumers. And then, of course, the strongest science to hold within the industry. We do provide a lot of engagements and training to the governments and the government agencies to make sure that we have the same awareness across all the different players in the industry. And what is the connection? What is the sole purpose of having this first few minutes of introduction on science-based regulations and how it is connected to GLP studies? Is that every single checkpoint in every single bullet point in this presentation, or in this slide at least, ensures that GLP is an essential part in every single process when it comes to food safety. Because if you have a highly compliant and highly regulated standard, when you're doing any of your safety studies, for example, residue studies, where you evaluate what will be the residue levels in the crop, you'll be able to create an assurance that these products are safe and done in a proper systematic manner. And then when it's being regulated and monitored by the authority, you can able to have this trust on the data package that's presented by the pesticide manufacturer, and also evaluated by all the different authorities across the world. And essentially, as I mentioned before, the first slide is the tolerance levels, to ensure that the tolerance levels are within the safety limits, and that would ensure the safety for the consumers or the general public. So essentially what's important is to do a thorough dietary safety assessment, making sure that before that, when we do the safety studies, we are doing them under GLP, so that these studies are done according to international standards, and have proper peer-reviewed quality assurance, so that these data are trustworthy and defensible if there's any other challenges that may occur down the line in the product lifeline. Just want to give a very brief overview about the risk assessment or requirements. So for some of the countries in Asia, so looking at the regulatory landscape and how these countries are moving towards more risk-based assessment or GLP study requirements. In 2016, we found that in the agricultural space, especially in crop protection, there's a lot of the countries in Asia Pacific that are still under developing their policies on risk assessment and GLP. You can see majority of them are within the bottom half of this group, and only a couple of them, like New Zealand, Australia, are within that risk assessment status. But 10 years later, we do see a lot of shift when it comes to development within these countries. We see a lot of Southeast Asia countries moving up into the ladder, trying to dip their toes into the risk assessment pool, and trying to get more gist about GLP requirements and what's needed to ensure that the data that's presented is reliable. And this is due to many factors. One of them, of course, still are more global demand for trade. So there's a lot of demand for countries like the US and EU require a lot of this trade of these specialty crops in Asia, for example, like durian, dragon fruit, papaya, that are essential for the usage in those countries in Europe and US. And these crops cannot grow in those countries. So they require those like durian to be grown in Malaysia and then be exported to the US and Europe, for example. But of course, they will need a proper safety evaluation, making sure that our crops are grown based on international standards and evaluated based on international best practices. And to highlight a few global trends in GLP residue study demand, I think GLP demand is similar across different industries, but for me, I always associate GLP residue data as a global passport for crop protection products because it gives your pesticide product global access to a lot of different markets across the world, especially in those high regulatory countries I mentioned in the US and in Europe, where there's higher demand of science-based evaluations. So Codex GMPR, which is under WHO arm, EU, Japan, Korea, Canada, all require GLP residue data, and these are essentially high markets for all these traded commodities, especially in Asia, specialty crops, as mentioned before. So they will want the tolerance to be performed according to their standard and the WHO standard. So rarely you see non-GLP data get into their markets, in their submissions because they will rely on GLP data to be there, so that it's trustworthy and reliable for them to evaluate their products there. EU and US and OECD always set the bar. They're always raising the bar when it comes to setting up better evaluations on pesticides, especially with the growing demand on organic farming, and the NGOs are pressing on the need for less use of pesticides in the field. So the US and the OECD and the Europe are raising the bar in the amount of studies they require, amount of toxicity studies they want you submit. And this gives countries a lot of burden, extra demand for trials and studies to prove that your products are essentially safe. And this raises up the value in Asia as well, especially when most of these crops or high value crops are coming from Asia in most cases. So you have more trials in more geographies, and rising technical expectations and capabilities. So you have labs in Asia raising their level of expertise and accreditations, because they will need to raise that demand for expecting those markets to be fulfilled. And national capacity is a bottleneck, as mentioned in Asia. There is an emerging market within many regions, including in Latin America, where similarly with diversity and all the different specialty crops, tropical, subtropical fruits, we have similar landscape with Latin America, like namely South America, Central America, where you will need to provide a lot of data reliable studies so that you'll be able to prove that your products are safe. And why does it matter to all of us is because the bottom line is if your data is non-GLP, it is harder for you to defend your data against the higher regulatory bodies, especially. And of course, when you are creating a tolerance for trade, it's even more difficult to justify, whether your product meets the safety limits, because if you are not running GLP data, you don't have a scientifically valid data study that could be essential for you to do the evaluation, or a third party, player doing the evaluation for you. So, but later I'll present some information on, not all non-GLP data is non-essential, but GLP data is definitely giving you a better safety net when it comes to defending your product against different markets in the world. I just want to briefly touch on the QA. So a QA is an important part of GLP, quality assurance. So it gives you an independent system of checks monitoring on your GLP studies. So it confirms, follows whether the study is done according to SOPs, whether your equipments are calibrated according to GLP standards, etc., and making the data basically more reliable and reproducible. And this is essential because in most cases, non-GLP studies don't have the QA component. So that practically makes the non-GLP even less defensible, because you have this QA component of independent third-party evaluation, where you'll be able to review all the protocol, the study planning, the actual results, the raw data, and also the final reporting, all the way to providing that QA statement where you've been verified to be GLP compliant. In context to our residue studies in agriculture, so one of the few QA checkpoints that we do is looking at study protocol, as mentioned. So looking at study setup, how is it essential, whether the trained personnel have a proper training, calibrated equipment, etc. Test item application, calibration of equipments, whether you've done correct dosage, proper labeling and proper logging of information and application information in the field. Sample collection also is very essential, because you want to ensure that the sample are collected based on best practices. There's no cross-contamination, and everything is being audited and logged and labeled. And storage and transport, there's unbroken cold chain when you are transporting your field samples from the residue studies to the lab when you're doing the actual analysis, making sure that there is enough, for example, dry ice, to make sure that those samples are safe and not being tampered with low temperatures or high temperatures, etc. And then essentially at the end is analysis and reporting, validated methods, QC of sample, accurate traceability, etc. And those are important as part of the study validation for residues. And going back to non-GLP and why it matters is, again, as I mentioned, top regulators only accept data that is proven reliable. QA provides that better assurance, so to speak, so that your data package is highly defensible. And why it matters non-GLP is that the same principle might keep the data trustworthy and reproducible. Non-GLP studies nowadays do have some option to run a QA. Of course, this will add extra cost, but you'll be able to do all the other GLP requirements. For example, like proper logging of information, etc. But you'll be able to do that QA where the peer review will be able to give you a report that says that your data is reliable, for submission. So QA doesn't add credibility. QA does add credibility when GLP is not required. But it does require additional costs due to that extra peer review evaluation. This is a pretty busy slide, so I won't dwell it too much. I just want to highlight some important key points on the differences between GLP and non-GLP from industry perspective when it comes to crop protection studies or residue studies, as we say here. As I mentioned, GLP studies are widely accepted by major high regulatory bodies like the US, EU, and of course in the WHO level. Non-GLP studies are more commonly applied in developing countries. They are still developing risk assessment and developing their own policies around GLP. So looking at some of the Southeast Asia countries here, they can argue that some of these countries are already on the verge of have developed their own GLP policies, but some of them are still on the verge of developing policy-wide guidelines on GLP on crop protection products. And quality system oversight, as I mentioned, QA is an essential part of GLP studies. Non-GLP, it's not essential to have QA monitoring. Because it lacks the external GLP compliance, and that it lacks that oversight. But of course, some non-GLP studies I mentioned do perform independent QA as a side program to make sure that their data is reliable. Data integrity obviously with GLP studies, you have more data integrity because you have strict record keeping, traceability, chain of study of test substances, archiving, etc. GLP, there is no mandated guidance on non-GLP and traceability and data integrity. It relies solely on internal practices rather than following a formal system like a GLP standard. So basically it's based on the lab or the company or the contractor who does the studies, whether they want to follow the internal guidance on that. And technical controls with technical methods, calibrated equipment, etc. for GLP and non-GLP, everything is more flexible. Standard lab certification, for example, ICO standards would require to do proper calibration and validation, which is already sufficient for non-GLPs, but of course it's not sufficient for GLP. And cost would be probably the number one drawback for most countries going to non-GLP requirements is because on average higher cost is expected for GLP studies because of the QA oversight, because of the independent evaluations, and of course the GLP certification testing facilities fees also has a lot of infrastructure that need to be covered. So so the cost can be more than 2 or 3 times the cost of the non-GLP study. So that's why there is a lot of hindrances on countries to go or embark on the journey of GLP infrastructure in the future. So what does a GLP or future-ready GLP ecosystem looks like from an industry perspective in agriculture or in crop science? Essentially, it boils down to 5 players or 5 partners who share the same outcome, is to ensure that we have a trusted and usable data to be accepted globally, defensible, if there's any challenges that might occur down the line. So regulators have to play their part to be very clear, tangible on expectations, and not just go with the crowd and making sure that they follow their own pace in developing their own GLP guidelines and risk assessments. The industrial responsibility will be on the sponsorship and the ownership, the accountability of the product safety and the human safety side, making sure that we are not just here to sell our products, seamlessly without any consideration for human safety. So food safety and food security is important. I can say at least from our side, from Bayer and the major industry players, the other companies like Syngenta, BASF, FMCs, Corteva, AgriSciences. But we expect the other industry players to play a very important part as well. The CROs, the contract research organizations that are helping, there are third-party basically contractors who does study for us in the field. They have to have that proper expertise and robust execution of those trials to make sure that they are done based on GLP requirements. And of course, you have the QA part of it as independent reviewers that does all the peer reviewing assessments for the GLP studies. And of course, digital and IT is an important part as well, because we are moving towards more digitalization of our data. So we have more validated systems. GLP are now moving more towards digitalizations. So cybersecurity, data protection is really important to give us more long-term, sustainability on data protection and data availability. Just a couple of slides left. And from our side, industry call to action is essentially to build a GLP system that produce data that can be safe, that can be trade, and it can be innovated globally. So we want to design data that has high integrity and also give us a better digital readiness, so they are available whenever we need it, especially when moving to a more digital space in the future. Regional capabilities. So we want to strengthen the CRO capability, the labs and the companies, the third-party players that are doing the studies for us, making sure that they also are raising their bar of safety and requirements. And collaborate for acceptance is we want to use this advantage or leverage on this authority industry dialogue engagements to further improve on harmonization, improve our GLP capabilities in the countries. And this will give us a more trusted science when we try to showcase our products and making sure that products do work in the field, and essentially the farmers are benefiting from it at the end. And talking about collaboration, I just want to, for my final slide, just to share on what we have done in collaboration with governmental agencies and authorities in Indonesia and Vietnam in the past month in July, where it is an industry-led initiative, capacity building, where we trained governmental agencies that are responsible for residue studies and for GLP requirements. We want to train them how we do perform the residue trials in the field based on GLP and OECD guidance, and also the analytical part as well in the lab. So we did quite a few trainings where PropLife Asia, which is the industry association mixture cocktail of different industry players, for example, from us, Bayer, Syngenta, Corteva, BASF, etc. And we provide this close hands-on training with authorities, and we be able to provide them with essentially the outcomes of providing a better science-based assessment and proper following the proper processes of running a GLP study in the field and in the lab. So we are doing all this capacity building exercises constantly with the national authorities, with the governmental agencies in different countries in Asia, to raise the bar, to raise the safety level of our products and the safety level for our consumers, so that essentially when the farmers are applying the products, they are not endangering themselves, and they are applying them based on label uses and not off-label uses, and giving more assurance to the consumers that once they are buying their food on the supermarkets, they can ensure that they are safe and not in a level which can determine their health. And with that, I end my presentation. So thank you for listening. I hope that I don't go overboard with the timing. Yeah, and if any of the questions, hopefully I can leave on my email, and people can just reach out to me, or want to know more about our company, Bayer, you can go to our website and see what we do as a company. Okay, thank you.
Dekat situ. Di sini terus. Ha? Yes. Sekejap. Thank you, Doctor. Thank you, Doctor, for the very comprehensive sharing. I think I like the last two slides where you have actually summarized it between the collaboration between us, industry, CMA, RAs, and etc. And also your experience in having GLP in Bayer, and also the one that you have summarized the differences or the difference between the GLP and non-GLP. So I think with the slides that you have actually shared, we have obtained a lot of information here. So thank you very much, Doctor, as time is flying. So we will not accept any, or do we have question, guys? No?
No, okay. So thank you very much, Doctor, for the sharing. Hopefully to see you on board again.
No
Thank you.
problem. Thank you very much. Thank you for the opportunity.
Thank you very much, Dr. Ahmad Razlan Rosli, for the insightful presentation and for sharing your valuable perspective from the industry. We truly appreciate your time and contribution to today's symposium. Thank you once again, Dr. Ahmad Razlan Rosli. Ladies and gentlemen, we shall now proceed with our next industry session. This session will further explore the practical aspect of conducting field trial studies under GLP, including key consideration from study, planning and execution through to documentation and reporting. Without further delay, let us welcome our next speaker, Mr. Tim Chin Min, Task Facility Manager from ALS Technichem Sdn Bhd, who will be presenting on field trial studies under GLP from planning to reporting. Ladies and gentlemen, please welcome Mr. Tim Chin Min.
Nak kena pinjam, kena pinjam tali. Tali ni. Mana-mana. Okay. Sebab nak kena sangkut ni, nanti orang dekat dalam tu tak dengar, Tim. Okay.
PowerPoint ya.
Morning session. ALS kan ni?
Saya ada pendrive.
Thank you. Yes.
It's okay. Okay, PDF. Ya, satu saja. Saya kontrol. Okey.
Boleh. Ya, antara media yang mana ni?
Apa?
Media yang mana? Yang ada video.
Tak ada video, animation saja. Boleh start terus.
Share screen.
Sampai saya sudah tengah hari. So tadi ada orang dengar orang cakap sudah lapar. So yeah, I try my best supaya bagi cepat habis lah. Okay, first of all, I would like to thank Jabatan Standard Malaysia for inviting ALS as one of the speakers. I'm glad to be here to share on today's topic, field trial studies under GLP from planning to reporting. But before going to the topics, I would like to present a brief introduction of ALS. ALS, Australian Laboratory Services, is a global leader in providing laboratory testing, inspection, certification, and verification solution. In Malaysia, we provide testing on the field as illustrated in this picture: food, agro,
environmental, soil, water, air, including the emerging contaminants such as PFAS, biomonitoring, microplastic, biotech analysis, tribology on lubricant analysis, pharmaceutical, and also semicon. ALS present in 70 countries with more than 22,000 staff worldwide. And this slide shows the distribution of ALS staff. In APAC, we have near to 4,000 staff. Let's just zoom into Malaysia. Di Malaysia, kita ada 5 ISO accredited lab dan 1 GLP lab. Shah Alam is our HQ, accredited for ISO 17025 since 1998. Pride, this is our Pride GLP lab and also ISO accredited. Johor Bahru, accredited since 1994. Kota Kinabalu, accredited since 2011. And in total, we have staff strength of 213 in Malaysia. As I mentioned, this is our accredited 17025 lab. And we have 2 certifications. One is on OECD GLP, on pesticide chemical testing and residue studies. And GMP, certification for safe, sustainable animal feed. Our laboratories are recognized by various regulatory authorities in Malaysia, including MOH on food, COVID, and also pharmaceutical by MPRA, Jabatan JAKIM on halal testing, and recognized by DOE and DOSH. And also we awarded IKM Laboratory Excellence Platinum Award. Yeah, we expanded our category area of expertise to residue studies last year. This picture shows actually the presentation ceremony by Pengarah Kanal Standardisasi Encik Miza. And as part of ALS commitment towards knowledge sharing or also responsibility to the scientific community, we periodically publish technical flyers, articles, and also we conduct at least 1 webinar monthly on several topics. By scanning this QR code, you will be directed to our website, which contains all these technical articles and also webinar invitation. Perhaps you can take a photo for your future reference. Yeah. Okay, without further ado, I will go into today's topics and I will cover on the following contents and also illustrate the actual study flow from beginning until the end. First, on the trial design, plot selection, and plot layout. Followed by application of test item. Test item means the pesticide formulation. Sampling of test crop. Next will be the transportation and receive in the lab. The lab will do the sample preparation, sample extraction, and determination of pesticide residue using instrumentation techniques. Field trials is also known as crop field trial, supervised field trial, and supervised residue trial. The purpose of field trials is actually to determine the magnitude of pesticide residue in raw agricultural commodities, including feed items. And this residue data actually is required for the pesticide registration to ensure that any residue pesticide at the time harvest does not exceed the MRL, and recommend a suitable waiting period between the last application or the last harvest, PHI, so that the residue of pesticide would not exceed MRL. And field trial is always accompanied by the lab work. So the test facility need to ensure that a workable method is available to analyze pesticide residue in the crop. These are the guidelines that we use for field trials. OECD document number 1, 6, 509, 506, and also GP4. I believe many of us already very familiar with the study plan for the lab-related study plan. So now I would like to emphasize on the field trial information. So study plan must contain critical good agricultural practice information such as test item technical information, the IUPAC name, CS number, type of formulation, target test crop, planting methodology, the variety, field, number of location, plot size, or number of plant required including buffering zones. Test item application information, method of application, application rate, spray volume, internal and number of treatment. And lastly will be the collection of the test crop. There are 3 types of trial design. The first one is residue decline study for perennial crops with multiple test crop sampling. That means we apply last application applied at the same date, and we harvest at a different interval that which it will give the multiple sampling. Perennial crops is a plant that provide yield for multiple growing season without needing to be replanted. For example, oil palms, solanaceae, like chili, cucurbits, like melons or cucumber. Reverse decline study. That means last application done on several days, and then we harvest at the same time to give the different intervals. It's applicable for annual crops. Annual crops is the plant that complete their life cycle in one single growing season. And for example, gramineae, paddy, and Brassica cubies. Many-tailed residue study, that means only mostly applicable for pesticide, is only one interval, one sampling. So the GLP field trial must have the conducted according to the common practice, agronomic practices, such as in glass house or in an open area, the type of irrigation and crop varieties. Typically, it's more on the typical varieties. Also depends on the maturation date period for the crops. Location, fertilizer application, and any practice that will contribute to the highest residue. Trial site selection. The field trial must be conducted at the region where the crops are widely planted. So the distance between each trial site must be at least 30 km apart. And trial at same location at different planting dates, at least 30 days interval are allowed. And second trial can be considered a separate trial if the second trial different in terms of agronomic practice. For example, using the rain shelter or versus open area is a different practices. And even though the trial is conducted at the same location and same growing season. Trial can be repeated at the same trial site on different season. However, the exact location of the test plot must be different from the previous trial. And the field trials, the site selected must be free from the pesticide study for at least 3 months. And other factor to be considered when selecting the field trials will be natural factor, drought, flooding, strong wind, pest and disease factor, and also human factor, theft. So kita cari lokasi supaya bila nak harvest, buah itu masih ada. Tak dicuri orang. Plot selection. So plot selection, the criteria based on the uniformity in crops, pests and field condition to meet the trial objective. And must be large enough to represent sample size, simulate the current commercial farmer practice. And actually kita ada guideline berapa crops yang kita perlu harvest. Untuk cili 2 kg dan berapa biji durian, ya ada spek dia. Kalau tak meet, mungkin PB akan tolak. Durian jemput, okey, tak ada masalah. Able to accommodate the sample required for lab testing, especially control. Control kita perlu banyak sebab kita mungkin akan guna untuk buat QC, buat spiking. So control normally kita perlu banyak. So parameter to be considered untuk padi 30 square meter, jenis tanah, the plot maintenance may include fertilizer, plants growth regulator, and ini adalah untuk uncontrolled plot, that means not treated. So uncontrolled plot should be located in the immediate vicinities of the treated, so that cultivation and cropping take place under similar identical conditions. Ensure that plots are adequately buffered and separated. There is a guideline, the distance between the treated and the untreated. So higher established barrier, shielding is acceptable, and also higher elevation compared to treated. Should be not located in the wind path from the direction of the treated plot, and water movement for paddy. Make sure surface water discharge flow from the treated plot does not enter into the control plot. Tak ada parameter ini kalau tak control dia akan memberi cross contamination. Ya. Kalau control plot contaminated, artinya study itu tak valid lagi. Okay, this one on the application plot sudah pilih, sekarang kita bagi application. So application parameter information that require spray volume, application rate, weight of AI ataupun weight of test item per hectare yang diperlukan, number of application and also number of interval. To achieve higher possible residue, application must be conducted based on maximum number of application, highest rates established within the crops, and shortest established application intervals. Application of test item must be conducted similar or and emulate with the agronomic activities in the farm. So equipment used with known pressure, spray capacity, and suitable nozzle for the application. So equipment need to be verified before the application. I would like to highlight this slide on the use of drones. So kita pun boleh guna drones untuk sembuh racun. Application equipment. The first one at the left is the mist blower. Sprayer for the higher vegetation, mainly apply to the fruit tree. Dia boleh sembuh sampai 10 hingga 15 meter. Tapi kadang-kadang durian lagi tinggi dari itu. Masalahnya durian itu lagi tinggi. Not suitable for herbicide. The center is a typical sprayer. knapsack is operated manually ataupun guna motorized pump. So there's nozzle yang digunakan, typical nozzle, flat fan, full cone, full hollow. Untuk carbon dioxide sprayer, this sprayer dia bagus untuk deliver precise and on the localized or defined area. So it's very good for bioefficacy study. Equipment yang kita guna, as I mentioned, is calibrated and verified according to the metrological traceability requirements. So we can see equipment yang guna weighing balance ataupun micropipette to prepare the spraying solution. Metronome is a device produce rhythm beat per minute. So applicator akan guna ini untuk to control their movement, spray movement, so that delivered consistent pesticide to the crops. Kalau applicator jalan cepat sangat, so the plant will receive more droplets and maybe cause runoff. Kalau dia lambat sangat, kalau cepat sangat mungkin plant tak kena. Droplets itu tak kena dengan konsisten. Standard weight to calibrate our balance and also sprayer calibrator. The activity in the field actually can be simplified to the three categories. First is the preparation of test solution, which will involve in test item preparation, the test item measurements. Second will be the sprayer verification. So determine the flow rate, calibrate, and application. So kat sini, many recordings happens here. So many recordings happen here. So the personnel need to ensure that all the data integrities and also data traceability are upheld. So sini sangat critical. Mungkin ada banyak rekod yang diperlukan kat sini. So best practice in test item application. Adherence to GLP, always verify calibrated apparatus and conducted during favorable environment in terms of safety, including proper PPE for applicator and weather and application timings. Use only clean water for the mixing of test item. Record all necessaries. Kat sini kita boleh nampak study personnel pakai PPE yang penuh. So bila saya tengok ini, saya boleh rasa sangat panas. Sebab dia spray, especially padi, kita buat application under the sun. Ya, tapi keselamatan adalah keutamaan. So lepas application, kita sekarang adalah menuai, harvest. So crop taken from field trial should be of the whole raw agricultural commodities, RAC, as it move in commerce. For some crops, they may be more than one RAC. For example, field corn, jagung, kita ada grains, kita ada fodder. Dia dua berlainan. So guideline yang guna OECD 059 dan juga GP4 mention. So general procedure, test crop must be collected without bias, representative of all portion to be of the crops. So avoid edges and end of the plot to be influenced by sprayer overlapping. Normally kat ends mungkin akan ada overlapping. So kita tak ambil kat penjuru. Prevent cross contamination between plot in terms of procedure, PPE and equipment use during sampling. Record all necessary information during sampling and label and pack test crop accordingly. Kita boleh nampak dari gambar the first on the top photo segregation of test crop. So kita segregate based on the plot. So proper segregation, even during harvesting, kita pun pakai proper PPE. Kita tuan-tuan puan-puan boleh nampak study personnel pakai apron. So dia sampling control dulu. Bila habis, setiap kali mereka akan tukar PPE untuk prevent contamination. Even packaging, kita pack using double plastic bag. Collection of test crop must be conducted based on approach study plan, based on the PHI. So for pre-harvest interval kosong, PHI kosong, test crop was collected after 2 hours of test item application. So do not conduct test crop collection during raining. Always, as I mentioned, always start sampling with untreated control. Kat video kita boleh nampak ini study director. Study director during harvesting. Actually dia ikut commercial practice. Untuk kubis commercial practice, bila sudah potong akan remove daun-daun yang layu. Perlu remove dia commercial practice. Even ada mention kat OECD guideline. So dia follow. Yang kelapa sawit susah sikit lah. Satu tandang, satu bunch 40-50 kg. Test crop storage, okay. Timing and also distance, do not fail. Sometimes the timing and also arrangement do not allow the sample to be sampled immediately and sent to the lab. So for this, to cater this, actually we have a freezer to store at the test field. So test facility need to ensure the proper precaution and integrity of the harvested sample test crop. So test crop must be stored under -18 degrees. So kita ada chart daily temperature using data logger, and from the picture we can see freezer. This freezer is actually located at the field trial sites. Proper packaging and arrangement in the freezer to prevent cross-contamination. The freezer actually lock, so hanya study personnel saja boleh access, and also the freezer are calibrated or verified using the thermocouple, temperature mapping kita buat, dan data logger, monitor daily temperature. So test crop transportation. So to transport test crop, actually always in the cooler box with ice for duration 12 hours. If the duration is more than 12 hours, kena guna dry ice. So special precaution is separation of untreated with treated. And during transportation, study personnel must record, buat semua record lah. That means the full trail of the sample from the site, from the collection until sampai lab. Mungkin di tengah-tengah ada transit because we have facility, but not facility insya-Allah kita akan berhenti kat sana sampai baru hantar ke Prai. So semua information ini kita akan record in our in-house form, change of custody, and also we take the photo of all location with the GPS and timestamp here. So kita boleh trace kat mana location, pukul berapa, dia kat mana semua. Information ini sangat penting untuk GLP. Okay, now we go to the lab. Bila sudah harvest akan sampai lab. So test crop to be analyzed actually based on Codex, OECD, or GP pun ada mention. So for example, sweet corn, kernel crop with husk, palm oil, you kena separate to cake, mesocarp oil, and kernel cake, kernel oil, fruitlet. 5 bahagian untuk kelapa sawit. Compulsory. So untuk durian, tahu mana? Sampling. Isi saja. Salah. The whole fruit. Yes. Durian the whole fruit. So sample preparation, as we know, sample preparation adalah process the sample into the test portion to be representative and also homogeneous. Tadi saya ada mention sampel terima 2 kg cili, kubis 12 unit kubis. So kita kena buat sampling, homogenization until dapat 5 gram untuk buat testing saja. So this part is very crucial. These are the equipment that used in the lab for sample preparation. Yeah. Yang kelapa sawit susah sikit, semua orang takut. Ya, sebab daripada tandang kita kena ambil buah dia, susah. So test method, as we know, workable test method, that means validated test method. So PB required method to be validated. This is the typical test method for pesticide. So it will involve sample preparation, as I mentioned just now, solvent extraction using a suitable solvent. Bila extract, kita akan buat clean up, and then baru detect by instrument. Yeah. So personnel must be competent. So dia kena tahu the knowledge or characteristic and also the test method. Facility penting because we are handling trace level analysis, so we need to prevent contamination. Because in ALS kita ada buat formulation, kita pun ada buat residue. So kita tak mau formulation punya glassware atau apa contaminate residue. So facility penting. Equipment, these are the sensitive equipment to be used for pesticide. LC-MS-MS, GC-MS-MS. HPLC, UV tak mungkin akan achieve sensitivity that required by the regulation. So just now I mentioned on maximum residue limit. The definition in the highest level of pesticide residue that is legally tolerated in or on food when pesticides are applied correctly in accordance to GLP. So MRL guidelines you can get from Malaysia Food Act, Codex, Japanese, and also EU. Example of the MRL taken from Codex and also Malaysian Food Act. Definition of residue. Some pesticides also required to determine metabolite. If the metabolite is as hazard as the parent ion, so kena determine metabolite. And for example, glufosinate actually is reported as the sum of the parent AI and also metabolite. So pesticide analysis is very challenging, and QC need to be carried out by the lab to eliminate any false positive or false negative reporting. So in the lab, we will perform procedural blend, sample blend, and to ensure everything is not contaminated, and also perform spiking recovery. Also, confirmation of analyte. When you use mass spectrometer, the MRM setting actually is already very specific. So on top of this can also use to confirm the analytical piece. So I believe this will be my last slide. Oh no, report, yes, still have report. So report, this one, this slide is more on the typical information required by the study report, except the additional dates on application, harvesting, storage, transportation is the critical. And then the other data they require for the field trials actually are quite similar as I mentioned just now in study plan. Because for GLP, the main customer actually is regulatory authorities for pesticide PB. So we submit the report, study report to PB. So PB must make, I think, valid regulatory decision on the report that they receive. So the report prepared need to have all the required information and also user-friendly and also not confusing. Ini PB suka. Kalau dia baca, dia confused, susah nak buat judgement. Not even PB, saya rasa CMA pun perlu report yang user-friendly dan senang tengok. Residue typically, it will take 5 months, depends on the crop. And take-home message will be my last slide. GLP field study is tedious, laborious task. However, the report generated from GLP study is less worried because of sole accountability on study director, quality assurance, test facility management, given a comprehensive responsible to ensure validity of the test method. And also it's less headache due to the easier technical evaluation based on systematic planning, conduct of study, and reporting. And last, less conflict due to harmonized test guideline. Tadi saya dah mention OECD, so it's a harmonized guidelines. Reconstructible, reliable studies, traceable, retrievable to origin data to the point of its origins, mutual recognition of study data across international frontiers. So, thank you. That's all of my presentation.
Thank you very much to Mr. Tim Chin Min for the insightful and practical presentation on field trial studies under GLP from planning to reporting. Field trial studies are an important yet highly specialized area within GLP and opportunities to hear directly from the test facilities about the practical aspects of planning, conducting, documenting, and reporting these studies are indeed quite limited. We truly appreciate our speaker for sharing this valuable experience and practical insight with us today. I believe this session has provided our participants with a better understanding of the expectation and challenges involved in conducting field trial studies in compliance with GLP principle. Once again, thank you very much for the excellent presentation and for sharing your valuable expertise with us. Ladies and gentlemen, before we break for lunch, we would like to take this opportunity to present a token of appreciation to Encik Baharudin Basri. May I invite Encik Warukiman Wamamat, Director of Accreditation Division, GSM, to present the token of appreciation. We will now break for lunch. Please enjoy the lunch provided and take this opportunity to refresh and recharge. The program will resume at 2:30 p.m. Kindly be back in the hall by 2:20 p.m. to ensure that we can continue the program as scheduled. Thank you and enjoy your lunch.
Every standard begins unseen, in fragments, in motion. It aligns, it connects, it takes form. Then it responds to purpose, to direction. Refined, tested, strengthened over time. Shaped with precision. Until it becomes complete. Perfected to lead. A mark of trust, a symbol of excellence. Your strategic partner in quality assurance and compliance.
Angkas Certification Sdn Berhad kita diperpadankan pada 7 Ogos 2018 dan mula beroperasi secara penuh pada 1 Oktober 2018. Kita telah mendapat akreditasi pada 3 Oktober 2019. Kita telah mempunyai 225 skop untuk akreditasi badan persejalan. Di samping itu juga pada 24 Februari 2025 kita juga telah mendapat akreditasi untuk badan pengujian cabaran dalaman. Kita nak menyediakan keseluruhan sistem yang perlu disemak oleh Jabatan Standard untuk melayakkan kita mendapat akreditasi. Kita ada dua aspek yang penting. Pertama, penyediaan dokumen dan juga kedua adalah kompetensi kaki tangan kita. Akreditasi merupakan satu CJ yang penting bagi badan persejalan terutamanya Angkas. Kerana pertama, tanpa akreditasi sudah pasti kita tidak dapat diterima oleh badan-badan regulator, badan-badan kerajaan. Yang kedua, melalui akreditasi ini juga sebagai satu bukti bahawa Angkas mempunyai sistem yang mantap, mematuhi standard akreditasi, dan juga secara tidak langsung ia membuktikan Angkas telah mematuhi kesemua keperluan-keperluan akreditasi dan ia telah memberi kepercayaan kepada pelanggan kita dan juga regulator untuk menerima CJ Angkas dan juga perkhidmatan daripada Angkas Certification. Dalam kita menyediakan sistem, kita telah membuat satu sistem yang baik bermula dengan visi, misi dan quality objektif, dan juga kita mempunyai standard operasi SOP yang tertentu yang semuanya telah disemak oleh Jabatan Standard sebelum mereka memberi akreditasi kepada Angkas. Setelah kita menjalankan SOP mengikut standard persejalan itu, maka proses kita akan menjadi lebih mudah, lebih cepat, dan seperti mana yang saya sebutkan bahawa kaki tangan kita adalah kompeten dan mereka lebih berpengalaman mengetahui kerana semua telah diletakkan di dalam SOP. Dan sebab itulah kita berlaku pertambahan, alhamdulillah pertambahan klien kita begitu membanggakan. Sekarang ini pun kita telah mempunyai hampir 800 CJ yang telah kita keluarkan yang merangkumi barangan air, barangan construction dan sebagainya. Success story yang saya amat bangga adalah kita telah menjadi satu badan persejalan yang diiktiraf di Malaysia oleh regulator-regulator yang berada di Malaysia, dan juga kita menjadi rujukan kepada regulator-regulator, dan juga kita menjadi pilihan kepada klien-klien yang berjenama besar di dunia. Kalau dalam bidang sanitary, kita ada jenama-jenama yang popular lah, Toto, Roka, Grohe, Hans Grohe, IKEA, semua itu merupakan klien kita. Saya rasa itulah success story kita, dan saya selalu memesankan kepada kaki tangan saya supaya terus menerus memastikan kita berada di landasan yang betul dan pastikan mereka ini betul-betul memahami proses kita dan melakukan operasi dengan penuh integriti, transparent, kompeten. Kita juga menjadi rujukan dalam apabila mereka menghadapi masalah teknikal. Jadi itulah saya rasa success story yang boleh dibanggakan. Angkas menjadi rujukan kepada semua, termasuk jenama-jenama besar dunia. Ada standard untung dan Angkas pilihan semua.
Ini merupakan Institut Pendidikan Perhutanan. Kami menjalankan penyelidikan berasaskan hutan, sama ada daripada segi huluan ataupun hiliran. Apa-apa produk perhutanan juga kami ada melaksanakan ujian. Oleh itu kami mempunyai pelbagai makmal ujian yang akan menguji produk-produk yang berkaitan dengan perhutanan ini. Makluman juga, kami telah bermula seawal 1998 dengan akreditasi kepada dua makmal kami, iaitu makmal analisis kimia kayu dan juga makmal komposit kayu. Sebelum kami mendapatkan akreditasi Jabatan Sdn Malaysia ni, kami sendiri telah mempunyai makmal namun tidak ada akreditasi. Jadi ini menjadikan satu masalah kepada pelanggan kerana tidak ada kepercayaan terhadap capability makmal ujian, dan juga untuk pasaran antarabangsa memerlukan ada akreditasi daripada 17025 daripada satu badan akreditasi untuk membolehkan ekspor ke luar negara. Manakala untuk pensijilan produk telah berlaku banyak isu-isu kualiti di pasaran tempatan, di mana kita seacap kali mendengar isu-isu murid tersepit di sekolah oleh kursi yang digunakan. Kita dapat satu keperluan untuk menubuhkan satu badan pensijilan produk yang boleh mengawal isu-isu kualiti perabot di Malaysia. Dengan adanya akreditasi daripada Jabatan Sdn Malaysia ini, kami dapati keadaan sistem kerja di FRIM bertambah baik, bertambah cekap, bertambah efisien, dan kemudian daripada segi kompetensi penjawat awam sendiri nampaknya semakin meningkat, di mana kami dapat merancang dengan lebih teliti untuk latihan teknikal yang perlu dihadiri oleh penjawat awam untuk badan pensijilan produk. Walaupun kami bermula pada 2014 baru dapat akreditasi, dan kami telah mendapat kerjasama yang baik daripada Kementerian Kewangan, di mana ianya telah memasukkan keperluan untuk mendapatkan pensijilan produk daripada FRIM kepada kontrak panel berpusat perabot. Terdapat syarikat baru-baru ini dapat berjaya mengeksport ke Singapura produk yang telah mendapat pensijilan daripada kami. Daripada segi pengujian perabot, kita ada 16 makmal yang telah berjaya akreditasi setakat ini. Kami dapat mengkomersialkan lagi perkhidmatan pengujian-pengujian kami ini kepada orang luar. Jadi pelanggan kami semakin hari semakin bertambah, dan mereka banyak berpuas hatilah dengan perkhidmatan yang kami berikan. Kami juga telah mendapat pengiktirafan daripada MGTC sebagai rakan verifikasi untuk produk My Hijau. Ianya telah dianugerahkan semasa iGEM 2025 baru-baru ini. Jadi itu adalah satu lagi kejayaan yang kami peroleh setelah mendapat akreditasi daripada Jabatan Sdn Malaysia. Sebab keperluan untuk menjadi partnership kepada MGTC untuk produk My Hijau ni perlu mendapat akreditasi daripada Jabatan Sdn Malaysia. Kami juga telah berjaya menambah bilangan peralatan untuk mesin-mesin ujian. Sebagai contoh, di makmal pengujian perabot kami telah menambah 31 alat ujian, di mana ini membantu meningkatkan lagi kami punya kapasiti untuk pengujian. Sebelum ini kami hanya mampu untuk melaksanakan skop pengujian bagi standard BSN. Namun setelah itu kami telah menambah kepada skop untuk pasaran Amerika untuk standard BIFMA, di mana keperluan setiap negara berbeza. Jadi daripada 345 ujian yang kami boleh laksanakan setahun, ini telah meningkat kepada 1445 ujian setahun. Peningkat ini terlalu besar, di mana ianya melebihi daripada 300 daripada sebelum ini.
Kualiti melangkaui jabatan, ada standard untuk
standard.
Putri Sentubung, putri sejinjang, penjaga gunung negeri Sarawak. Manisnya si ada dapat dilawan, anak dan dewa turun kayangan. Putri Sentubung, putri sejinjang.
Bangsa yang sihat, aset negara, kualiti hidup dipelihara, berpasukan, berganding tangan, bertingkatkan taraf kesihatan. Secara profesional khidmat diberi, kecemerlangan matlamat kami, dimanapun jua di setiap waktu, kami sedia membantu. Kami penyayang pada semua, bagi kami tiada beza. Bertanggungjawab amalan kita, kesihatan dan maruah dijaga. Bangsa yang sihat, aset negara, kualiti hidup dipelihara, berpasukan, berganding tangan, bertingkatkan taraf kesihatan. Secara profesional khidmat diberi, kecemerlangan matlamat kami, dimanapun jua di setiap waktu.
Yo. Yo. Di balik pintu makmal ini, tekad kami menyala. Patologi kimia mengukur rahasia cerita tubuh manusia. Mikrobiologi menjejak musuh halus tanpa pernah alpa. Hematologi membaca denyut darah, tanda kehidupan nyata. Oh, setiap langkah berpandukan manual quality. Arahan kerja dijunjung tinggi, tiada ruang salah interpretasi. Tata kelola rapi, jadi budaya bukan sekadar janji. Persijilan menjadi cermin kami, buktikan integriti.
Inilah makmal kami, berdiri teguh bersama. Kepimpinan cemerlang memandu hala tuju yang sama.
Keputusan tepat, cepat, selamat itu amanah utama. Keselamatan pesakit diutamakan, itu janji kami selamanya. Patologi kimia on track, kira angka macam pro. Mikrobiologi cek, singkir musuh mikro. Hematologi steady, baca darah macam
Premier Integrated Lab atau PIL sebelum ini dikenali sebagai Pantai Premier Patologi. Kami telah beroperasi sejak Julai 1997 dan merupakan sebahagian daripada keluarga IAH Healthcare Berhad. Di PIL, kami menyediakan perkhidmatan diagnostik dan ujian makmal analitikal yang lengkap dan berkualiti untuk para pengamal perubatan termasuk Dr. Am dan juga pakar. Makmal pertama kami menerima akreditasi pada tahun 2007 iaitu Referral Core Lab. Sejak itu, rangkaian kami terus berkembang dan kini sebanyak 16 makmal PIL di Semenanjung, Malaysia dan Sabah telah berjaya mendapat akreditasi di bawah MS ISO 15189 standard. Antara skop akreditasi kami termasuk hematologi, imunohematologi, chemical patologi, microbiologi, histopatologi dan juga cytopatologi. Sebelum kami mencapai akreditasi, memang banyak cabaran yang kami hadapi. Antaranya, keputusan ujian yang kadang-kadang tidak konsisten, dokumentasi yang tidak lengkap atau sukar untuk diaudit, dan proses kerja yang belum begitu seragam. Kami sedar bahawa untuk terus relevan dan dipercayai dalam industri diagnostik, kami perlu mematuhi standard antarabangsa yang diiktiraf. Akreditasi ini membolehkan kami membina keyakinan pelanggan, memperkukuh reputasi di pasaran domestik dan antarabangsa serta memastikan mutu diagnostik pesakit sentiasa berada pada tahap tertinggi.
Dengan mematuhi MS ISO 15189, kami dapat menjamin konsistensi, ketepatan dan kebolehpercayaan keputusan ujian faktor kritikal dalam penjagaan kesihatan. Selepas akreditasi, budaya kerja kami berubah sepenuhnya. Prosedur kini lebih sistematik, audit dalaman dijalankan secara berkala dan latihan berterusan memastikan pematuhan standard. Komitmen kami terhadap kualiti diterjemahkan melalui polisi kualiti yang jelas dengan visi untuk menjadi rangkaian diagnostik paling dipercayai dan misi menyediakan perkhidmatan makmal bertaraf dunia melalui teknologi canggih dan kepakaran profesional. Hasilnya, kepercayaan pelanggan meningkat. Kami berjaya mengembangkan perkhidmatan di luar rangkaian hospital IH, memudahkan persiapan bagi akreditasi berkaitan hospital, contohnya JCI, MSPH, SCHH dan juga akreditasi makmal yang lain. Contohnya College American Pathology CAP Laboratory Accreditation, kerana dokumen yang diperlukan telah tersedia dan prosedur kerja sudah teratur serta mematuhi amalan standard kualiti. Salah satu kejayaan terbesar kami berjaya mengembangkan perkhidmatan di luar rangkaian hospital IHH. Pihak hospital memberikan kepercayaan kepada PIL untuk mengambil alih pengurusan makmal serta memajukan operasi mereka. Dengan keyakinan bahawa PIL mampu meningkatkan mutu perkhidmatan makmal dan mendorong makmal untuk memperoleh akreditasi MS ISO 15189. Kami juga berjaya mendapat pengiktirafan luar, antaranya First Antimicrobial Stewardship Center of Excellence Asia, Healthcare Asia Award 2024 dan Univance Award 2024. Selain itu, PIL juga telah berjaya meningkatkan bilangan pelanggan, menandakan perkembangan positif dan penerimaan yang semakin meluas dalam pasaran.
Every standard begins unseen, in fragments, in motion. It aligns, it connects, it takes form. Then it responds to purpose, to direction. Refined, tested, strengthened over time. Shaped with precision. it becomes complete. Perfected to lead. A mark of trust, a symbol of excellence. Your strategic partner in quality assurance and compliance.
Angkas Certification Sdn Bhd kita diperpadankan pada 7 Ogos 2018 dan mula beroperasi secara penuh pada 1 Oktober 2018. Kita telah mendapat akreditasi pada 3 Oktober 2019. Kita telah mempunyai 225 skop untuk akreditasi badan persejalan. Di samping itu juga pada 24 Februari 2025 kita juga telah mendapat akreditasi untuk badan pengujian cabaran dalaman. Kita nak menyediakan keseluruhan sistem yang perlu disemak oleh Jabatan Standard untuk melayakkan kita mendapat akreditasi. Kita ada 2 aspek yang penting. Pertama, penyediaan dokumen dan juga kedua adalah kompetensi kaki tangan kita. Akreditasi merupakan satu CJ yang penting bagi badan persejalan terutamanya Angkas. Kerana pertama, tanpa akreditasi sudah pasti kita tidak dapat diterima oleh badan-badan regulator, badan-badan kerajaan. Yang kedua, melalui akreditasi ini juga sebagai satu bukti bahawa Angkas mempunyai sistem yang mantap, mematuhi standard akreditasi, dan juga secara tidak langsung ia membuktikan Angkas telah mematuhi kesemua keperluan-keperluan akreditasi dan ia telah memberi kepercayaan kepada pelanggan kita dan juga regulator untuk menerima CJ Angkas dan juga perkhidmatan daripada Angkas Certification. Dalam kita menyediakan sistem, kita telah membuat satu sistem yang baik bermula dengan visi, misi dan quality objektif dan juga kita mempunyai standard operasi SOP yang tertentu yang semuanya telah disemak oleh Jabatan Standard sebelum mereka memberi akreditasi kepada Angkas. Setelah kita menjalankan SOP mengikut standard persejalan itu, maka proses kita akan menjadi lebih mudah, lebih cepat dan seperti mana yang saya sebutkan bahawa kaki tangan kita adalah kompeten dan mereka lebih berpengalaman mengetahui kerana semua telah diletakkan di dalam SOP dan sebab itulah kita berlaku pertambahan, alhamdulillah pertambahan klien kita begitu membanggakan. Sekarang ini pun kita telah mempunyai hampir 800 CJ yang telah kita keluarkan yang merangkumi barangan air, barangan construction dan sebagainya. Success story yang saya amat bangga adalah kita telah menjadi satu badan persejalan yang diiktiraf di Malaysia oleh regulator-regulator yang berada di Malaysia dan juga kita menjadi rujukan kepada regulator-regulator dan juga kita menjadi pilihan kepada klien-klien yang berjenama besar di dunia. Kalau dalam bidang sanitary, kita ada jenama-jenama yang popular lah, Toto, Roka, Grohe, Hans Grohe, IKEA, semua itu merupakan klien kita. Saya rasa itulah success story kita dan saya selalu memesankan kepada kaki tangan kita, dengan saya, supaya terus menerus memastikan kita berada di landasan yang betul dan pastikan mereka ini betul-betul memahami proses kita dan melakukan operasi dengan penuh integriti, transparan, kompeten. Kita juga menjadi rujukan dalam apabila mereka menghadapi masalah teknikal. Jadi itulah saya rasa success story yang boleh dibanggakan. Angkas menjadi rujukan kepada semua termasuk jenama-jenama besar dunia. Ada standard untung dan Angkas pilihan semua.
Ini merupakan Institut Pendidikan Perhutanan. Kami menjalankan penyelidikan berasaskan hutan sama ada daripada segi huluan ataupun hiliran. Apa-apa produk perhutanan juga kami ada melaksanakan ujian. Oleh itu kami mempunyai pelbagai makmal ujian yang akan menguji produk-produk yang berkaitan dengan perhutanan ini. Untuk makluman juga, kami telah bermula seawal 1998 dengan akreditasi kepada dua makmal kami, iaitu makmal analisis kimia kayu dan juga makmal komposit kayu. Sebelum kami mendapatkan akreditasi Jabatan Sdn Malaysia ni, kami sendiri telah mempunyai makmal namun tidak ada akreditasi. Jadi ini menjadikan satu masalah kepada pelanggan kerana tidak ada kepercayaan terhadap capability makmal ujian dan juga untuk pasaran antarabangsa memerlukan ada akreditasi daripada 17025 daripada satu badan akreditasi untuk membolehkan ekspor ke luar negara. Manakala untuk pensijilan produk telah berlaku banyak isu-isu kualiti di pasaran tempatan di mana kita seacap kali mendengar isu-isu murid tersepit di sekolah oleh kursi yang digunakan. Kita dapat satu keperluan untuk menubuhkan satu badan pensijilan produk yang boleh mengawal isu-isu kualiti perabot di Malaysia. Dengan adanya akreditasi daripada Jabatan Sdn Malaysia ini, kami dapati keadaan sistem kerja di FRIM bertambah baik, bertambah cekap, bertambah efisien, dan kemudian daripada segi kompetensi penjawat awam sendiri nampaknya semakin meningkat, di mana kami dapat merancang dengan lebih teliti untuk latihan teknikal yang perlu dihadiri oleh penjawat awam. Untuk badan pensijilan produk, walaupun kami bermula pada 2014 baru dapat akreditasi, dan kami telah mendapat kerjasama yang baik daripada Kementerian Kewangan, di mana ianya telah memasukkan keperluan untuk mendapatkan pensijilan produk daripada FRIM kepada kontrak panel berpusat perabot. Terdapat syarikat baru-baru ini dapat berjaya mengeksport ke Singapura produk yang telah mendapat pensijilan pada kami. Dari segi pengujian perabot, kita ada 16 makmal yang telah berjaya akreditasi setakat ini. Kami dapat mengkomersialkan lagi perkhidmatan pengujian-pengujian kami ini kepada orang luar. Jadi pelanggan kami semakin hari semakin bertambah dan mereka banyak berpuas hatilah dengan perkhidmatan yang kami berikan. Kami juga telah mendapat pengiktirafan daripada MGTC sebagai rakan verifikasi untuk produk My Hijau. Ianya telah dianugerahkan semasa iGEM 2025 baru-baru ini. Jadi itu adalah satu lagi kejayaan yang kami peroleh setelah mendapat akreditasi daripada Jabatan Sdn Malaysia, sebab keperluan untuk menjadi partnership kepada MGTC untuk produk My Hijau ni perlu mendapat akreditasi daripada Jabatan Sdn Malaysia. Kami juga telah berjaya menambah bilangan peralatan untuk mesin-mesin ujian. Sebagai contoh, di makmal pengujian perabot kami telah menambah 31 alat ujian, di mana ini membantu meningkatkan lagi kami punya kapasiti untuk pengujian. Sebelum ini kami hanya mampu untuk melaksanakan skop pengujian bagi standard BSN. Namun setelah itu kami telah menambah kepada skop untuk pasaran Amerika untuk standard BIFMA, di mana keperluan setiap negara berbeza. Jadi daripada 345 ujian yang kami boleh laksanakan setahun, ini telah meningkat kepada 1445 ujian setahun. Peningkat ini terlalu besar, di mana ianya melebihi daripada 300% daripada sebelum ini.
Kualiti melangkaui kenyataan. Ada standard untuk
ada dapat dilawan, anak dan dewa turun kayanya. Putri sentubuh, putri sejinjang. Bangsa yang sihat, aset negara, kualiti hidup dipelihara, berpasukan, berganding tangan, bertingkatkan taraf kesihatan. Secara profesional khidmat diberi, kecemerlangan matlamat kami, dimanapun jua di setiap waktu, kami sedia membantu. Kami penyayang pada semua, bagi kami tiada beza. Bertanggung jawab amalan kita, kesihatan dan maruah dijaga. Bangsa yang sihat, aset negara, kualiti hidup dipelihara, berpasukan, berganding tangan, bertingkatkan taraf kesihatan. Secara profesional khidmat diberi, kecemerlangan matlamat kami, dimanapun jua di setiap waktu.
Yo. Yo. Di balik pintu makmal ini, tekad kami menyala. Patologi kimia mengukur rahasia cerita tubuh manusia. Mikrobiologi menjejak musuh halus tanpa pernah alpa. Hematologi membaca denyut darah, tanda kehidupan nyata. Oh, setiap langkah berpandukan manual quality, arahan kerja dijunjung tinggi, tiada ruang salah interpretasi, tata kelola rapi, jadi budaya bukan sekadar janji, persijilan menjadi cermin kami, buktikan integriti.
Inilah makmal kami, berdiri teguh bersama. Kepimpinan cemerlang memandu hala tuju yang sama.
Keputusan tepat, cepat, selamat, itu amanah utama. Keselamatan pesakit diutamakan, itu janji kami selamanya. Patologi kimia on track, kira angka macam pro. Mikrobiologi cek, singkir musuh mikro. Hematologi steady, baca darah macam
Premier Integrated Lab atau PIL sebelum ini dikenali sebagai Pantai Premier Patologi. Kami telah beroperasi sejak Julai 1997 dan merupakan sebahagian daripada keluarga IHH Healthcare Berhad. Di PIL, kami menyediakan perkhidmatan diagnostik dan ujian makmal analitikal yang lengkap dan berkualiti untuk para pengamal perubatan termasuk Dr. Am dan juga pakar. Makmal pertama kami menerima akreditasi pada tahun 2007 iaitu Referral Core Lab. Sejak itu, rangkaian kami terus berkembang dan kini sebanyak 16 makmal PIL di Semenanjung Malaysia dan Sabah telah berjaya mendapat akreditasi di bawah MS ISO 15189 standard. Antara skop akreditasi kami termasuk hematologi, imunohematologi, chemical pathology, microbiologi, histopatologi dan juga cytopatologi. Sebelum kami mencapai akreditasi, memang banyak cabaran yang kami hadapi. Antaranya, keputusan ujian yang kadang-kadang tidak konsisten, dokumentasi yang tidak lengkap atau sukar untuk diaudit dan proses kerja yang belum begitu seragam. Kami sedar bahawa untuk terus relevan dan dipercayai dalam industri diagnostik, kami perlu mematuhi standard antarabangsa yang diiktiraf. Akreditasi ini membolehkan kami membina keyakinan pelanggan, memperkukuh reputasi di pasaran domestik dan antarabangsa serta memastikan mutu diagnostik pesakit sentiasa berada pada tahap tertinggi. Dengan mematuhi MS ISO 15189, kami dapat menjamin konsistensi, ketepatan dan kebolehpercayaan keputusan ujian faktor kritikal dalam penjagaan kesihatan. Selepas akreditasi, budaya kerja kami berubah sepenuhnya. Prosedur kini lebih sistematik, audit dalaman dijalankan secara berkala dan latihan berterusan memastikan pematuhan standard. Komitmen kami terhadap kualiti diterjemahkan melalui polisi kualiti yang jelas dengan visi untuk menjadi rangkaian diagnostik paling dipercayai dan misi menyediakan perkhidmatan makmal bertaraf dunia melalui teknologi canggih dan kebakaran profesional. Hasilnya, kepercayaan pelanggan meningkat. Kami berjaya mengembangkan perkhidmatan di luar rangkaian hospital IHH, memudahkan persiapan bagi akreditasi berkaitan hospital, contohnya JCI, MSPH, SCHH dan juga akreditasi makmal yang lain. Contohnya College American Pathology CAP Laboratory Accreditation kerana dokumen yang diperlukan telah tersedia dan prosedur kerja sudah teratur serta mematuhi amalan standard kualiti. Salah satu kejayaan terbesar kami berjaya mengembangkan perkhidmatan di luar rangkaian hospital IHH. Pihak hospital memberikan kepercayaan kepada PIL untuk mengambil alih pengurusan makmal serta memajukan operasi mereka. Dengan keyakinan bahawa PIL mampu meningkatkan mutu perkhidmatan makmal dan mendorong makmal untuk memperoleh akreditasi MS ISO 15189. Kami juga berjaya mendapat pengiktirafan luar antaranya First Antimicrobial Stewardship Center of Excellence Asia, Healthcare Asia Award 2024 dan Univance Award 2024. Selain itu, PIL juga telah berjaya meningkatkan bilangan pelanggan, menandakan perkembangan positif dan penerimaan yang semakin meluas dalam pasaran.
Every standard begins unseen, in fragments, in motion. It aligns, it connects, it takes form. Then it responds to purpose, to direction. Refined, tested, strengthened over time. Shaped with precision. Until it becomes complete. Perfected to lead. A mark of trust, a symbol of excellence. Your strategic partner in quality assurance and compliance.
Angkas Certification Sdn Bhd kita diperpadankan pada 7 Ogos 2018 dan mula beroperasi secara penuh pada 1 Oktober 2018. Kita telah mendapat akreditasi pada 3 Oktober 2019. Kita telah mempunyai 225 skop untuk akreditasi badan persejalan. Di samping itu juga pada 24 Februari 2025 kita juga telah mendapat akreditasi untuk badan pengujian cabaran dalaman. Kita nak menyediakan keseluruhan sistem yang perlu disemak oleh Jabatan Standard untuk melayakkan kita mendapat akreditasi. Kita ada 2 aspek yang penting. Pertama, penyediaan dokumen, dan juga kedua adalah kompetensi kaki tangan kita. Akreditasi merupakan satu CJ yang penting bagi badan persejalan, terutamanya Angkas, kerana pertama, tanpa akreditasi sudah pasti kita tidak dapat diterima oleh badan-badan regulator, badan-badan kerajaan. Yang kedua, melalui akreditasi ini juga sebagai satu bukti bahawa Angkas mempunyai sistem yang mantap, mematuhi standard akreditasi, dan juga secara tidak langsung ia membuktikan Angkas telah mematuhi kesemua keperluan-keperluan akreditasi, dan ia telah memberi kepercayaan kepada pelanggan kita dan juga regulator untuk menerima CJ Angkas dan juga perkhidmatan daripada Angkas Certification. Dalam kita menyediakan sistem, kita telah membuat satu sistem yang baik, bermula dengan visi, misi dan quality objektif, dan juga kita mempunyai standard operasi SOP yang tertentu yang semuanya telah disemak oleh Jabatan Standard sebelum mereka memberi akreditasi kepada Angkas. Setelah kita menjalankan SOP mengikut standard persejalan itu, maka proses kita akan menjadi lebih mudah, lebih cepat, dan seperti mana yang saya sebutkan bahawa kaki tangan kita adalah kompeten dan mereka lebih berpengalaman, mengetahui kerana semua telah diletakkan di dalam SOP. Dan sebab itulah kita berlaku pertambahan, alhamdulillah, pertambahan klien kita begitu membanggakan. Sekarang ini pun kita telah mempunyai hampir 800 CJ yang telah kita keluarkan, yang merangkumi barangan air, barangan construction dan sebagainya. Success story yang saya amat bangga adalah kita telah menjadi satu badan persejalan yang diiktiraf di Malaysia oleh regulator-regulator yang berada di Malaysia, dan juga kita menjadi rujukan kepada regulator-regulator, dan juga kita menjadi pilihan kepada klien-klien yang berjenama besar di dunia. Kalau dalam bidang sanitary, kita ada jenama-jenama yang popular lah, Toto, Roka, Grohe, Hans Grohe, IKEA, semua itu merupakan klien kita. Saya rasa itulah success story kita, dan saya selalu memesankan kepada kaki tangan kita, dengan saya, supaya terus menerus memastikan kita berada di landasan yang betul, dan pastikan mereka ini betul-betul memahami proses kita dan melakukan operasi dengan penuh integriti, transparan, kompeten. Kita juga menjadi rujukan dalam apabila mereka menghadapi masalah teknikal. Jadi itulah saya rasa success story yang boleh dibanggakan. ANKAS menjadi rujukan kepada semua, termasuk jenama-jenama besar dunia. Ada standard untung dan ANKAS pilihan semua.
Ini merupakan Institut Pendidikan Perhutanan. Kami menjalankan penyelidikan berasaskan hutan, sama ada daripada segi huluan ataupun hiliran. Apa-apa produk perhutanan juga kami ada melaksanakan ujian. Oleh itu kami mempunyai pelbagai makmal ujian yang akan menguji produk-produk yang berkaitan dengan perhutanan ini. Makluman juga, kami telah bermula seawal 1998 dengan akreditasi kepada dua makmal kami, iaitu makmal analisis kimia kayu dan juga makmal komposit kayu. Sebelum kami mendapatkan akreditasi Jabatan Sdn Malaysia ni, kami sendiri telah mempunyai makmal, namun tidak ada akreditasi. Jadi ini menjadikan satu masalah kepada pelanggan kerana tidak ada kepercayaan terhadap capability makmal ujian dan juga untuk pasaran antarabangsa memerlukan ada akreditasi daripada 17025 daripada satu badan akreditasi untuk membolehkan ekspor ke luar negara. Manakala untuk pensijilan produk telah berlaku banyak isu-isu kualiti di pasaran tempatan, di mana kita seacap kali mendengar isu-isu murid tersepit di sekolah oleh kursi yang digunakan. Kita dapat satu keperluan untuk menubuhkan satu badan pensijilan produk yang boleh mengawal isu-isu kualiti perabot di Malaysia. Dengan adanya akreditasi daripada Jabatan Sdn Malaysia ini, kami dapati keadaan sistem kerja di FRIM bertambah baik, bertambah cekap, bertambah efisien, dan kemudian daripada segi kompetensi penjawat awam sendiri nampaknya semakin meningkat, di mana kami dapat merancang dengan lebih teliti untuk latihan teknikal yang perlu dihadiri oleh penjawat awam untuk badan pensijilan produk. Walaupun kami bermula pada 2014 baru dapat akreditasi, dan kami telah mendapat kerjasama yang baik daripada Kementerian Kewangan, di mana ianya telah memasukkan keperluan untuk mendapatkan pensijilan produk daripada FRIM kepada kontrak panel berpusat perabot. Dapat syarikat baru-baru ini dapat berjaya mengeksport ke Singapura produk yang telah mendapat pensijilan pada kami. Dari segi pengujian perabot, kita ada 16 makmal yang telah berjaya akreditasi setakat ini. Kami dapat mengkomersialkan lagi perkhidmatan pengujian kami ini kepada orang luar. Jadi pelanggan kami semakin hari semakin bertambah, dan mereka banyak berpuas hatilah dengan perkhidmatan yang kami berikan. Kami juga telah mendapat pengiktirafan daripada MGTC sebagai rakan verifikasi untuk produk My Hijau. Ianya telah dianugerahkan semasa iGEM 2025 baru-baru ini. Jadi itu adalah satu lagi kejayaan yang kami peroleh setelah mendapat akreditasi daripada Jabatan Sdn Malaysia. Sebab keperluan untuk menjadi partnership kepada MGTC untuk produk My Hijau ni perlu mendapat akreditasi daripada Jabatan Sdn Malaysia. Kami juga telah berjaya menambah bilangan peralatan untuk mesin-mesin ujian. Sebagai contoh, di makmal pengujian perabot kami telah menambah 31 alat ujian, di mana ini membantu meningkatkan lagi kami punya kapasiti untuk pengujian. Sebelum ini kami hanya mampu untuk melaksanakan skop pengujian bagi standard BSN. Namun setelah itu kami telah menambah kepada skop untuk pasaran Amerika untuk standard BIFMA, di mana keperluan setiap negara berbeza. Jadi daripada 345 ujian yang kami boleh laksanakan setahun, ini telah meningkat kepada 1445 ujian setahun. Peningkat ini terlalu besar, di mana ianya melebihi daripada 300% daripada sebelum ini.
Kualiti melangkaui kenyataan, ada standard untuk
ada dapat dilawan, anak dan dewa turun gayanya. Putri sentubuh, putri sejinjang. Bangsa yang sihat, aset negara, kualiti hidup dipelihara, berpasukan, berganding tangan, bertingkatkan taraf kesihatan. Secara profesional khidmat diberi, kecemerlangan matlamat kami, dimanapun jua di setiap waktu, kami sedia membantu. Kami penyayang pada semua, bagi kami tiada beza. Bertanggung jawab amalan kita, kesihatan dan maruah dijaga. Bangsa yang sihat, aset negara, kualiti hidup dipelihara, berpasukan, berganding tangan, bertingkatkan taraf kesihatan. Secara profesional khidmat diberi, kecemerlangan matlamat kami, dimanapun jua di setiap waktu.
Yo. Yo. Di balik pintu makmal ini, dekat kami menyala. Patologi kimia mengukur rahasia cerita tubuh manusia. Mikrobiologi menjejak musuh halus tanpa pernah alpa.
So welcome back to the symposium. So first of all, I would like to say thank you to the organizer for give us the opportunity, okay, to share our journey, especially on the GLP experience. So me myself, Hasni Zawati Muhammad Dahlan. So I'm representing my Lab Director, Ketua Makmal Bioserasi iaitu Dr. Siu Iling. So, saya kuat sikit suara. Okay, Dr. Siu Iling dia mohon maaf juga because tak dapat hadir. Because actually dia memang like really looking forward to attend this symposium. So tapi dia ada komitmen yang lain lah. Okay, so if you have like any questions after this, maybe we can save some for Dr. Iling to answer later. Okay. So for today, okay, so rather than we discuss GLP from a theoretical or regulatory perspective, maybe we would like to share, let's look at what happens in a test facility when we actually receive and conduct a GLP study. So we will look at the study journey, so the people, the system involved, data integrity, the QA, the operational challenges, and as well as the financial consideration. And finally, what we have learned from our GLP journey. Okay, so saya nak pinjamlah quote daripada Puan Fariza. Puan Fariza always share with us that GLP, okay, so involve a giant lot of papers. Yes, we do agree. You see the piling of documents over there. Okay, we have the study plan, the SOPs, all the records, everything. So many records, yeah. Okay, each function got so many records, yeah. Okay, so maybe I would like to add a few more definitions. GLP also involve great lot of procedures because in each every activities we need to have a documented procedures. So like for Makmal Bioserasi, ourselves we have around 115 SOPs, active SOPs, yeah. So kita belum kira yang obsolete and whatsoever, everything. And then GLP also gather lots of people. Okay, so yeah, it's not like a one-person job or one department punya job. So every time we have the GLP studies, so kita kan ada like 4 to 5 minimum of people to work with. Okay, so we have of course the SD, the SPs, the TFM, archivists, and maybe we need like supporting staff like for IT, maintenance, and everything. Okay, so ramai-ramai lah ya kita discuss on the GLP studies. Okay, and then yeah, GLP. So ni phase whenever TFM hand over the study director appointment letter to the SD. Okay, then she wish good luck SD, please proceed. Okay, but then TFM senyum lah kan, tapi SD dah ketar-ketar tangan dia terima the appointment letter. Okay, because dia akan like hold the responsibility lah, okay, overall responsibility of the studies. Okay, and then like more practical. Okay, GLP, so get it, okay, log it and prove it. So why? Because whenever we have a GLP studies, so we need to have the study information, get the right study information, get the right test item condition, get the right people to conduct the study, okay, get the right documentation, and then log the evidence. Of course, in any activity, we need to record. Okay, everything needs to be recorded. And then proof. Proof, why? Because so we can we have the confidence in the data and can reconstruct the study in the future. Okay. So from compliance to confidence, yeah, GLP from compliance to confidence. What makes the GLP study data trustworthy? Because it consists of 3 main elements, okay, which is the quality, whether the study properly planned and conducted, and then plus the data integrity, whether we can trace where the data came from, who generated it, and how it was controlled. And last one is the confidence. Can another person reconstruct the study and understand how the conclusion was reached? So basically, GLP is ultimately about building the sufficient evidence to give our stakeholders confidence in the study data. So I would like to introduce about Makmal Bioserasi. Okay, so Makmal Bioserasi Alaf UKM is basically a testing facility where we focus on biological safety testing, okay, supporting the biocompatibility as well as the toxicity assessment, which involve the in vitro and in vivo approaches. So we support the industries, universities, consultants, and as well as the R&D teams in evaluating the safety of products and materials. So the quality managed data and report we support few stages from the product developments as well as the biological safety evaluation, the technical decision making, as well as the regulatory submission preparation. And our vision is to be one-stop center for biocompatibility and toxicity testing in Malaysia as well as in Asian region, while our mission is to deliver the quality services in biological safety assessment that conform to the international standards. So our journey started in 2003, where at that time we start with the research activities, okay, and then slowly we provide the testing services. So when providing testing services, we aware that there's a need on the quality system of a laboratory. That's why we start with the accreditation in ISO 17025, and then while progressing and involved in a preclinical studies, so we start to prepare ourselves to involve in a GLP world, say like dunia GLP lah kan. Okay, and then in 2019 we certified by Standard Malaysia, okay, for the pesticide and industrial chemicals. And then in 2021 we venture into like more test item scopes, okay, so we certified by NPRA. So for future we want to become the one-stop center. Okay. So these are the overview of our facility. So we have two main buildings. At the front two, Block A, and then at the back Block B. Where Block A we focus on research related activities. Okay, and then the Block B, the 3-story building there, we conduct the accredited study, involve GLP and as well as the ISO studies. And then we have our animal study room. Okay, the whole ground floor there. So yeah, it's at the moment it's enough for us to conduct the GLP studies. And then we have the in vitro laboratory as well as the general lab, okay, for us to prepare the samples, reagents, everything. So moving to our organization, so actually Makmal Biosarasi is under the Center for Natural and Physical Laboratory Management UKM, ALAF UKM, where our director is Associate Prof. Dr. Wan Muhammad Aizat Wan Kamarudin. And this center is directly under the Deputy Vice Chancellor of UKM, Research and Innovation, Prof. Dr. Ismanizan Ismail. Okay, and then under that we have a few Deputy Director. So we have the Prof. Dr. Zaidi. So under this strategy dan pembangunan, we have 4 alami natural labs, yeah. So we have the PPTC, the Pusat Penyelidikan. TC is Tasik Chini. We're covering the lake ecosystem research. And then we have the PPL, the Pusat Pengurusan Langkawi, covering the geoforest, geopark ecosystem research. And then we have the Bukit Fraser, PPBF, covering the tanah tinggi punya ecosystem, yeah. And then we also have the Ecomar, covering the marine ecosystem at Mersing. So that's why we call as a Makmal Alami lah. And then we also have the physical lab, which are the iCream and as well as Makmal Biosarasi. So iCream is covering like analytical and physical studies, while us, Makmal Biosarasi, covering the biological study. And then zooming down to our GLP organization chart, so we have Dr. Siu Yiling as the TFM, and me myself as the Deputy TFM. And then we have 3 main section, which are the quality assurance section, leading by Ms. Farhana. Actually, Ms. Farhana is today with me. And then we have the in vitro and in vivo units, also led by Puan Mimi. She also ada kat sini hari ni. Yeah. And of course, we need to have the archive section. Okay. So overall, we have around 14 plus 2 vets, animal vets. Okay. So these are our certificates. So we have from the MPRA, the DSM, as well as the ISO 17025. Where our area of expertise, we cover the toxicity and also mutagenicity. Okay. So this is our services. So we cover a few type of products, okay, such as the medical device, the pharmaceuticals, food and supplement, chemicals, pesticides, cosmetic, as well as scheduled waste. Okay, so these are the list of the testing that we offer. Okay, And then selain daripada lab testing, we also provide training. So like recently we have this training on OECD GLP. So we can conduct a training on quality system, okay? ISO 17025 as well. And then we also offer training on the test method and technical training, such as the animal husbandry and so on. And then we also provide the technical consultation because I think GLP facility kita tak lari lah daripada we need to advise as well for the manufacturer on what are the type of testing that they need to conduct. Okay, so yes, we can provide that. And of course, as a university-based research facility, testing facility, we also support the R&D development. So what are the benefits of GLP? What does this GLP bring to Makmal Biostrasi? So I can highlight these 6 benefits. So number 1 is the credibility. Okay, so a structured GLP system, it provides evidence. Okay, in a study that we conduct, so in control and traceable manner, which make us Makmal Biostrasi reputation for the quality and integrity. Okay, number 2 is the client or a sponsor confidence. So whenever the sponsor engage with Makmal Biostrasi, so they are not simply purchasing a test, okay? So they are placing confidence in the facility, the personnel, and also its system. And number 3, regulatory confidence and acceptance. So it supports the generation of reliable and traceable data, which helps to make them like easier process to register their products with the RA. Okay, and then number 4, the culture itself in Biostrasi, where GLP they encourage the defined responsibilities, control procedure, as well as the proper documentation, where it foster a strong culture of quality at Makmal Biostrasi. Even now, as simple as, I don't know, like activity, semua pun mereka nak record. Because dah jadi culture in Biostrasi. Even si Farhana also want to record my speech today. Okay. And then number 5 is staff competency. Yeah, of course, maintaining GLP requires a structured training, the competency assessment, and continuous development. So it enhances the skill and professionalism of our personnel. Last but not least, of course, institutional and business value. So GLP creates institutional and business value by support the regulated industry and potentially expands its testing capabilities and market reach. So basically, GLP is not simply a certificate on the wall, so it becomes part of the lab identity and operating culture. Okay, so yeah, so behind the GLP certificate, so people, apa yang mereka boleh nampak ya in a GLP facility? Yes, of course, they can see the lab, they can see the equipment, and they can see the final report, and of course, they can see the GLP certificate. However, what happens behind these certificates is an entire system, yeah, that operates and makes the quality possible. So we need to have a well-trained and competent personnel. We need to have the SOPs and procedures. We need to conduct a proper scheduling and as well as planning. So we need to have some of QA activities, yeah, some inspection and everything. And then we need to have a proper document on how to control these deviations, how to manage deviation and kappa. We need to have a procedure for data review as well as the data integrity. So we have the equipment that need to be calibrated. And then we have the management to have to discuss on the decision and oversee the overall study conduct. And of course, we need to have the documentation and records. So the certificate is visible. However, the system behind it is something that people cannot see. So GLP is not a responsibility of one person, yes, or one department. We have this GLP ecosystem, okay? So it involves like a few designation and people that are working together and create this ecosystem. So we have the sponsor who initiate the studies, and then we have the TFM to provide the facility, resources, and equipment. And then we have the study personnel who conduct the study. And of course, study director that have the overall responsibility to, of the conduct of the studies. And QA to ensure the compliance, yeah. And then sometimes maybe we need to talk with the regulatory authorities and the other stakeholders as well. So it's not responsibility of one person, it's an ecosystem of responsibilities where everyone has a defined role, and reliable data depends on the strong science, documentation, as well as the effective teamwork. Okay, so now kita pergi ke training, bahagian training. So training give people the knowledge, but training alone is not enough. Of course, kita perlu demonstrate that the trained person can actually perform the assigned task correctly. So where the competency assessment comes in. So the process is like this, where we identify the training needs, and then we provide the practical training if applicable, and then we conduct the competency assessment. So once the person is competent, so we authorize that this person can produce a data. Okay, and competency also is not a one-time exercise where it needs to be maintained, reassessed, and updated, especially when the procedure or responsibilities change. Why? Because every piece of data is generated by a person or by a system that controlled by a person. So if we want to trust the data, we first need to trust that the people generating it are competent and to do their job. So one of the most important lessons that we have learned in a successful GLP study, it begins before the day one. So planning is one of the first quality control in GLP studies. Before any study initiated, we need to consider whether the proposed test method is appropriate. Do we have a sufficient test item? Are the facilities available and suitable? Are the personnel trained and competent? Are the resources available? And then the study, is it realistic or not? And if there are any ethical or regulatory that need to be addressed, like our Bambang Bersasi conduct animal study, so of course we have to apply like for animal ethics and so on. Therefore, planning is not simply an administrative work, but failure to address this before initiation of study can create problems later. And then we have our master schedules. So we also need to look at the master schedule, particularly when we have like a few studies running at the same time. Why? Because we need to avoid any resource conflicts and also to ensure the proper sequencing and maximize the facility and personnel efficiency. So now every study starts with a plan. So this is our study plan where we record like what are we intend to do and when we want to do it. Okay, so who will do it and how we are going to perform this study. These are properly stated in our study plan. But however, what actually happened in the lab? So things can happen. Okay, so yeah, every day like conducting study, of course, sometimes that things that are not according to our plans occurred. Okay, example like this one, the animal study. Suddenly this animal like, I want to sleep like the whole day today. Okay, so not according to our plan. So what we should do? And then the test item. Sometimes the sponsor declared that this TI can dissolve in the water, but then when we do experiment and then it not dissolve. Okay, so this thing can happen. Procedure deviation. It mentioned in the study plan 5 mil from our theoretical calculation, but then in actually it's different. Okay, and then this is the most common deviation occurs to control the temperature and humidity within the specification. Okay, and even sometimes our positive control also does not outcome as per expected. So dia assume like the red color tu macam cantik kan, control, but then berasap pula kat situ. Okay, so these things can happen in the lab actually. So what we should do when things don't go according to the plan? Okay, so a GLP study does not mean that nothing unexpected can happen. So GLP provides a control framework for managing the unexpected events. So when this thing happens, so we need to understand and we need to act. Okay, so we need to record the event, assess the impact, and then plan for the immediate action as well as the preventive action, and comes to QA to check the compliance. So, and finally, we need to ensure that the raw data the final report reflects the original raw data. So the objective is to ensure the study record accurately represents what actually happens. Yeah. So this is important part of data integrity. So now comes to the QA part. Okay. So ramai orang like salah faham. Asal QA dia lah yang tanggungjawab to take care of our quality system. But actually it's a responsibility of everybody. So QA provides the independent oversight during the study, where they conduct the study base, the process base, as well as the facility base. So an important point that the QA sendiri does not own the quality. Okay, so the study, the SD is responsible for the overall conduct of the study, the SP responsible to perform the study, the TFM provide the resources, and the QA come to check independently whether the system is working properly. So you can see, yeah, the first picture, when QA comes in into the lab, so everybody run away. Why? Someone want to hide the data, someone want to check the test item, so everybody is afraid of QA. But if everybody understand everybody's semua orang punya tanggungjawab dan responsibilities, so QA is not there simply to find the mistakes. Okay, so kalau semua orang pun bekerjasama dan faham, dia akan jadi macam di next picture lah. So this our SD. And then there's our QA inspecting like in a very calm situation. Okay. Yeah. Berat kan anak tanggungjawab QA. Okay. So now we look at the data life cycle. Okay. So where does the GLP data come from? So start from the data generation, from the activity in the labs. And then we record the data. And then processing the data maybe involves some calculation and so on. And then we review the data. And then from the data transfer into the final report. And last, we need to record or archive the data. So at every stages, there is a potential risk to the data integrity. So maybe a value can be incorrectly recorded, a calculation can be wrong, data can be transcribed incorrectly, and a record can become unavailable. So how to control this? Yes, of course, we have the ALQUA principles. And hopefully these ALQUA principles is not just a training slide, yeah. So everybody knows what is ALQUA, the attributable, usable, contemporaneous, original, and accurate. Okay, so these principles help us, okay, to ensure the data integrity. However, the challenge is to apply it every day, yeah. So many data, dia mengundang lah macam more risk, ya. Bila banyak data, banyaklah risiko dia, risiko salah dan sebagainya. So these principles should be visible in our daily behavior. So now from the paper-based, the traditional paper-based, we move to digital, and of course the now AI. So if traditional paper-based, like paper records, the handwritten entries, physical signatures, and physical archive. So at Mama Biostrasi, we still practice like 90% of manual and physical records. So now moving to the paper to digitals. So for electronic system, we have to look at the other profile, risk profile. So of course, this digital and AI memudahkan or maybe increase our effectiveness in managing records and everything. However, kita introduce the system with new risks. Yeah. So we need to consider, especially on the AIs, is introduced without controls. So the key control here, we need to include the authorized access, the validated fit for purpose system, the audit trails, backup and recovery, the cybersecurity, change control, and so on. So moving from paper to the electronic system does not automatically mean that data integrity is better, but the risk profile changes. Okay. While bringing the GLP significant benefit to Makmal Biostrasi, maintaining a GLP facility also creates operational challenge. The first is the people. Okay, so we need a sufficient personnel, but more importantly, we need a competent personnel. And then we have people, then they need to go for training, competency, and so on. And then for the capacity, multiple studies may compete for the same equipment. And also like 5 studies conducted by a same personnel. Okay, and then in scheduling, so a delay in one activity can sometimes affect the other study in a master schedule. So example like kita dah estimate like animal will arrive like this date and certainly dia tak sampai, so dia berangkai lah the effects, yeah. Okay, and then the quality workload. So of course GLP studies involve bebanan kerja yang tinggi. Okay, so require a significant work, including the documentations, the inspection, the deviations, CAPA, training and inspection readiness. And last one, for the infrastructure and equipment, so must be appropriately maintained, calibrated, or qualified where applicable. So GLP is not something that we achieve once and then we forget. Another aspect that sometimes is less visible is the financial and resource requirement for maintaining the GLP system itself. So GLP requires a continuous investment across the people, infrastructure system, as well as the study operations. So we need to invest on people, to send them for training, and then facility maintenance, yes, it does involve a quite high cost, okay? And then equipment investment, so whenever to conduct study, we need the suitable equipment, and then the quality system resources. If we use the electronic system, so we need to cover the software, and software also not cheap, yeah. And then validation, data storage, and so on. And then the direct operational costs that involve like purchasing the animals, the consumable reagents, and so on. So it's not a one-time investment, and it is a continuous commitment. And however, quality has a cost, but poor quality data maybe can give us greater cost. So from a test facility perspective, so we need to balance 3 things, which are to maintain the quality, using the resources effectively, and to ensure the appropriate cost recovery. So instead of like asking us how many studies that we can conduct, maybe you can ask a better questions that how many studies that can we conduct properly and sustainably. If the study volume exceeds the capacity, well, quality can eventually be affected. And this is particularly important for us as a university-based testing facility where scientific, regulatory, and financial responsibilities need to be balanced. So I would like to share like a few take-home message from the GLP journey. So GLP itself is a system, not a certificate. And then quality starts before the study initiation, and people are just as important as the procedures. Unexpected events should be managed transparently, not hidden. Data integrity must be built into every activities, and maintaining a GLP requires continuous operational and financial commitment. Okay, so what we document today may be the evidence someone needs years from now, because we do experience like after 10 years, the customer come back to us and asking, can we, mohon dapatkan report semula. So yeah, so we can help like, not like in a short time, but a longer times. And also, we need to keep up with the evolving GLP guidance and regulatory expectations. So we have these OECD guidelines that always like updated. Selain daripada OECD guideline, we also need to adhere to the test guideline. Yeah, we have the OECD test guideline for chemicals, pharmaceuticals, and then if we conduct the medical device study, we need to keep us updated with the 10993. Okay, other than test guideline, then we need to understand as well the national requirement for registration. There's a lot of guidelines that we need to like keep updated ourselves. So from compliance to confidence, for Makmal Basrasi, the GLP journey is continuous. So we strengthen our people, system, the facilities, and the operational sustainability. So we cannot predict everything that will happen, but we can build a system that allows us to respond appropriately, document transparently. Okay, so these are the picture where we obtained the GLP certificate in 2019, and then the other picture is just recently, in 2026. So I think over the past 7 years, although the people still look the same, the shape is still look the same, okay, and they're still happy, meaning that we survived the GLP process. Yeah. I think that's all from my side. So thank you for your attention. So maybe any simple question from the floor? Okay, thank you.
Thank you very much, Puan Hasnizawati, for the informative presentation and for sharing your valuable experience with us. Ladies and gentlemen, we have now arrived at one of the key highlights of today's symposium. We are honored to have an OECD GLP expert joining us online for a series of engagement sessions. This session will provide us with an opportunity to gain international perspective and updates on GLP developments. Our first online engagement session is entitled Sharing on OECD GLP Meeting Updates in Paris. For this session, we will be viewing a pre-recorded presentation by an OECD GLP expert who will share the key updates and developments discussed during the meeting in Paris. Ms. Eva, welcome, and the floor is yours.
Hello. My name is Eva Lainala. I work at the OECD, where I'm head of the OECD's Mutual Acceptance of Data program and the Good Laboratory Practice program. So thank you very much to colleagues in Malaysia for inviting me to provide you this update on recent developments at the OECD regarding MAD and good laboratory practice, and to provide you an overview of the OECD system. So I'll go through a few slides and provide you this update. Throughout the presentation, you'll see different links to the OECD websites. All of the information that I'm presenting to you, the documents that I refer to and such are available on the OECD website. So first, a little bit about the OECD. So the OECD is an intergovernmental organization. It's made up of 38 member countries, but we also work with partner countries and also civil society, trade unions, and business associations. So the OECD works on a number of different topics to align international standards and to find solutions for a range of different social, economic, and environmental challenges. The OECD was created in 1961, and we work through a series of committees which guide our work. And in the area of chemicals, we have the Chemicals and Biotechnology Committee of the OECD. And it's within this committee that we develop and coordinate activities on chemical safety and biosafety at the international level. The program has three main objectives. One is to improve the protection of human health and the environment through improving chemical safety and biosafety, but at the same time to harmonize approaches which then make chemical control policies more transparent and efficient. And this harmonization then saves resources for government and industry. And also through this harmonization, we prevent unnecessary distortions in the trade of chemicals, chemical products, and products of modern biotechnology. So when we think about the mutual acceptance of data system, really we have two components. We have a scientific standard that is agreed upon, and then we have a quality assurance standard that is agreed upon. And when both of those are in place, you have mutual acceptance of data. So the two main pillars of MAD are good laboratory practice and its principles and test guidelines. So the principles of good laboratory practice are really a quality system for the processes and conditions under which non-clinical health and environmental chemical safety studies are conducted. Then OECD test guidelines are standard methods and approaches for chemical safety testing that have been validated and harmonized across countries. So when data is generated according to these two pillars, we have mutual acceptance of data in that data shall be accepted in other adherents for use in chemical non-clinical chemical safety evaluation. So what are the benefits of mutual acceptance of data? Well, this system supports policy harmonization and simplification because we have better alignment between countries. This enables better visibility and predictability for both countries and industry of the types of data that will be needed and also accepted. It simplifies and speeds up market access for innovative and safer products. It includes the standards for state-of-the-art science, which then enables competitiveness, and the MAD framework can also create business opportunities for laboratory facilities. When the data is generated, it saves time in its use and assessment because of the harmonization of the data that's being received. This then also enables more similar regulatory decisions to be made on chemical safety, and lends itself also to support more transparency in regulatory decisions, which is also important for public acceptance. And as I mentioned, it aids in removing non-tariff barriers to global trade of chemicals in various sectors. We have in the past quantified the benefits of the OECD program on chemicals, and the program annually saves more than 310 million euros per year for both governments and industry. This was a value that we calculated in 2019, and based on inflation, that value would already be more than 400 million euros per year. And most of this savings is due to non-repeat testing, which occurs because of the mutual acceptance of data system. So in 1997, we had an OECD Council decision on the adherence of non-members to mutual acceptance of data. So we have, as I mentioned earlier, 7 non-members who are full adherents to MAD. This included Malaysia, who became a full adherent in 2013. We also have an accession process to the United States, to the OECD, where 5 countries would become new MAD adherents if they move through the accession process. So this would bring us to around 50 countries around the world that would be adherents to MAD. A little bit about the test guideline program and some updates in this area. So what is a test guideline? A test guideline is a harmonized protocol agreed by countries for testing a chemical to determine its intrinsic properties. So we have different sections of test guidelines. There's more than 100 different test guidelines. These can range from things like physical chemical properties to effects on environmental systems, health effects, and there are also some guidelines related to pesticide residue chemicals. Just in July of 2026, a number of new or updated and corrected test guidelines were published. So 7 new test guidelines were published, 5 were updated, and 6 were corrected. So there are 3 new in vitro tests that provide mechanistic information on metabolism, genotoxicity, and endocrine disruption. There were 2 new test guidelines released that are specific to nanomaterials, looking at dustiness and solubility in environmental media. There were also 2 new test guidelines published regarding terrestrial toxicity, so toxicity to bumblebees and earthworms. And there were also a number of updates made to skin sensitization test guidelines, in which an increased number of validated options were included within these test guidelines for different methods and approaches that can be used to probe skin sensitization. We have also released just this last month a OECD Test Guide Explorer, and this is meant to help users more easily identify the test guidelines of their interest. Now that we're getting a much larger number of test guidelines, it's a Power BI-based interface where you can browse the different OECD test guidelines in this interactive interface. You can search by test guideline number or method name. You can filter by endpoints or assay types. So this dashboard should help users find access the test guidelines that they are most interested in. You can also have access to former versions of test guidelines which may have been updated over time. So you can find that on our OECD website. Switching over to the Good Laboratory Practice Program. So as those of you in the room will know, good laboratory practice is really referring to a quality system for the management control of research laboratories and organizations which are conducting non-clinical health and environmental safety studies for regulatory purposes. And GLP is in place to ensure uniformity, consistency, reliability, reproducibility, quality, and integrity of chemical tests. So the OECD principles of good laboratory practice were already established more than 40 years ago in the early 1980s. And these principles cover a wide variety of the responsibilities and requirements for test facility organization and personnel, so that different aspects are governed in facilities, such as establishing and maintaining test systems, to handling the various test substances and reference substances, having standard operating procedures, study reporting, and also archiving and retention of records and materials. As many of you know, the OECD principles of GLP apply to non-clinical safety testing in a range of different types of test items, ranging from pharmaceuticals to pesticides to industrial chemicals and many in between. And depending on the jurisdiction, these principles are applied to the non-clinical safety testing of other regulated products, such as medical devices. So how does the GLP compliance monitoring and information exchange work at the international level between MAD adherents? Well, first of all, if you think about it in the context of one country, you'll have a GLP test facility that is monitored and inspected by a national compliance monitoring program. That GLP test facility generates data for study sponsors, who can then submit that to their receiving authority, who is making regulatory decisions in the country. And if that receiving authority has any questions about the nature of the conduct and the quality of the conduct of that study in regards to good laboratory practices, they can contact their national compliance monitoring program, who can also for example, have study audits and such at a particular test facility. If we think about the exchange then between two different adherents to the mutual acceptance of data system, so not only can the study sponsor submit that data to the receiving authority in the country where it was generated, it can also be submitted to the receiving authority in other adherents that have the same data requirement, where then they would accept that data. The receiving authority in the other country, so for example, in adherent B, if they have questions about the conduct and the quality of the study, they can contact their own national compliance monitoring program, who has good contacts with their counterparts in other adherent countries, where they can have exchange on the GLP compliance of a particular test facility, or even ask that a study audit be conducted in the other country. So to help with information exchange and collaboration between MAD adherents, we have the OECD Working Party on Good Laboratory Practice, to which Malaysia is an active participant. So within this working party, we have exchange of annual overviews of inspection. We have exchange of when there has been identification of non-compliance in facilities. We also discuss quite a bit different best practices and experience related to inspection. There's also the development of guidance, both for compliance monitoring authorities, but also for facilities and implementing good laboratory practice. We also have a periodic on-site evaluation, of which is very much a peer review of each other's compliance monitoring programs. And through all these mechanisms, there's knowledge transfer and training that occurs at the same time for compliance monitoring authorities. Just to highlight our website, our public website on good laboratory practice. Here you can find the description of the program, but also all of the OECD documents related to GLP, which include different guidance documents, advisory documents, and position papers. We also have here frequently asked questions and their answers, and contact points for all of the GLP compliance monitoring programs, along with their websites. What are newer developments from the Working Party on GLP? Well, the last document that was published was now back in December 2024, which was a position paper on GLP and IT security. We have just recently finalized 2 new frequently asked questions on study report amendments and relational databases. In progress, we have a series of discussions related to GLP study activities and conduct, which are multi-site, and some of these multi-sites occur across countries, not just within a country. These have elements of these types of studies have increased, also related to remote working, but also due to the increase in multinational companies working across borders. So the Working Party on GLP is looking at development of guidance and additional frequently asked questions to address issues that arise from the conduct of such studies. I also wanted to point out to you this guidance document that was released about 1 year ago. So countries are doing their part through the mutual acceptance of data system to reduce duplication of testing and to promote mutual acceptance of data. But also there are opportunities for companies to share data to support mutual acceptance of data. So this guidance was written in close collaboration with industry, and it offers practical solutions for companies on how to share health, safety, and environmental data in ways that are fair, transparent, and efficient. It also addresses questions of ownership, cost, and consistency when sharing of data between companies. It builds on the OECD's recommendation of the Council concerning access and protection of proprietary rights to non-clinical health, safety, and environmental data and information on chemicals and its associated best practice guide. And overall, it also aims to reduce administrative burden, enhance regulatory harmonization, and strengthen trust around stakeholders. So I encourage also companies to consider the utility of this document to also promote sharing of data and reducing duplication of testing. So that is the end of my presentation. I thank you very much for your attention. Again, here is our website on the work of mutual acceptance of data. We also have the websites on good laboratory practice and as well as test guidelines. Here you have also my email address, and I've also provided you here the email address of my colleague, Anne Gourmelon, who is the head of OECD's test guideline program. So I wish you a good continued workshop and conference, and thank you very much for this opportunity to provide an update from the OECD.
Following the presentation, any question related to the session will be addressed by Puan Fariza, our Malaysian GLP expert who attend the OECD GLP meeting in Paris. Should you have any question, please feel free to raise them.
Ada dah. Online.
Online. Oh, okay.
Ada saya.
Okay, baik. I'm trying the best to I'm trying to ask the question on behalf of the audience. Okay. Actually, one question raised in the morning. There was a concern raised by our colleague from this Jabatan Pertanian regarding whether the conformity, the QA statement, whether the QA statement should have only the signing of the QA or should also have the signing of sponsor. So far, we have not come across, to my knowledge, looking into the OECD documents, the QA statement is exclusively for the QA. So the question is, if the sponsor, we need something from the sponsor. I think it's very valid for the RA. They need some confirmation from the sponsor that they are accountable and have read the content of the study report, for example. So what will be the means, example, the norm that been practiced among the OECD members that is acceptable, at the same time does not overlap with the OECD, what has been mandated in terms of the QA statement exclusively for the QA. So how to manage this scenario to show that the report already been looked at by the sponsor before submitting to RA, at the same time not to create extra requirement that the QA statement must be signed, because suddenly we are debating from the original document. So maybe I seek some advice regarding the signing of where should the sponsor sign, whether on the final report or somewhere. So that is some idea, some guide we seek from Puan Fariza, and also for fellow other, we have also from MPRA experience on this. So maybe we need some idea on that. Thank you very much.
Thank you, Doctor. Terima kasih, Doctor. Thank you for the question. Yeah, I think this is very, very sebab many lab or test facilities asking, do we actually need, or why do we do not need the signature, all right, of a sponsor in the final report? Okay, so first, before we go further, document number 1 has mentioned, in the section 1, one para saying that the the required of assigning the final report is depends on the requirement of regulatory authority. Okay, so at the moment we have pesticide board and we have NPRA. If both of these RA says that yes, each of the final report has to be endorsed by sponsor, then we have to do so. Okay, but at the moment we don't have such requirement yet from PB. Untuk NPRA also no. Alright, at the moment no, not yet in Malaysia. But kalau doktor tanya overseas, there are regulators overseas that needs the reports to be viewed or endorsed by the sponsor. But again here, what is the role of the sponsor? OECD has come up with a document number 21 or 20. That's related to sponsor responsibility. So sponsor have to understand their role in GLP so that they do not actually conflict or act as impartial when they review the documents or the final report. Because we have seen some sponsor that do not really understand their role and is taking challenges to the test facility when they review the study plan or the final report. Okay, so that would be my answer based on doctor's question. So please refer back to document number 1. Role of sponsor in signing your final report or your study plan. And secondly, please make sure also we understand what is your role as sponsor. Okay, so I think 21 right now, 21 untuk sponsor. Okay, am I okay, Mari? Did I question well? My good friend.
Agree.
Thank you. So can we proceed? Okay, doctor. All right. Can we proceed? Okay.
Ladies and gentlemen, we shall now continue with our second online engagement session with our OECD GLP expert. The topic for this session is sharing on data integrity, computerized system, and artificial intelligence. This is certainly a highly relevant topic as laboratories continue to embrace digitalization and emerging technologies. Ladies and gentlemen, please welcome our OECD GLP expert from Poland, Mr. Marek Ubraniak.
Hey. Good afternoon. My name is Marek Ubraniak. I hope that you hear me well and everything is okay. On the YouTube, I see it's all right, so it should be okay. In the case of any problem, just please tell me. I will try to repeat or do something to improve it. So one more time, good afternoon. My name is Marek Ubraniak. I work for Bureau for Chemical Substances. I'm a GLP inspector, and as a GLP inspector, I work already something around 11 or 12 years. I'm a Polish representative at the OECD group, EU group, and EFSA, and some other things. I should start from say thank you for inviting me to that very interesting meeting, symposium. I started not from the beginning because it was too early for me, but I saw already several presentations. It was very interesting, especially the presentation about the field phase studies, which we have a lot in Poland, and I see that similar procedures at the field in Malaysia and in Poland. So that was very interesting for me. Okay, back to the presentation. I focus on two documents, advisory documents, document number 17 and document number 22. That's both of that documents. They are very important. They are critical for GLP, and that's a lot of the information. So half of the hour is not enough to say everything about them. So I choose by myself just the things which I interesting from my perspective, and the things which many times make some problem in Poland. I try to give you some information about Polish experience in that two advisory documents, introduce them. So I show you some deviation which we observe at the inspection. Maybe that will be more interesting. It's better to see how the mistakes of or other people to don't do it by yourself. And just a few words about the Polish monitoring program. In Polish monitoring program, we have now 49 test facility, including 5 facility from the China. We have only 1 monitoring program in Poland, which cover all of the areas. We conduct inspection every 2 years. So each of the facility inspected every 2 years. Here you have our webpage, our national law, register, and current certificate. They look like that. In general, that's all about the Polish. Maybe I add, because that will maybe interesting for you, about the this 49 facility in Poland. We do not have some huge test CRO like this international big one. We have several international CROs, but they are not that huge in Poland. So most of the facility are like, they are the medium. The biggest one is something like 1,000 people involved in the studies, in GLP studies. So as I mentioned, I would like to say you about these two advisory documents. So about document number 17 and document number 22. They are maybe not very new documents, but like I told you, they are, from my perspective, they are critical. They saying about the data, about the raw data, how to collect data, what to do with the data, how to store the data, and all the requirements for GLP. Still, the most important document is document number 1, and that is we treat it in Poland as a law. These two documents, they are only, or maybe not the only, but they are advisory documents. So the group, OECD group, decide that we need something like clarification for document number 1, and that documents should help you and should help us to conduct the inspection, and for you should help to conduct the studies. That's very good. I think that I have quite good documents, and I would try to briefly show you some information about that document and about both of this data integrity and computerized system. I would like to highlight that is a Polish perspective. So still the Malaysian perspective is at the inspection can be a little bit different. At the OECD group, we try to get one point of view. We try to speak in one language, but still, it's like Fariza said previous. Each country have their own monitoring program, and we have some small differences. And so still, this what is important for you is a Malaysian requirements. That's a Polish point of view. And so first, what I would like to speak, and from what I would like to start, and where we can start, is at data. So what is a data? According to document number 22, data is a quantitative or qualitative facts, figures, and statistics collect for reference or analysis. So all the data which you generate in your facility will be treated as a data, and all of that data which will be used for a GLP study must be treated in the same way. The data include all original records and verified copies, or original records included raw data and the metadata, and all subsequent transformation that are generated are record at the time of the GLP activity, and allow complete reconstruct and evaluation of the GLP activity. We as an inspector, I inspect usually retrospectively. I inspect retrospectively studies, some just few study at the inspection which we can observe at the moment, but usually we check the study retrospectively. So that's the reason why the data is so important. And to confirm that the study was performed properly and so on, for all of that, we need proper storage of the data, conducting of the data, retention of the data, and archiving of the data. So that all the presentation will be about the data, more or less. And how data can be captured. Data can be captured manually, so that the writing on the paper, by automatic record. So here will be usually all of the computerized system, using a hybrid system. So the automatic, the data from the usually the hybrid means that the data which is generated at the computerized system is collected by, for example, writing the data at the paper. So many times, simple spectrophotometer or pH meter and so on, you just wrote the data from the computerized system of the paper. And the last one is other media, other means of media. Some of the facility in Poland, when they conduct the toxicological studies, sometimes is the phase of the study is about the behavior of the animals, and they make some film, some movie about how the animals, what the animals doing at the special plate, and that movie that film will be treated as a raw data for that precise study, for that precise phase. So that can be many different ways, and it will depend only from the facility and from the equipment which facility have. Second, and the principles of GLP define a raw data, metadata, and what is the raw data? Raw data, the easiest way how to explain it, and that is understandable for me, is raw data means first captured data. So is by the principles defined as all original test facility records and documentation or verified copies, therefore which are the result of the original observation and activities in a study and allow complete reconstruction and evaluation of the GLP activities. So the raw data is that information, that data which you first capture on the paper, in your computerized system. That can be the photo. It depends from which system what is the system from which you generate the data, and what is your procedure. The document number 22 explains something additional. So the raw data was quite important, especially was understandable for everyone, but the document explains something like a metadata. And the metadata are providing information used for the identification, description, and relationship of the data. And the metadata gives the information about the data. So it's a data about data. So they explain you. So I like this photo, this slide at the right side presentation. That's a photo of the cat, and that is our raw data. So the photo of the cat, and the metadata you have at the right side. And the metadata will be information about who made the photo, what is the format, what is the title of the photo, what is the place, what was the equipment, and so on and so on. So metadata gives you information about your data. And metadata is very important because without the metadata, many times you cannot describe your data. So as a metadata is treated audit trail, which answer for the question who created the data, when, how, and so on. And the metadata, what is important, and many times facility forget about that, metadata is an integral part of the data. So you cannot archive the data, the raw data, without metadata. So if you have chromatographic system, you always must remember that you must archive chromatograms with all the audit trials. Some of the facility forget about that, and many times they miss that in the validation, or later at the inspection. Sometimes it's impossible to find the audit trial or the rest of the information. And the same point of view, the same we will treat the samely each of the computerized system. Next thing is a verified copy. And and the verified copy is a faithful representation of the original at the time copy is generated, and a verified copy may be stored in a different format or document type to the original. And how you will make your verified copy must be described in your standard operation procedures. It can be made by a duplicate of the original, maybe done by photocopy, scan, picture, screenshot. It's up to you, up to the facility. But what is important, that deletion of original data is not permitted. So verified copy doesn't mean that you can remove your original data in the place and then replace it by the verified copy. Verified copy can be treated only as a support for your raw data.
Every standard.
When you like a supporting activities, for example, for archives, maybe some duplicate of original documents. I had one situation when the raw data were, when the verified copy was treated as a raw data. It was in the facility which analyzed the human blood. At the analyze, they dropped some blood on the raw data, and that was treated as a contamination. The facility said that they cannot storage anymore that document because that can be health problem for the staff. They do this photocopy of that document. In that moment, when they do that photocopy, were quality assurance, study director, and so on, and was precisely described. They have a standard operation procedure, so what to do in that situation, and still it was treated like an exclusion. They need to remove that original document because that can be harmful for the people, and they use verified copy. But only in that case, we in Poland accept that, more or less. Still, if it will be one page or something, it will be one situation, it can be acceptable, but you cannot treat the verified copy as a replacement of the original data. So only original data, only raw data is treated as this document, this data which can be used to support your report. The verified copy is only like a supportive document. Next slide is about the data structure, and that's what's still the problem in Poland. When I started my work 10 years ago or 11 years ago, computer system wasn't that popular in Poland. Now each of the facility have more and more the computer systems. They store the data in the clouds and so on. So we have two format of the data. So we have a static format and dynamic format. And from my perspective, so from the inspector perspective, more interesting is dynamic format because it sometimes happens with this data. But I will start from this static format. Static record format, such as a paper or electronic record, is one that is fixed and allows no interaction between the user and the record content. So, like you have on the slide, on the picture, you have pH meter. There is a printer, and all the data are printed, and you cannot change the data. So the same is with the writing of data on the paper. You cannot change it. But with the dynamic format, it's a little bit different situation. So records in the dynamic state are mostly electronic records that allowed for an interactive relationship between the user and the record content. So what that means, the best example for the dynamic format is chromatograms. So in the moment when the operator will change the baseline manually, then you have a dynamic format of the data. So each of the change of the baseline must be treated like a change of the raw data. And what we require from the Polish facility, they must store the original format, the chromatogram format, before the change, before the manual change, and after that manual change. They must clearly describe the procedure in study operation procedure, and they must put that information in the study report, clear information that they change the baseline manually. And additionally, we require in the system, so in the chromatographic system, that will be clearly mentioned somewhere. So something like a tick or automatically will be mentioned that that record, that baseline was changed manually. Usually, it should be done by the system. Okay, and deviation. So some example of the deviation. I told you that I prepare some information about deviation which we have in Poland. So original chromatograms, including those generated prior to manual integration, must be archived, and the final reports should also indicate whether manual integration of chromatograms was performed. So that's one thing, that is the example of the deviation which we saw in one of this facility. Second is there is no data in the scale memory, so facility use Alibi system. That's a Polish system. It's like a black box for the balance, and all the data, the facility was decided they will use that black box, and all the data, all the raw data will be in the black box. But once or several times they forget to use the black box. In the black box was, after the waiting, they need to push the button, save the data. When they forget it, they need to use the paper document. And at the inspection, we saw that several times they forget, and it was something like inconvenience between the data, so some mistakes, some differences. So in the case when the facility start to use some computer system like Alibi, they must use it all the time. They can use only the paper document in the situation of some problem. So in that case, it should be explained that was a problem with the system. This should be described, the service should be called, or they should repair it. There should be something like a kappa for that, and so on. But in that facility, they didn't have it. So we treated like a deviation with the problem to traceable of the data. Next thing is a data lifecycle. Data lifecycle is introduced in the document number 22. It was something new for me. Maybe not new for me, but document number 22 introduced several things like that. So like data lifecycle. Today in one of the presentations mentioned about the data lifecycle, and the data lifecycles line up that you must remember not only about the preparation of the data in the moment, so the raw data in the moment, but the same important is the data approval, transcription, data processing, data migration. That's critical many times. Computerized system transaction, data retention, backup, and archive. So all of that steps in the road of your data in your facility is very important. Each of them must be part of or should be part of the validation, and you must know what's happened in your data in each of that step. It should be mentioned after the archive about the retirement of the, for example, computerized system, but that will be in the different slides. But in that case, you must remember about all the life cycle with your data, what will be happen before, after, and even at the migration. In Poland, many times the problem with the facility, the facility have a problem with the migration. They many times forget to validate that steps, but that is a critical one. The next slide was already said today about the ALCOA. So ALCOA means that's an acronym. In document number 1, you don't find the ALCOA, the acronym, but all the requirements, so the attributable, legally, contemporaneous, original, accurate, and so on, all that the requirements for your raw data they are already in document number 1, but nobody used the acronym ALCOA. The first was ALCOAs, later IMA create ALCOA Plus, and the data should be complete, consistent, enduring, and available. And ALCOA is, it was created in the moment when we didn't have so many computer systems, but still the advice for the facilities in Poland, which we have usually, if you are not sure what to do with your electronic data, you should think what you will done with what you will do with your paper data. So according to Alcoa, because for the people for the facility, much easier is to understand and to compare electronic data with the paper data. They are more like they used to it, and many times they forget that the requirements, Alcoa requirements, are the same for the paper and for the electronic data. So still, if you have any doubts about how to treat your electronic data, you should think how you will manage the paper documents, and the procedure should be the same. Some deviation, the laboratory notebook, so the electronic one. I tried to say more about the electronic document, about more about the computerized system and about the electronic raw data. So the laboratory notebook from September 2020 was completed at 4:08, but the related activities were performed at 10:30. The data can be changed many times without confirmation the changes, no audit trial. So the facility create their own computerized system. They create something like a laboratory notebook. They validate it, and according to their validation, the data cannot be changed. And that was correct. It was impossible to change the data, but it was only in the case when someone put the button save. Before that, you can change the data how many times, so many times as you wish. So by the 6th hour, nobody pushed the button save, and all the phase of the study was done in the system, in the computerized system, in the laboratory notebook, was described step by step. But by the 6 hours, everyone can change everything in the raw data. So we treat that as a deviation, and the facility has got lots of the problem. That was not only that, but that's a good example for non-compliance in the study, because it was impossible to traceable when they changed any data and what did they change and so on. That was the problem with the validation, a major problem with the validation. The next example is in the test facility computer system. It is possible to entering data with the date of the previous day, so they can change the data, and it was possible to enter and change the raw data many times until the document is saved. So it's a similar situation like a previous one. So they can change several times the document until it will be saved. So that is improper and it's not allowed. And we try to in Polish facility to don't allow for something like that. That's one or two cases. So it's not very popular, but still it happened. Second thing is a risk assessment. That's another thing which was introduced by document number 22. In Poland, the facility start with the introduce risk assessment and risk management. And that's a very useful tool for some cases, from quality assurance, for a validation, for to measure the risk which can be found in the study. And risk management should be applied through the life cycle of a computerized system. So like I show you this cycle, in all of that steps, it should be introduced the risk assessment should be used or should be in charge. Risk management contains of risk identification, assessment, migration, and risk control. And risk assessment should be used to develop an adequate validation strategy to scale the validation efforts. So in the validation, one of the important part of the validation should be assessment of the risk, which so find the place, find the moments when the risk is the highest, and in that moment should be described, and it should be precisely check at the validation process. Second thing is the risk assessment of computerized systems that are used for both GLP studies and non-GLP studies should include any potential impact of non-GLP activities on GLP compliant activities. So in the facility where which conduct not only GLP study but as well non-GLP study, non-GLP activities, the risk assessment, risk management can be used for show the risk and the places where the risk can happen. In document number 22, you don't find the information how your risk assessment should look like. The document doesn't explain that you should use like this slide, which I present, these squares, green, yellow, orange, and red. It's up to you, it's up to facility, but the risk management should be introduced in the facility. Still, that is based on your knowledge, and it should be adequate to your system, your facility. In smaller facility, our advice is that the risk assessment may be maybe not that high level, because still that's the risk assessment. So if the facility is small, if they have several studies, then the risk is lower. In the bigger facility, lots of the people love the complicated studies, lots of the computerized system, then that risk is much higher. And in that case, that should be more advanced, the risk assessment involved in that facilities. Few words about the responsibilities for data generation, from generation data to the archive. So for the study director, so for the personnel which is involved in the study, so first the most important person or people, they are the study directors, and they should ensure that all raw data are fully documented and recorded, and that is a main responsibility of the study director. The computer system which are used in the study should be validated, including requirements associated with the data integrity. And the study director is responsible for the computer system which she used for the precise study. The third thing is, after completion of the study, all the data, study plan or the document should be transferred to the archive, and that is the third responsibility of the study director. For the study personnel, is responsibility for recording raw data promptly and accurately and in compliance with principle of GLP. So that means that still the study director as well, both of that position, they should be well trained. It was mentioned today that the people are one of the most important part of the GLP study, and I fully agree. In the facility where is a good staff, good trained personnel, they work much better, and the study there's much higher quality of the study, to say. The third position is a test facility management, and the TFM responsibility is for the organization and functioning of the facility. So for the creating approving the personnel, validation, equipment, and so on. Second place, quality assurance personnel. QA should conduct inspection to determine if all studies are conducted in accordance with the principles of GLP. I should maybe advise, but in Poland we advise facility that quality assurance should take, maybe it's not that's a requirement. They should take a part in the validation of computerized system. So they should, the QA, even if they are not the specialists in the IT, always they can check the URS, always they can, with the validation, always they can check validation plan reports, and they can support the IT personnel many times, or study director or personnel responsible for validation. And the last position is an archivist. Archivist is the individual responsible for the management, operation, and procedure for archiving in accordance with principles of GLP, including archiving of data physically and electronically. Very important, because still in Poland, sometimes facility they forget about the electronic archive. Maybe not the forget about the electronic archive, but they forget about indexing of the data. So the same like the paper documents, the electronic data should be indexing, should be something like a list of the documents which are archived, should be some procedure to confirm that all the data is properly sent to the archive, properly storage, and validation should cover as well back up the data from the archive, this electronic data. It should be confirmed in the validation that that the data can be opened after 1, 2, 3, or 10 years. In Poland, is required 10 years of archiving. So even after this 10 years, the data should be opened. It must be opened by the personnel or at the inspection or for the receiving authority. Here I have only one example of deviation. QA account to the analytical equipment such as HPLC, LCMS should include also audit trail function. Many times the audit trail is for the usually it should be used by the quality assurance, should be one of the steps for the inspection for the quality assurance. They should check the audit trail as well. For the inspector, we can use it. And that is a function which gives the information about QA that everything was done properly. They should have access, and the same they should be traceable. Few words about the computerized system. This is what we understand about how we understand the computerized system. So the computerized system is a function. We in GLP understand the computerized system. A computerized system is a function integrated with the computer system and performed by trained personnel. The function is controlled by the computer system. The controlling computer system is comprised of hardware and software. The controlled function is comprised of equipment to be controlled and the operating procedure performed by personnel. So general computer system means software and hardware, and it's up to the facility, up to the system, what software and what hardware you will have. That's computer system. Controlled function or process is operating procedures and people and the equipment, and all of that together, it's treated like a computerized system. So the computerized system, many times in facility, we have a problem. They don't describe properly computerized system. In the documents for the computerized system should be clearly described which software they use, what is the hardware for that precise computerized system. So should be even number of the equipment. So the computer, if it's HPLC, so it should be mentioned the pump, auto sampler, and so on and so on. Second thing is operating procedure should be described procedure for that precise equipment and additional equipment, and as well the personnel who is involved in that, like a personnel who is responsible for maintenance, validation, and so on for that equipment should be mentioned in procedure on some document in the facility. In the document, we have several, in the document number 17, because code, and 22 as well, is a mention about costs. So that's a commercial off-the-shelf product. And example of that product is, that's a usually low complex automated equipment, like a small system. So analytical equipment, like electronic pipette, balance, photometers, and so on. And the computerized system may fully or partially rely on COTS products. And COTS product may be used without modification, with limited configuration, with heavy configuration, or even customized coding. And as with any other type of the software, COTS products require appropriate validation depending on the risk and the complexity of the customization. And that is, from my perspective, very important, because that is a place where the facility forget. So in the moment, COTS means the equipment which you facility receive from the supplier with the full validation, with the description about the use of the equipment, and usually you shouldn't have possibility to change anything in that system. If the system is created just for you, if the system, and the COTS means that the products from the shelf, so product which is like this analytical equipment, like a balance, electronic balance with some advanced system. That's product which you as a user cannot change, is unchangeable. But in the moment when the COTS is changed, like you see here, customized coding, in that case, that customized coding must be validated as well and should be described that if it's additional change in the COTS, it not always mean that it's still the COTS. In that case, that product many times it will be require additional validation or partial validation. It still depends what means customized coding. And the last thing, user requirement specification should be written for any application that is based on a COTS products. So in your user requirements should be clearly described the application for which that COTS will be used. Second point is a qualification, and the formal qualification can be used for the commercial off-the-shelf systems rather than validation. So if you have just simple equipment like this electronic pipette and so on, then many times qualification is enough. Many times qualification is enough, but not always. It still depends from the facility and from the equipment. And the second thing is the facility management must decide and define criteria for when to apply computerized system validation and or qualification approach. At the next, there's a few slides I will show you how it is look like, but general, the facility will decide that you will qualify or validate the equipment. But for that, you should have some basis. Many times in Poland, as a basis, they use GAMP5. In GAMP5, you have description what that means. So when you should qualify, when you should validate the equipment, you have 5 different levels of the equipment, from less to more advanced equipment, and then on the base of that, you will decide to validate or qualify your equipment, and that should be scoring for each of the equipment, and should be the list of the equipment with the scoring. So the simplest one, more advanced, and highly advanced equipment which must be validated, and it should be clearly described. The qualification approach should be detailed in procedures. So your SOP should be clearly defined how your qualification will be look like, and the best if it will be described for each of the equipment. The next thing, which is probably the most important, is a validation. Validation is demonstration that a computerized system is suitable through its life cycle for its intended purpose, is of fundamental importance and is referred to as computerized system validation, and all computerized system used for generation data and so on, data intended for regulatory submission or support regulatory decision should be validated and operated and maintained in ways that are completed with GLP principles. We understand that any equipment, any advanced equipment which you buy in the facility must be validated, and must be all the new equipment must be validated with the different. All the new equipment and equipment used in the study, in GLP study, must be validated. So the validation of new established computer systems should be done prospectively, and retrospective validation is not allowed. The documents, the document number 1, 22, 17, they doesn't describe they describe what means the validation, but they don't describe which validation procedure you should use. This what is usually done in Poland is the system which is called V. It's according to GAMP to GAMP requirements. They many times, usually they create, the Polish facility, they create a validation plan, then the raw data for the validation, and at the end of that is a validation report. Still, they need the user requirement specification, so URS for that before the validation. But validation must cover the like I show you this life cycle of the equipment, should cover the generating data, the moment when you use the equipment to generate data, the processing data, archiving, and then the retirement of the data. So all of that should be covered by the validation. If you have something like a migration of the data, if the migration is required in your system, in your procedure, then the migration should be as well part of the validation. And on that slide, I like it because that explains something, but not too many. So that is the information. I will explain why I said like that. So from the left side, you have a qualification, and from the right side, you have a validation. So here you have a simple equipment, and the right side, you have a complex one. And somewhere between them is the line which decides should you validate or should you qualify your system. My advice is everywhere where you have some doubts, you are not sure validate or qualify, I think better is to validate the equipment. So each equipment which is changeable by you, so at least you have you can give the several different point of access. Each of that should be validated. Validation doesn't mean that you must validate all the system by yourself. You can always base on the information from the supplier, because maybe they have already some part of the validation. But in that case, it must be described properly and must be all the information about the previous validation of the equipment, and it should be confirmed. And still, validation is for you, for the facility, to be confirmed that your raw data doesn't disappear. It will be opened after the years, and at the inspection, nobody say that your data is inconsistent, because in that case, the reason of the inconsistency of the data on some problem with the data will be non-compliance for the study. So still, the validation is from my perspective, validation is for facility. You must be sure that your equipment is fit for purpose. You must confirm that in the validation. Qualification is for smaller equipment and less complicated, and without the changeable parts by the operator and so on. Change control and configuration. I don't know what is my time, but okay. In the case of, you must remember, you as a facility must remember that each change of the computerized system, which is validated, will require many times the validation. So if you change some important part of the equipment, if you change the software, that can be, in that case, maybe needed the validation. You should have the procedure, what to do in that situation. And when you will decide to validate or revalidate the equipment, or fully validate, or maybe partial validation will be enough. But still, you should have something like a change control, and in the logbook of the equipment, usually is in that place, is described each change of the configuration, control, and so on. It should be described in your document for the precise equipment. I mentioned about migration several times, but that is still critical. You must be sure that the data which is transferred from one system to another doesn't disappear, can be opened, and is unchangeable at the migration steps. And you should confirm that in your validation procedure. Next thing is electronic signature. That's at the right side, you have a Polish certificate. All the documents which we send now to our facilities from our program, now they are signed electronically. So all the documents are signed electronically. 25% of Polish facility, 25% of the Polish facility, 25% use the electronic signature, and still some of them left, but signature is much easier for some of the signing some of the data. For example, temperature data. So if they have a PDF only, they sign it by the electronic signature. It depends from the data and so on, but it's acceptable until it's recognized by local law. And if you use your internal electronic signature, which I think is not the best solution, it should be always validated. Two examples of the deviation. So one example is one of their facility create their own electronic signature and they use it for the signing SOPs, but was a small problem, they forget to validate it. And it was critical. That was in the COVID time. They use it just for signing the SOP, several of them. But still, that was Next thing is the audit trail. Audit trail is critical of inspection. The quality assurance as well. Audit trail answer for the audit trail data. What the audit trail might, you must be sure that your data will be, if you will back up the data, all the audit trail will be back. Graphic system, when it is not possible to enable the Yes? Do you hear me back? Did I back? Oh, I'm sorry. I'm really sorry. That's a problem with the internet. I'm really sorry. I back in the moment. Okay. So for chromatographic system where it is not possible to enable the audit trail function system, user form should be created. It's only acceptation in the system. We only accept that in the older version of the system, computer system. New equipment which is buy by the facility must be, they need to have audit trail on, and it cannot be turned off. About the audit trail, if we speak about the data, the life cycle of the data, that's my last two slides. So the data should be archived securely under the control of the unique archivist, and the requirements for the electronic data is the same like for the paper documents. And you must remember that for all of that is need, like I said previously, indexing of the data, and the data should be clearly described which data is archived, and should be confirmed that the proper storage of the data and retention and backup of the data is possible. That we left. Just 2 words about the cloud computing. That's just additional. I didn't have a time for that. I would like to mention only the last sentence because it's very important. So it is the ultimate responsibility of the test facility to assess and demonstrate whether the cloud-based services can ensure data quality, data integrity, and data availability, and do not affect GLP compliance of the test facility. I want to summarize that it's up to you to decide to use the computer clouds, and that is the risk of the facility. You will use the computerized, the cloud computing, and that is decision on the facility, and still the responsibility is on the place of the test facility. Which cloud do you use? That depends from you, but still you remember about the risk. That's all. Thank you very much. Sorry for the problem with the internet, and sorry that I speak so many things. I said you so many things. Thank you very much.
Okay, mic tu juga. Ada soalan ke kita? Kita ada soalan ke? Ada ke? Kalau tak ada, kita kena gerak lagi satu. Tak ada, eh? Okay, Marik, we don't have question. So thank you very much for the sharing. So it's a very, very comprehensive explanation and also slide given on a very hard topic. So thank you very much.
Thank you. Goodbye. Thank you for your
So
invitation.
Erna is in. Okay.
Thank you very much, Mr. Marik, for the insightful presentation and for sharing your perspective on data integrity and computerized system. Ladies and gentlemen, we now come to the final presentation session of today's symposium. Our final online engagement session with the OECD GLP expert will focus on sharing on management of test item. The management of test item is an important element in ensuring the integrity, traceability, and reliability of GLP studies. Without further delay, let us welcome our OECD GLP expert for the final session, Dr. Erna Hissing.
Erna, you're on now.
Yes, thank you. I'm trying to see how I can share my slides.
If you have problem, then we can just assist you on that. Okay.
I think, is it okay now?
All right, it's on. It's
yes,
on
you
now.
can see the slides. Okay, thank you. Do I need to still to have presentation mode? I think so, right?
Yes. Yes, please.
Is it okay like this?
No, it's not on presentation mode.
Is it now not on presentation mode? For me, it is.
Can you click that again? The presentation mode?
No, I do escape.
Or you want us to assist you with your slides?
Yes, can you see the slides right now? This is now presentation mode for me.
Oh, yeah, it's not.
And how about this?
Zoom. Okay. Okay, we will assist you. All right, can we assist you with that?
Okay, you can
All
also,
right.
if it's better, you can also share the slides, and then I will just say continue the slides.
Okay.
Shall I stop sharing?
Yes, please. Will you able to see that? No. So kita tak nampak lah dia. Apa dia? Host kena share screen lah kan? But we cannot see her.
I can now see the slides, and I can see the room, and I can see myself.
Yeah, what we can see here is only the slide, but we could not see you.
Okay.
Okay, let us pin you. All right. Pin dia. Oh, okay. All right, give us a moment.
Yes.
Nampak dia.
Sebab di presentation dia tempat sepuluh. Kita kena bawa presentation tu one minute. Bisa saya present kat depan. Lepas tu dia harus tempat.
Still the- Can you give another try, Erna? That you share the
To
screen?
share, you mean? I can share my screen again.
Because yeah, we're not sure why we can't see you.
Alright, give me a minute, yeah.
I'm now sharing. But it's still not.
So nak suruh dia share kan kita share. Kalau dia share mungkin kita screen total freeze. Dia never bergerak. Kita share je. Kita share. Kita share. Dah?
Hang on. Dah, come on. Sekejap, you trying to figure out.
Yang tu boleh.
Shoulder?
Yang boleh. Betul. Tapi keluar kita, bukan keluar dia.
Okay. Yep.
Ada je. Please. Ke? Oh, kalau tak, ni lah kita boleh tunggu. Buat dua muka tu tak boleh. Hold on, Erna. We still troubleshoot.
Yes,
Yeah.
I'm.
Are you able?
Well, my Zoom is saying that I'm sharing my screen.
Okay.
Shall I stop sharing again?
Okay, you can share if you want.
Yeah, I'm sharing now, but you don't see it?
Tak keluar muka dia. Okay, you share. All right. Are you going to use the slide mode?
Sorry? Do you see now my slides?
He is sharing now. Okay. No. Okay. Okay, you share the slides, Erna?
Yes, I'm now sharing, but you don't see them?
We're seeing them, but it's not presentation mode.
I will once again I will do the presentation mode, which is now slideshow.
Okay. Tapi tak boleh. But it does not show presentation.
Like, see if we can get it to show on the website.
Jonah, can you move the slide?
Apa yang kita ada ni, kita dah solution dah.
Solution dah ada kita ni. Kalau dia stop sharing.
Could you stop sharing for a while?
Yes.
All right, give us another try. Ke suruh boleh minum?
You can see our screen.
You can see our screen, right?
Yes.
Okay. All right. Hold on.
berjaya, just buat minimize video ni dan korang share
Yes,
slide je bagi
itulah.
nampak semua.
Itulah saya pun rasa macam tu. Minimize. Kenapa? Share biasa tu pun. Kenapa share biasa tak jadi?
I don't know why Zoom perangai macam.
Oh, okay.
So,
So now,
solution?
what's the
so my last solution is I walk her through how to get to show on her side. Basically,
panjang lah pula cerita.
I will try to make it easy lah.
Hmm.
Okay, is that alright?
Okay.
So,
Okay, he will guide you to share the slide because we could not see the slide when you appear. All right? Okay.
Okay.
You stop sharing.
yeah, I stopped sharing. So now,
Okay. Now
I
okay.
can start.
Okay, she will guide you. All right.
Right. Hi, can you hear me?
Yes.
Okay, so what I would like you to do is share the presentation once more.
Okay, I will now press share. Yes.
Okay, all right, go ahead. Okay.
And then I will go to my presentation, to the screen, and I'll go to the PowerPoint and I'll now press share.
Right. Okay.
Mhm.
So once you shared it, you should see like there should be a green bar right above. Do you see a green bar that says you are sharing such and such on your end?
I do not have a green bar.
I have a green bar. Okay, hold on. Let me see if I can do something. All right. When you press the presentation button, what happens next?
Which presentation button?
Okay. Okay, when you go to presentation mode.
I'll go to presentation mode.
Yes,
Yes, now
yes.
it's full screen for me.
Okay, so full screen for you, but we couldn't see it, so no worries. Can you navigate to Zoom, press the share button once more, and try to find the full screen of the presentation slides?
Sorry, can you repeat that?
Okay, so go back to Zoom.
But now I'll have to go out of the. I'll stop
Because,
sharing
okay,
now. Then.
All right, so okay, go back to Zoom. Yeah, okay, so when she presses that, kalau dia tekan tu, dia akan full screen kat monitor dia. Dia takkan nampak benda lain. Tapi Zoom tak tangkap, dia tangkap tu je. It's only capturing your PowerPoint instead of the full screen.
Hmm.
Tak, memang dia share tapi tak full.
Okay. It's just that I can, yeah. Yeah, because you're only seeing the slides on your end, right?
Well, now I'm looking at your room, but it's saying that I'm sharing my screen.
Okay, hang on. Keluar dan masuk balik. Keluar masuk balik as in? She's keluar and try to masuk dulu sebentar.
Adanya dia ni apa nama dia keluar tu?
Hang on. I think I have an idea. Wait, hang on. Okay, I have a last resort. You would have to stop sharing for now.
Okay.
And we'll feature your face and do one last thing. It's a bit messy, tapi it will do. Kita buat macam ni, okay? All right, because the issue is on your end is that you only have one monitor and you couldn't switch out to show the full slides. So no worries, we'll show the slides in our way. It's a bit janky, but it'll work.
Okay.
So
But I think for you, it's more important that you see the slides than you see me.
Yes. Okay, tak apa, we will proceed. No worries, we just have to share. It's just now I have one more problem. Side by side, overlap kat side tu sekejap. Hang on. Ini kita punya. Yeah, kita punya. I'm trying to find the settings. Hang on, I'm gonna swap out display settings. Tapi dia akan tunjuk ni. Hang on. No, tak nampak dia. You will not be able to see her. No. So we have to do it this way. Do I do it this way? I can do that lah. Tapi, but this slideshow tak boleh guna. Okay. No, we will have to navigate on our end while she reads her slides. Okay, so
Yes.
you will present as usual, but we will control a copy of the slides on our end to show to the audience.
Yes.
Okay, so it should be okay from here. Yeah, so you just give us the cue when you say you want to go on to the next slide, and we'll follow your pace.
Okay, so I will not see the slides in the Zoom, but I will just go through my own presentation, right?
Correct.
Okay.
All right.
And then you see also my face and the presentation?
Hold on. Okay, so this is what you're seeing right now.
Okay. Yes.
Yeah, it's a bit all over the place, but you know, we make it work.
That's fine.
Okay. It's just that one.
Okay, I will go through my own I will go through my own presentation, and I will just let you know when to go through the next
Yeah,
slide, right?
okay. So just
That's
tinggal
fine.
aku pula macam mana nak tunjuk slides untuk kat YouTube. I need to have a. Do you have a copy of the slides? Sini. I need a copy untuk YouTube sebab kalau kita buat macam ni, I tak boleh tangkap untuk YouTube. So I have to do something on my end, tapi I just need a copy. You boleh tak WhatsApp?
WhatsApp lah, WhatsApp you.
Okay.
Dalam external tadi tu? Dalam external.
Ada tak dalam external drive tadi? Okay, here you go. Apa pasal?
Hold on for a while. They are taking coffee. So another 5 minutes while
Okay.
we set up, because we have an online also. So the participants are not able to see it at the moment. So we'll try to
Okay.
adjust, so that the online also could see the presentation.
And will you put your presentation? It's now not in presentation mode. Will you put it in presentation mode when you start?
We can't change to presentation mode. Once we change to presentation mode, we can't see you.
Okay.
Yeah. So my apology is a bit messy, but yeah, that's the best at the moment.
Yeah. And it's better for me to go through my own slides because I see that the video, the audio and the video are not synchronized for me. So that's a little bit strange. So it's better for me that I just go through my own presentation, and I will just guide you whenever you need to go to the next slide. So please let me know when I can start.
Get our coffee in. Yes, table so that we can start. All right, in 5 minutes time, kita bawa makanan dekat meja. Semua dah kesejukan. Hello? All right, you may start, Erna.
Okay, and then I will go through to my presentation, my own presentation. So I'm not seeing the Zoom mode anymore. Okay, so for the first slide, first of all, I'm very happy to be a guest speaker for your symposium. I know for you it has been a long day. This is the final presentation. For me, it's still morning. Yesterday evening, I returned from holiday, so I was already very happy that I remembered my password to log in. Still, we had some hiccups with starting this presentation, but I think we will be fine. I will try to be brief and clear because I know that you just had some coffee to be a little bit awakened again, but I know it's always quite challenging at the end of the day. So I try to be as quick, but also as clear as possible. I always like to start with a picture from windmills and tulips because most people worldwide like those pictures. So that's why I chose this one as the first slide. And my presentation will be about test item and test item characterization and its formulation, which are two different things. Please go to the next slide. So, yes, is this working like it is? So you're now on the second slide?
Yes, all good.
Yes, okay. I will continue. Okay, please let me know if there are issues. So I'm now in the second slide. So the agenda. So I will talk about the definition of both, which is both described in the principle, so in doc number 1, but also in document number 19, which is a more elaborate document on test item and test item characterization. I will talk about the types of test item that we encounter, the characterization, as I already said, the formulation of the test item, and what is our role as an inspector, what kind of things we need to consider during inspections related to test item and its formulations. I will also present some examples and make it a little bit interactive if we have time. That will only maybe a few minutes that you can discuss a little bit. And I will, as always, end with a conclusion. So now I will go to the next slide. So we have a definition as in the principles as well as in document number 19. And in the principles, it's very basic, like test item is defined as an article that is the subject of a study, which is quite simple, but it's also very important that you really need to think about what is the test item, what kind of information do I need to gather to determine the safety of a specific product, and what in the end is also used in practice. So what can I see as test item? And in document number 19, it's more elaborate definition. So this is saying the conclusion of a GLP study provides information on the properties of the test item, which allows an assessment of the risk it presents to the safety of humans, animals, or the environment. And you should always bear in mind that GLP studies are performed and in the end, they will end up at the desk from an assessor who needs to assess whether a certain product is indeed safe to provide to humans, animals, or the environment. So in the end, the assessor needs to have confidence in the information that he or she receives and that he or she needs to assess. Next slide, please. So we have different types of test item. So there are like industrial chemicals, agrochemicals, pharmaceuticals, which can be used both for human and in veterinary industry, cosmetic products. If the country where you live, if it's allowed to test those kind of products, for instance, in the Netherlands, it's not allowed to do animal testing on cosmetic products. Only you can use products which we are already tested in the past. Food and feed additives, both for human and for animal purposes, and also medical devices are also used in GLP studies. And like in Europe, this is laid down in the MDR, the medical device regulation, which is not that old. And there it's specifically described that you should do your safety studies under GLP. So you have different types of origin, like the simple chemicals, but also biological, synthetic, natural, living organism, transgenic organism, items from complex industrial or biological processes, and complex mixtures. And you can understand that all these kind of complex test items all bring their own challenges in GLP studies, and within the GLP studies, you have to deal with those challenges, which will not mean that it's impossible, but you have to really describe properly what you can test and what you cannot test based on the characteristics of the test item. Next slide, please. So characterization of a test item, this provides the evidence to support its suitability for use in GLP studies. So this means that the study director and of course also the test facility manager, he or she must be certain that the test item is really the test item that they want to test. And that sounds quite obvious, but in the end, if you have a bottle with a certain powder of tablets, you really need to know that it's really the test item that it should be, and that it's also of sufficient quality. And it's also that in the end the assessor knows that the test item which was tested is really representative for the use in practice. Next slide, please. So in the principle in 6.2, the following is described about characterization. So each test and reference item should be appropriately identified. That could be with a code or with a CAS number, with a name, or with biological parameters. For each study, the identity, including the batch number, which is really important, the purity, the composition, the concentrations. And you should bear in mind that if you talk about the concentration of a test item, it's not the formulation, because sometimes the test item can really be just a concentration, which is in the end applied to the test system. Or other characteristics to appropriately define each batch of the test or reference item should be known. And number 4, the stability of the test and reference items and the storage and test conditions should be known for all studies. And point 5, if the test item is administered or applied in the vehicle, the homogeneity, concentration, and stability, which is easily said as HCS, which I will do in the following slides. So the HCS of the test item in that vehicle should be determined. And for test items used in field studies, for example, tank mixes, these may be determined through separate laboratory experiments, because it's not practical to have that analysis done in the field itself. Next slide, please. So in document number 19, there is much more explained about management of and characterization and use of test items. In the definition of terms, it's explained that test item is defined as an article that is the subject of a study. And the conclusion of a GLP study provides information on the properties of the test item, which allows an assessment of the risk it presents to the safety of humans, animals, or the environment. And in document number 19, in characterization point 34, and this is really a repetition of what it's also said in the principle, so this part I will skip. And in number 35, it is explained no or inadequate information on the characterization of the test item constitutes a deviation from the principles of GLP. And that is really important. So the impact of the deviation has on the validity of the study data and the extent of compliance with the principle of GLP should be described by the study director in the GLP compliance statement of the final study report. So this means that if you need to exclude some information from GLP, that does not mean that in the end the whole study would not be valid, as long as you describe it properly, what it could be, what the impact could be on the study, as long as the assessor has sufficient information about the properties of the product. So deviations from principles of GLP does not invalidate a study, but it should be clear what the impact is. So this is important that this is described well in the GLP study report. Next slide, please. So what more is described in document number 19? This is about the source of the characterization of data. So at point 40, the characterization of the test item may be carried out by the sponsor, which is quite often done, a supplier, or the test facility. If characterization is performed by the sponsor or supplier, test facility management should ensure that documented procedures are in place to verify the integrity and quality of the information provided. What also is important that the characterization itself of the test item, it's not mandatory to have that work done under GLP. So the characterization is something else than the test item formulation, which also then constitutes the homogeneity, concentration, and stability. That is something which should be done under GLP, but characterization, that is not something required to do under GLP. 41 is saying, in every case, the final study report should describe who is responsible for test item characterization and who performed it. And the report may also provide other relevant information, such as the quality system under which the characterization was performed. And this is already saying that, like GLP is not mandatory. What you quite often see is that a COA, a certificate of analysis, is provided, which is, for instance, stating that the test item was manufactured in a GMP facility, which is okay for test item characterization. Next slide, please. What else is described in number 19? This is about the HES. So in point 62, as required by the principle of GLP, if the test item is administered or applied in a vehicle, the homogeneity, concentration, and stability, the HES, of the test item in that vehicle should be determined and reported. And most of the time, the determination is part of the study. For test items used in field studies, again, the tank mixes, these may be determined through separate laboratory experiments. 63.63 is saying there's an expectation that data on HCS of the test item in a vehicle are generated in compliance with the principle of GLP. So this is different than the test item characterization. The HCS is really expected to be performed under GLP. Then we have also a different situation where you use ready-to-use formulations, which will be explained on the next slide. And in point 65, it says that it's recognized that it may not always be technically possible to generate information on HUS for a test item administered or applied in a vehicle. And the lack of such data and its impact on the validity of a study should be justified in the final study report. So again, it does not mean, because sometimes it's just not possible to do these kind of tests under GLP. It does not mean that it's invalidating an experiment, but it really should be described in the report. So again, that the assessor has the correct information and do a proper assessment on the quality of the study performed. Next slide, please. So here, this is a little bit of visualization of a test item and a test item concentration, or the HCS. So you can like have a liquid in the liquid vehicle, or you can also have a test item in feed. That's quite often a little bit more complex with the manufacturing of the feed and also the analysis, but those are two examples which could be possible. And then in the end, these can be applied in different ways. So you can put them as an injection in testing animals, or you can provide it via feed to animals. And the material, which is the text in blue, the material actually applied to the test systems, that is really important. That should be analyzed in accordance with GLP. So it's really about the final product, which is applied to the test system, for which you need to know the HES. Next slide, please. So here's the information about a mixture or formulation which is supplied as a ready-to-use. So point 64 in document number 19 is saying, if the test item is supplied prepared as a mixture, as a formulation, or in a vehicle, and the data on HES is not generated in the GLP test facility, also then the impact of the validity of the study and the integrity of the test item should be assessed and made clear in the final study report. So I think that's clear, that if a test facility receives it like a formulation which is already prepared somewhere else and it's not under GLP, that also should be described in the report. Next slide, please. So our role as an inspector. So with respect to the test item, what do we need to do? So we should check that the test facility has procedures and records on the determination of the identity, the purity, the composition, stability, and also, which is quite important, prevention of cross-combination of test items. So if you go to the test item chamber where the test facility is keeping all the test items, you need to make clear that cross-contamination is not possible, so that they have a good processes to separate the different test items. There should also be procedures and records on the receipt, the handling, the sampling, the use, accountability, which is also important as an inspector, that you need to check that the amount of test item which is used in the studies, and then that they keep records of what is used, what they still have, what they destroyed, so that if you put everything together, that there is no missing test item, which, for instance, could have been used in studies which were not reported. So you should do a little bit of calculation to make sure that everything is accounted for. And also the storage of the test item, whether it's stored according the required temperatures and other requirements, it's important. We should also check that test item containers are properly labeled, which is quite obvious. And the storage conditions are appropriate to preserve the concentration, the purity, and the stability of the test items. So there should be a certificate of analysis or a comparable document, which is also describing the storage conditions where the test item should be, how the test item should be stored. Next slide, please. So with respect to the formulation, which is something else than characterization, the inspector should check that there are procedures for mixing substances to prevent errors in identification or cross-contamination. There are procedures and written records for the determination of the concentration, homogeneity, and stability of mixtures containing test items. And also the containers which are holding the mixtures or dilutions of the test items are properly labeled and stored. Next slide, please. So with respect to identify the test item, what do we look at? So we need to ask ourselves, so what is the test item? I will have some examples for that so that you can think about what is the subject of the study? What is the nature of the test item? who did supply it, in which conditions, so that both the conditions of the manufacturing, but also how was it shipped, under what conditions, do they maintain like the temperature records from the data logger, is this according to the requirements, how was it received, which documentation was provided by the supplier, how was the test item stored at receipt, during the study and at the end of the experiments, so that the date and the conditions of storage during experiments, are they consistent with the stability data? Which procedures and records are available at the test facility concerning the test item, and who is responsible for receipt, storage, release, and dispense. In the larger test facilities, you see quite often there's a specific test item office, which is responsible for the whole process. But in the end, you should always keep in mind that whenever the study director receives the test item from the office, the study director is still the final person responsible to make sure that the test item is fit for use in the study. Next slide, please. So with respect to characterization of the test item, what else should we consider during inspection? So do the expiry or retest date cover the duration of the experiments? So it should be at least cover the last administration time point. So especially for like the 9 months of the 12 months studies, you quite often see that there is some retest required, or that new batches are supplied. You really should check that the stability of the test item is covering the complete period of the study and not only the start of the study. Is the identification of the test item correctly defined in the document provided by the supplier, on the label of the tube, in the study plan, in the raw data, and in the final report? Is there a document providing the composition of the test item, including the quantification of each component? For instance, if you use mixtures of different items. Is there a certificate of analysis indicating the purity of the test item, which is very important? And also here you need to check, sometimes you need to do some corrections for the purity within the test. And you see quite often on certificate of analysis that there are different purities mentioned, determined with different methods, and you should really check that the correct method was in the end used for the calculation within the study. You should look at how the test item was administered, in the pure form or in the diluted form. And in the diluted form, you then already have like the formulation requirements. Is there an extraction or filtration step, which is really important, because you always need to know what is the test item after you have extracted a liquid or filtrated a liquid. What is the remains of the test item? So that's important to consider. Do we have analytical information on the concentration obtained of the diluted test item? So that is the formulation of the analysis, or the concentration after the extraction filtration steps. So that information should be available. And were the formulations analyzed within the period of stability of the formulation, which is of course also very important. So not only the stability of the test item itself, but also the stability of the formulation. So if you do the test and you have the formulations, but you will only test those like in 1 month, and that doesn't mean anything anymore if your formulation is only stable for 1 week, for instance. And do we have sufficient information about the homogeneity of the prepared test item in the raw data and in the final report? Next slide, please. So are potential deviations identified by the test facility and assessed? This is also quite an important part of an inspection of a study. So did the test facility identify missing key information For example, lack of information regarding the purity or absence of analysis of formulation on prepared test item. And which is also very important, if this is identified within a certain study, you should, as an inspector, also look at the broader context of this deviation, because it could mean that many other studies are affected. So you should also look at that impact of deviation. And if there is missing key information related to GLP, is this information described in the deviations from the GLP principles? And is there an impact assessment done on the validity of the data? Next slide, please. So I have 3 examples. I think the first one is quite simple, so I will not give you any time to discuss, but I will do that for the next 2 examples, so that you can, with your neighbor, maybe talk a little bit about this. So we have here, if you consider definition of the test item in document number 19, the conclusion of a GLP study provides information on the properties of the test item. So we have substance A. We have substance A administered to the test system, and the goal of the study is acute toxicity of substance A. And the test facility received A, so there's no preparation. So it's very straightforward. So what is the test item and which characterization data are expected? Next slide, please. So the answer, of course, is that in this case, A is the test item. And what we expect is that characterization data on the test item A are required. So at least the identity, including the batch number, the purity, the composition, the concentrations, if it's a liquid and not just a solid product, and the stability. So next slide, please. So we have here example 2. So we have substance A, which is diluted in 4 solutions B, and the solutions B are administered to the test system. And the goal of the study is the acute toxicity of substance A. And the test facility received A, and they prepared themselves B. So in this case, what is the test item? Which characterization data are expected? And is there any need for other data? So you can now maybe discuss for a few seconds with your neighbor to think about this. What would be the requirements and what would you expect related to the data at the test facility? So I will go to the next slide now, because I think this is also not too difficult. So if you have now the next slide, which is slide 20, some examples number 2. So again, A is the test item, because still the goal of the study is that you need to have a conclusion on the acute toxicity of A. So the test facility received A and prepared B. So what you need to have again is characterization data on A, because that is the test item. And in addition, you need, because the diluted formulations B were administered to the test system, you need data on homogeneity, stability, and concentrations of B are required. Since the test facility received A and prepared B, data are generated by the test facility and the study. And those data should be determined analytically in a GLP compliant phase. And if this is not done as such, then it's a GLP deviation. I think that's clear. Next slide, please. So this is the third and the last example. So this may be a little bit more challenging. So the objective of the study is to determine the no observed adverse effect level of substance A in a 6-month toxicity study on dog. Substance A is diluted in solution B. Then solution B is again further diluted in solution C. And those solutions are filtrated because they were not clear solutions to provide solution D. And solutions D in the end were administered, applied to the test system. So again, the question is, what is the test item in this case? Which characterization data are expected? And what other information you need to be sure that what you in the end apply to the test system is of the appropriate quality. So will give you a minute to discuss with your neighbor what kind of what information you should expect related to this study.
All right, that's the end, right?
Yeah, I will go to the next slide. If you stopped discussing, then I will go to the answer. Are the expectations in this example? So again, A is the test item because the goal of the study still is a conclusion on A. And you could regard in this example already to. Yeah, if you receive as a test facility solution D, then you can say, well, this is a ready for use formulation. But in this case, it was not ready for use. So this was all performed by the test facility itself. So the characterization data on A are required because A is the test item. Next, you need to have the data on homogeneity, stability, and concentrations of D. This can be conducted at the test facility in the study or provided by the sponsor. But once again, if this is not conducted in GLP compliance facility, then it should be stated as a GLP deviation. And the raw data of the manufacturing from A to D can be provided to increase the study director's confidence. Because here you really need to include the fact that you are filtrating the solutions from C to D, and that you also need to make sure that in the end you have the correct concentrations of the filtrated formulation which you apply to the test system. And now I come to the concluding slide. So conclusions, next slide. I think you have the next slide.
Yes, it's the conclusion.
Okay, good. So characterization of a test item is of major importance because the test item is the base of the study. A deviation on the characterization can jeopardize the GLP status of one or more studies. So this is what I explained earlier. If you see something in a test facility, you need to check whether the deviation can also affect other studies, because that is quite often the case. Information about the characterization of the test item should be described in the final report. And if not, ask for information and proofs of communication or details. What we quite often see is that they receive the test item from a sponsor, and that additional information is later provided, like via email, like storage conditions or other information. Also, that information should all be part of the study file, and you should have access to that, and that information should be there. and deviation should be described in the GLP compliance statement, and impact on the validity of the data should be assessed. Once again, a deviation from the GLP does not necessarily mean that a study is invalid, but it should be described, the impact should be described, and in the end, the assessor needs to be able to assess whether the study is of sufficient quality and whether the study, the test item in the end is safe for use in man, animals, or in the environment. So I think the next slide is only to thank you for your attention. It was a long day, and please feel free to have questions. And I will now return to the Zoom.
Right. All right. Thank you, Erna. I think we are running out of time now. So so far there is no question. So thank you very much for your time. So I'll give it to MC. MC. Yes.
Thank you very much, Dr. Erna, for the excellent presentation and for sharing your expertise and experience with us. Ladies and gentlemen, before we conclude today's program, we would like to take a moment to express our appreciation to all our distinguished speakers for their valuable contribution and insight throughout the symposium. As a token of appreciation, we would like to present a small token of appreciation to our speakers. May I invite Puan Fariza Wan Abdullah, Head of the Medical Testing and Biobanking Unit, to present the tokens of appreciation to our distinguished speakers. Okay. First, we would like to invite Puan Fadilah. First, we would like to invite Puan Fadilah Hasbullah to receive the token of appreciation. Next, we would like to invite Puan Hasnizawati Muhammad Dahlan to receive the token of appreciation. Next, we would like to invite Mr. Tim Chin Min to receive the token of appreciation. And lastly, we would like to invite Puan Sofia to come forward to receive the token of appreciation on behalf of Dr. Ahmad Razlan. Thank you to all of our speakers for your valuable contribution and for sharing your knowledge and expertise with us. Now, I would like to invite Puan Fariza to deliver the closing remarks. Puan Fariza.
So it's a long day. Okay. So tahniah bagi tepukan semua kepada yang masih lagi berada di sini ya untuk mendengar. So saya dah nampak dah di antara improvement yang kita kena buat sebabnya sesinya banyak ya tetapi tak ada masa untuk bertanya soalan ya. Sebabnya kita tak boleh buat lebih sehari. Ya, ongkosnya tak cukup. So, kita cuba masukkan apa yang patut ya dan saya cuba concludekan daripada apa yang kita plan untuk hari ini sebenarnya ya, kalau kita perasan ya we started with CMA, betul? Okay, so that is the Malaysia punya program. Then lepas tu kita go through siapa pula? RA, ya. So both RA ya, yang ada dengan kita untuk kita faham what is the GLP requirement yang ada di Malaysia, ya. And then we go down to saya buat slide sebenarnya. Okay. Alright, saya buat slide untuk conclude ya apa yang kita belajar hari ini. So in other words, GLP framework, can we have the first tekan? Tak jadi. Tak jadi lah pula. Macam mana boleh tak jadi? Ya Allah. Ada kan ujiannya. Tak apa. Saya ada slide saya kat sini. Kenapa tak jadi? Ha? Okay. Alright. Dah gerak dah. Saja dia kan. Okay. So the first part kita tengok adalah based on the CMA. Yeah. Both CMA, yes, Standards Malaysia dan juga MPRA, we have explained what are the compliance program. So dekat sini can I see hands yang compliance saja, compliance test facility. Boleh angkat tangan, compliance test facility. So kita ada 1, 2, 3, 4, 5. Okay, we have total 13. Can we see the sponsors, the industry? Pesticide dulu. Okay, so this pesticide industry, can we see the pharmaceutical? Alright, so yang angkat tangan kali yang terakhir dan yang kedua tadi adalah the industry, termasuklah potential mungkin laboratories or test facility. Okay, so I think this is what we wanted to share with you, what we have in the country. Yeah, by starting with the both CMA, yes, sebab kita ada pharmaceutical industry, kita ada pesticide juga. Yeah? So you have gained that information from both the CMA. Kemudian, seterusnya, start again. Okay, so what we were trying to tell you is that apa kita punya compliance program, okay, we inspect and we verify. Ataupun kita inspect dan kita mengesahkan ya what you have in the laboratory in order to comply with the RA, regulatory authority that has been presented by Encik Bahar tadi and also Puan Fadilah sendiri ya for the pharmaceutical product. Okay, next. So this is where they inform us and what are the requirements that will be assessed or that will be looked into during the inspection. And so this is where we also have the association. Okay, we also have the stakeholder. We have from Bayer to share with us their experience in GLP, which is worldwide, although they are not in Malaysia compliance program yet. And we also have ALS to share with us what is field study in GLP. Yeah, it's very interesting. Yeah, we have gone through two inspection of a study audit to see how the harvesting that's been done during the field studies. Yeah, and we also have sharing from Biostrasi to see how the activities that's been done. Yeah, what's the outcome? Yeah, you can see a lot of the GLP terminology from them. Yeah, with all the cartoons. So those are what we can understand, based on the implementation of biostrategy. And all this is to actually support and align with the requirement that we have under OECD. Next, the association and also the test facility, how they implement the OECD GLP. Lagi. Implementation and also generate the final report. So finally, as a conclusion, we are here based on our framework in GLP, and we do not stand alone. We have to complement each other. All right, in making sure that GLP in Malaysia will sustain. All right, we launched this program in 2010. Now is 2026. So that is berapa tahun? 16 tahun. 16 tahun, already 2 cycle of evaluation from OECD. The next one will be 2032. 32, they are coming back to Malaysia to evaluate us again. So we need this complement each other to make sure that the status of Malaysia as a full adherence to MAD will sustain and maintain. So without you, we are nobody. So let's hold hand and keep this strength and also keep this framework of GLP for Malaysia. So with that, I have to thank you everybody for your attendance. Daripada jauh dekat semua ada. And also with all the hiccups yang berlaku hari ini. So saya juga mohon maaf bagi pihak jabatan. We will try to improve this in next round. Do you think we need this for 2 days? Future. More. So kalau more kita buat online dah. Kalau fizikal, kita buat separuh hari saja. Wanita, mana dia? Up and down, nak dapat kelulusan. What happened today ni? Kali berapa bulan? Supposed to be in May. Naik turun. So barulah August sampai. So a lot of strength you guys do. So thank you very much, and I apologize for all the shortcomings. So hopefully you go back with information and also whatever that's been shared to you as a platform, and to move on with a better improvement of the system. So thank you very much.
Ladies and gentlemen, with that, we have come to the end of the National Symposium on Good Laboratory Practice GLP 2026. May I once again remind all participants to complete the necessary attendance requirement. For physical participants, please remember to scan the QR code once again to record your checkout before leaving the venue. For our online participants, please ensure that your attendance requirements are also completed accordingly. As mentioned earlier, the feedback form will be emailed to all registered participants on 19 August. Kindly complete the feedback form to fulfill the requirement for the issuance of your certificate and attendance. Please be informed that the certificate and presentation slide will be emailed to you once you have completed the feedback form. Once again, thank you for being part of this program. Thank you, and you should send your
link check out punya QR sebentar saya.
Thank you.